guys? Am I perfectly audible to all of you? Am I perfectly audible to all of you? Hi everyone, am I perfectly audible to all of you? I believe it's a yes. So hi students, do let me know in the chat section, am I audible? Am I audible? Hi, good morning, good morning, good morning everyone. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma and welcome to this amazing session where we are going to practice the questions. Yes, right? So marathons are parallelly going on. Now it's the time for the practice as well. That is why we have scheduled this session so that you people can check your preparation, right? So that you guys can check your preparation. So what's up, people? Tell me, am I audible? Am I audible? Is it a yes? So students, just imagine one thing, that if today we are going to practice, let's say 1,000 questions. So don't you think that from 1,000 questions, because they are the PYQs. So don't you think that from that 1,000 questions, very easily, at least 100 plus questions can come? Don't you think so? At least 100 plus questions I'm talking about. I'm not talking about you'll get 20 questions, 30 questions, 50 questions. No. All I'm saying is that whatever questions we are going to practice today, they are from the NCERT, right? They are from your previous year paper questions. So of course, without any doubt, minimum 100 plus question, or I would say 120 plus question, you people can expect from this particular session. Right, from this particular session. So you know that our marathons are going on, right? Tomorrow, day after tomorrow, yes, day after tomorrow, you guys are having your chemistry revision. Then after that, my dear students, I'll be taking the individual sessions for your disorders practice, for your diagrams practice, for your examples practice. Meanwhile, the short revision sessions will also be there on the channel and your physics revision will be there. So we are left with the second part of the 12th biology that I will be taking on Friday. Don't worry about that, right? Don't worry about that. Okay, so let's start the question practice today. Let's start the question practice, guys. But stay tuned to Unacademy NEET English channel. And if you are new to our channel, do not forget to subscribe our channel. And here you guys can see that few days back, I have posted this planned family session for all of you. And here there is something amazing. That is your iconic subscription as well. So just click on the link given there, right, and be the part of our batches so that we can start our NEET 2026 preparation from the day one. Okay, what say, guys? What say? So let's start the preparation from day one only. So tell me, are you ready? Yes, people, are you ready? Are you ready to practice the question, guys? Quick. And let me tell you the sequence. Firstly, I will come biology, then chemistry, then there will be the physics. And this is the pattern that you need to follow while attempting your paper as well. So when you will attempt your NEET final exam, right, make sure you start with the biology. You start with the biology, then go for chemistry. Try to save as much time as you can in the biology, then chemistry, and then go for the physics, my dear people. So this is what you need to take care, right? This is what you have to manage. Is there any issue with the audio? Is there any issue with the audio? Guys, I don't think so. But if there is, do let me know. Yeah, PDF of the session you guys are going to get. So, are you ready to start the session? Tell me, please. Are you ready to start the session? Okay, I have increased the volume as well. So, are you ready to start the session, people? So, if it is a yes, I want to see some energy in the chat section, guys. If it is a yes, I really want to see some energy in the chat section, people. Quick. Are you ready to start? Some fire emojis should be there in the chat section. And what about the likes? They are quite less. Let's make it 1,000 plus. Quick. Let's make it 1,000 plus. Quick. Let's start the session. So I really wish you all the best, right, for these questions, and I really hope you get. I'm not just hoping, I'm sure about it that you are going to get maximum questions from this particular session. So here we go with our first question. That select the incorrect statement with respect to mesosomes. So I have covered everything, 11th part, 12th part. So don't worry, the questions are mixed. So quickly answer the question, guys. Quickly answer the question. Arey, let's not talk about the duration today. Let's not talk about the duration today. Let's focus on the question practice, guys. So be quick. Answer my question, people. Answer my question quickly. Yes. That answer should be with some fire emojis. The answer should be with some fire emojis. Quick, everybody. Quick. What should be the answer? So it is biology. I'm not going to give you 60 seconds to solve a question. So let's move to the answer quickly. So they are saying you have to select the incorrect statement. Which statement we need to select? We need to select the incorrect statement. So while reading the question, right, please mark these things. Are they asking incorrect or the correct one? So they are formed by the extension of plasma membrane into the cell, which is true. Present in the form of vesicle, tubules and lamellae, which is again true. They contain photosynthetic pigment in some prokaryotes. It is incorrect, and they help in cell wall formation. So here instead of photosynthetic pigments, you know that they will have the respiratory enzymes. So of course, when it comes to the correct answer, this is going to be the third one. Let's move to the next question. Next question is saying, what would happen if in a gene encoding a polypeptide? That's a nice question. That's a nice question. From cell biology, we are jumping to the molecular basis of inheritance. What would happen if in a gene encoding a polypeptide of 50 amino acids, 25th codon is mutated to UAA? What's going to happen, guys? What is going to happen? They are saying that if in a gene encoding a polypeptide of 50 amino acid, 25th codon is mutated, what's going to happen? What's going to happen, people? Give it a try, and guys, your energy should be high. Okay, this is the sequence that you need to follow for your exam. Start with the biology, go for the chemistry, then go for the physics. Yes, I'll explain it, everybody. I want the answer. I want the answer to be there in the chat section. Quick. Quick. So it's simple. If it is UAU, if it is mutated to UAA, we know that it is a stop codon. Which codon it is? Stop codon. It will stop the translation. So 25th codon is mutated means you will get the amino acid till 24th codon, everything is fine, right? And these are the number of amino acids that you people are going to get. So a polypeptide of 24 amino acid will be formed. Let's move to the next question. Double membranous cell organelle is what? Right? Double membranous cell organelle is. People, your energy should be high. Quick. Double membranous cell organelle. Yes, everybody. So chloroplast is the one which is having two membranes. Your ribosome is a non-membrane bound organelle. Rest all are having single membrane. Next question. All of the given functions are associated with endomembrane system except all of the given functions, they are associated with endomembrane system except, guys, quick, do not underestimate the question practice at this point of time. It is very, very, very important. So do let me know, people. Do let me know what are we supposed to do here. Yes, what should be the answer here? Guys, everybody be quick. Be quick. Exactly. So endomembrane system, will they help in synthesis of glycoprotein? True. Detoxification of drug, autolysis, and so they are saying which functions are associated with endomembrane system, except, so what should not be there? What should not be there? 110% photosynthesis cannot be associated with endomembrane system. In endomembrane system, you have Golgi apparatus, endoplasmic reticulum, lysosome, and then you talk about the vacuole. So definitely they are not at all helping in the photosynthesis, but still they can help in the other activities as well. For an example, your lysosome, right? It is a suicidal bag, so that is how it will help. Your endoplasmic reticulums, your smooth endoplasmic reticulums, they will help in the detoxification as well. So when it comes to the glycoprotein synthesis, that will also be there, right? So this is what we have to remember, and this is that is how we have to cater the other options as well. Now, in the genetic code dictionary, how many codons are used to code for all 20 amino acids? How many codons do we have there? How many codons do we have there? What should be the answer here, people? Quick, what should be the answer here? You know the answer, guys. It's a very, very, very simple question. It's a very simple question. It is having the very simple answer. Let me check the answers. Very good, very good, very good. It should be D for doctor. You people are right. 61 codons will give us the 61 codons will give us what? They will give us the amino acid, because 3 codons are the stop codon. Let's move to the next part. Select the incorrect statement with respect to the plasma membrane. Select the incorrect statement with respect to the plasma membrane. Yes, people, everybody here in the class. Incorrect statement with respect to plasma membrane. With respect to plasma membrane. So they are saying it is composed of lipids that are arranged in a bilayer. True. Tail of membrane lipids are hydrophobic. They are saying tails are hydrophobic. True. Non-polar heads of membrane lipid are found in interior head. No, that is what the arrangement is. Please keep it in your mind. These are not the non-polar heads, but rather these are the polar heads, and the tail is non-polar. So sometimes, right, the tail is non-polar. So sometimes in such kind of things, we just get confused. So make sure you remember that. So of course, third will be the incorrect one. Now, the beginning of diplotin is recognized by which of the following events? Right, the beginning of diplotin is recognized by which of the following event? We are talking about the beginning of diplotin here. We are talking about the beginning of diplotin here. So diplotin, so which phases do we have? It is LZPDD, leptotene, zygotine, pachytene, diplotin, dikinesis. So when it is the diplotin, because in dikinesis, so in diplotin, what will be there? Is there the dissolution of synaptonemal complex or terminalization of chiasmata? What should be the answer? What should be the answer, people? Right, I have given you 2 options here. Right. Right, bivalent formation will not be there. That will be there in the zygotine. Right, that synapses will be there in the zygotine. This will be there in the pachytene. So, of course, this one, terminalization of chiasmata, it is there in dikinesis. It is a transition to metaphase as well. And here the answer will be dissolution of synaptonemal complex. The 4th one will be the answer. So, one question for sure will come from this topic. Next question. After a mutation at a genetic locus, the character of an organism changes due to the change in. After a mutation at a genetic locus, after a mutation at the genetic locus, the character of an organism, it changes due to what? Due to yes, due to, of course, because there is a mutation, the protein structure will change, and that is why the mutation is, that is an important MCQ. Okay, let's move to the next part, bache. No, so if you are thinking DNA replication will change, protein synthesis pattern will change, RNA transcription pattern will change. No, ultimately there will be a change in the protein structure. That will be the most appropriate answer here. Okay, let's move to the next question. Which of the following plastid is a storehouse for oil and lipids? Right, for oil and lipids, which one is the storehouse, guys? Which one is the storehouse? Which one is the storehouse here? Quick. Of course, it will be elaioplast. Eleuroplast for the protein. Amyloplast for the carbohydrates, amylase. Elaioplast will be for the oil and lipids. Am I right? Am I right? It will be for the oil and lipids. Next question. If a diploid cell has 18 chromosomes, how many chromosomes do we have in a diploid cell? 18 chromosomes. So they are saying how many bivalents are possible to form during the cell division. Yes, students, what should be the answer? What should be the answer? Again, it's a very simple question. You people can expect, right, at least from biology, you people can easily expect your 70 to 80 questions, right? It's a fact, right? Because I have covered the entire syllabus. So it is going to be very helpful for you. This session is really, literally going to be very, very, very effective for all of you. So quickly answer the question. What should be the answer here? So they are saying that if a diploid cell has 18 chromosomes, how many chromosomes do we have? 18. How many chromosomes do we have? 18. So means 2n is equals to 18. Now when it is the bivalent that has to form, bivalent means pair. So that pair has to be this. 9 chromosomes should be there. How many chromosomes should be there? Sorry, how many pairs should be there? 9 pairs should be there. Now let's move to the next question. Here we have the direction of RNA synthesis, direction of reading of template strand. So according to you, which one is the correct? These kind of questions, they are very, very, very important one, and majority of students, they make the mistake here. So quickly answer this question of mine. Quickly answer this question of mine. Yes? Quick, students. So they are saying direction. Whenever it is the direction of synthesis, right? Whenever it is the direction of synthesis, my dear students, right? Direction of synthesis will always be in 5' to 3' direction. Be it RNA or it's a DNA, it will always be this. It will always be 5' to 3', right? So of course, I can cancel this option. I can cancel this option. Now they are saying the reading of the template strand, that will be 3' to 5'. So now you know your answer. Yes, bache, what should be our answer? What should be our answer? Question number 11th. Question number 11th, what should be our answer? Of course, the first one. So direction will be this. The direction of template strand is this. Phase of cell cycle in which mitochondria and chloroplast, they get doubled. The question is, they will get doubled. They will get doubled. The doubling of the organelles. The doubling of the organelle. Mitochondria and chloroplast, they have specified people. They have specified here. Which phase should be there? Yes, please. Don't get confused. I can see majority of incorrect answers as well. I can see majority of incorrect answers as well. Not expected, guys. Not expected. Read your NCERT carefully. Read your NCERT carefully. Hanji, what answer? It is the G2 phase. It is the G2 phase. So, if you have marked the G2 phase, then you got the 4 marks. And if not, then deduct your -5. Right? Deduct -5. Now, let's move to the next part. How many kingdoms with respect to Whittaker's kingdom classification system exhibit only autotrophic and only heterotrophic modes of nutrition respectively? How many kingdoms with respect to the Whittaker's kingdom classification system, they exhibit only autotrophic and only heterotrophic mode of nutrition? The question is this is a very good question. This is a very good question. Only autotrophic and only heterotrophic mode of nutrition. It's a very good question. When it comes to the autotrophic, the one, they'll make their own food. But the point is only autotrophic. What is the point here? Only autotrophic. Only autotrophic means your plants, right? Some bacteria, they are autotrophic, but they are heterotrophic as well. Only autotrophic is plant. Now, only heterotrophic will include your fungus, achlorophyllus. They will never ever synthesize their own food. And along with that, animals are also there. So the answer should be this. The answer should be this. It should be 1 and 2. Now let's move to the next part. Commonly used vectors for human genome sequencing are. It is the human genome sequencing. It is the human genome sequences. Commonly used vectors which are going to sequence, right, which are used for human genome sequencing. Commonly used vectors for what? For human genome sequencing. Very simple and a repeated question, guys. So yes, it is BAC and YAC. BAC and YAC. So, do you know this BAC and YAC they stands for? Do you know what this BAC and YAC stands for? Anybody? It is bacterial artificial chromosome, right? It is yeast artificial chromosome. It is bacterial artificial chromosome. It is yeast artificial chromosome. Where do we discuss it? In HGP, that is your Human Genome Project. Next question. Terminalization of chiasmata occurs in. Again, from this particular part, many questions will come. Trust me on that. Many questions will come from this part. Terminalization of chiasmata is the word that we have here, right? It is the terminalization of chiasmata, everybody. What should be the answer? What should be the answer? Yes. So, don't you think it is the dikinesis of prophase 1? Dikinesis of prophase 1, definitely. Let's move to the next question. Which of the following protozoan will cause the sleeping sickness? The word is sleeping sickness here. The word is sleeping sickness here. Quick. Quick. Which of the following given protozoan cause sleeping sickness? So it will be your trypanosoma, different species of trypanosoma, right? The answer should be trypanosoma. Let's move to the next question. Which of the following is not required for any of the techniques of DNA fingerprinting available at present? Not required for any of the techniques of DNA fingerprinting. What should be the answer here? Yes? Yes? So do we discuss PCR there? Yes, ma'am. Do we discuss restriction enzyme requirement, DNA hybridization? But we will not talk about zinc finger analysis. So that is our answer. Next question. Pairs of homologous chromosomes, they separate during. The pairs of homologous chromosome, they will separate during. Pair of homologous chromosomes, they will separate during. Yes, everybody? They will separate during. So majority of the I have seen majority of the time students get confused here. The answer is anaphase 1 because it is the metaphase 1 students where chromosomes they'll form the metaphasic plate, right? Where chromosomes they'll form the metaphasic plate. So of course, indeed, without any doubt, my dear students, without any doubt, it will be the anaphase 1 in which it will be the separation of bivalent. But when you people talk about this anaphase 2, remember splitting of Just like mitosis, splitting of centromere and separation of sister chromatid will be there, right? Separation of sister chromatids will be there, right? So that is something you have to keep in your mind. Next question, from which of the given regions of root tip root hairs develop? Which of the regions of root tip root hair develops? What should be the answer, people? What should be the answer here? What should be the answer here? I know my students know the answer, right? I know my students, they know the answer. Root hair. Of course, it will be the zone of maturation, right? From zone of maturation, the root hair. The root hair zone is your zone of maturation because cells are mature, they are having the permanent function. So do we have any student right now, right, who is sure that, ma'am, I have answered everything correctly, right? Do we have any student right now who's saying that, yes, ma'am, I have answered everything correctly? Yes, please. Yes, please. So how many are there? I want to see some fire in the chat section. I want to see some fire in the chat section. If you have answered all the questions correctly, show some fire here in the chat section. Yes, everybody. Show some fire here in the chat section if you have answered everything correctly so far. Too wrong. No, 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 not acceptable. If you have made mistake in such simple questions, so you have to write down that question and you have to solve it. Because as I said, right, in biology, near about 80 or 80 plus questions you can get from this part because it is the complete syllabus test. And just think once, if you will solve 300 or 400 questions of biology, that too from the complete syllabus, don't you think 70 to 80 questions can come? Don't you think 70 to 80 questions can come? Tell me. 4 wrong. Nah, nah, nah. Nah, 4 is too much. Okay, next question. What is going to happen during decomposition? So it's a statement, long statements are there, so read it quickly. Done? Done with the answer? Done with the answer? So they are saying during decomposition is correctly described. Humification is the accumulation of dark colored substance, humus, which undergoes microbial action at a fast rate, slow. Catabolism, breakdown. Okay, for anaerobic conditions, no. Leaching, water soluble inorganic nutrients, no. Fragmentation, carried out by organisms such as earthworm. So fourth has to be the correct answer. It has to be fourth. Done. Let's move to the next question. If a myocyte, the word is myocyte here. The word is myocyte here. If the myocyte has 36 chromosomes in G2 phase, what will be the number of chromosome in each of the meiosis 1 products? It's a good question. Trust me, it's a good question. It's a very good question. It's simple as well. Myocyte. What is the meaning of myocyte? Myocyte is a diploid cell which will undergo meiosis. That's the point. Now when we talk about the cell cycle, one thing that we discuss is we have G1, we have S, we have G2 there. So whenever we talk about the cell cycle, we know that number of chromosome will always remain same. Number of chromosome will remain same. That is what we know, isn't it? That is what we know. That number of chromosome will remain same, and it is the DNA that will get doubled. So DNA will become 4C from 2C. That is something we know. So if in G2 you have 36 chromosomes, how many chromosomes do we have in G2? 36 chromosomes. So it's obvious, meiosis 1. After meiosis 1, the two cells are haploid, right? Right? And after that, this is what we are going to see. So after meiosis 1, the chromosome number will be reduced to half. So the answer here should be 1. Am I right? Am I right? Am I right? Next question. So, okay, there is a floral formula given. So if you know this, and trust me, that will be sufficient for you. If you will watch my lecture now, watch my short lecture, right? So you just need to focus on what? You just need to focus on the floral diagram, right? And the floral formula. Ye dekho, this lecture I'm talking about. New plant families. Okay, new plant families in 15 minutes where I have discussed the floral diagrams, floral formula, important diagrams, estivation. These are the things that you need to understand. Okay, so do watch this particular session. Now, what about this? What about this? Yeah, what about this? So now you will be like, ma'am, I don't know about the examples. I don't know about the examples. So according to you, this floral formula is off. This floral formula is off. Yes. See, from P word, you can judge one thing. From P word, you can judge one thing. It is an extra question. It's a different family. But somewhere, see, this is, perianth word is given here. Means your sepals, right? Your sepals and petals, they are fused. They'll form tepals. The group of tepals, it is your perianth. It is your perianth. So it is basically lily, right? In Liliaceae family. I know Liliaceae is not the part of your syllabus anymore, but it is just a kind of question, right, from the previous year question. So the answer is 1 here, right? The answer is 1 here. Okay, the answer is 1 here. You can even take the screenshot for the quick revision. Next, select the correct statement for the given diagram. Now you have a diagram based question, and that is very common. That is very, very, very common for your NEET. Yes, diagram based question is there, people. Diagram based question is there. What should be the answer here? What should be the answer here? Yes, students. Is it a type of connective tissue? True that. Here you are talking about the cartilages and bones. Remember? Cartilages and bones. Right? These are that concentric layers are lamellae. Right? So do revise these diagrams as well. Let's move to the next question. Which one is not a defining feature of life forms? You know na, from the chapter Living Word, what is living is the part of our syllabus. It is the part of our syllabus. Right? No, it simply, right? One student is answering loose and dense connective tissue. Let's forget about the loose and dense connective tissue, my dear people. Just focus on this particular part. So, which of the, like here, just focus on the diagrams here. I'm talking about this question. Focus on the diagrams here. So it is basically a type of connective tissue. Bones and cartilages you are talking about here. And now here, which of the following is not a defining feature of the life forms? Not a defining feature. It is your reproduction. Why? Because all living beings, they do not reproduce. All living beings, they do not reproduce. So because they do not reproduce, so it is not a defining feature, because defining feature is something which is present in all the living beings without any exception, plus It is not shared with non-living beings. Let's move to the next question. Which one is again a non-defining property? Non-defining property. So growth is also non-defining. So whenever we talk about such properties, remember it is your growth, it is the reproduction which are non-defining. Rest, your metabolism, cellular organization, consciousness, they are the defining one. Next question, my dear people. Dense connective tissue, they are divided into dense regular and dense irregular tissue on the basis of which feature? On the basis of which feature? So the answer here is what? The answer here is the orientation of fiber. The answer here is orientation of fiber. Next, select the odd one out with respect to the classification of wheat. With respect to the classification of wheat, right? Which one is the odd one out here? Which one is the odd one out here? Quick, guys, quick. Yes. So, do they belong to family Poaceae? Yes, class monocots, division Angiospermae, but not the order Sapindales. Here, it has to be Poales, right? Sapindales, it is the order for your mango. Just keep it in your mind. Again, it is the NCERT. Do not forget about it. Next question, select the incorrect match with respect to the taxonomic categories of the housefly. With respect to the taxonomic categories of the housefly. Quick. With respect to the taxonomic categories of the housefly. Quick, quick, bache. Yes. Quick. So what should be the answer here? What should be the answer here? It's simple. Genus Musca, order no, class here. It should be class Insecta. Family is Muscidae, phylum is Arthropoda. Next, given below are two statements. You have assertion as well as reason. You have assertion as well as reason. So what should be the answer here? What should be the answer here? Yes, please. Given below are the two statements, right? Assertion and reason. Stilt roots are a type of adventitious roots. They are formed by direct elongation of radical. One thing that you have to remember is that if you are talking about the adventitious roots, they will arise from any part other than radical. Arise from any part other than radical, right? They will arise from any part other than radical. Just keep it in your mind. So of course, if this is the criteria, right students? If this is the criteria, then what should be the correct answer? Of course, assertion is true, but the reason is not correct here. Let's move to the next question. Which of the following are the features of bacteria except? Which of the following are the features of bacteria except? Right, bache? Quick, bache, quick. Yes. So they are saying features of bacteria except. Simple. Cell wall. Is it made up of chitin? No. It is made up of murines, peptidoglycans, right? We call them as NAM, NAG. We call them as NAM, NAG. Let's move to the next part. Vascular bundles are conjoined and open in. Vascular bundles are conjoined and open in. Quick. Vascular bundles are conjoined. So conjoined, xylem and phloem is present at same radius. Open for secondary growth. So basically, if they are open for secondary, so if they are open for secondary growth, so like this, you have xylem, you have phloem, you have the vascular bundle in between. So indeed, dicot stem should be the answer. Next question, ovary is superior in the flowers of. It's a good question. It's a good question from morphology. Ovary is superior. Ovary is superior in the flowers of. Yes, people. Ovary is superior in the flowers of. Of course, superior ovary. Hypogynous flower. Superior ovary. Hypogynous flower. Exactly. It is the NCERT based question. It is the NCERT based question. The brinjal should be the answer. The brinjal should be the answer. Next, cell wall is impregnated with silica in. Again, direct MCQ from NCERT, direct PYQ it is. Guys, speed up, speed up. Cell wall is impregnated with silica in. With silica in. So of course it is the diatoms. That is why they form diatomaceous earth, right? That is why they form diatomaceous earth, right, bache? Next question. There is no secondary growth in monocot root because anatomy will give you 3 questions minimum. Anatomy will give you 3 questions minimum. So make sure you do not miss any question from this part. So they are saying there will be no secondary growth in monocot root. Because, yes, please. Yes, please. Because, right, absence of cambium, of course, right? They do not have the cambium, and it is the cambium that will help in that. Am I right here? Am I right here? Yes, everybody. Next question. Which is the correct matching from the following? Which is the correct matching from the following? So I am even picking up the questions from the anatomy, from the morphology. So as I said, right, you can very easily expect 80 questions from this particular part. Okay, so make sure you solve all the questions, people. Make sure you solve all the questions. Yes, please. Are you done with the syllabus reading? Statement reading. D for doctor. Palmately compound leaves leaflet attached to the common axis rachis. Okay. Phyllotaxy is the pattern of leaf arrangement on stem or branch. Pinnately compound leaves, leaflet attached to a common point that is at the tip of the petiole. Okay. So, venation, arrangement of veins and veinlets. So D and B to for sure is correct. For sure is correct. And when it is palmately, right? In the palmately leaves, bache, we don't talk about the rachis. It is something like that. pinnately compound leaves, they are like this, and here the midrib is known as rachis. So of course, when it comes to the correct answer, which one is the correct answer? It is the it is the fourth question. Next, now member of Basidiomycetes is member of Basidiomycetes is member of Basidiomycetes. You are talking about the fungus here. Remember, pineapple with pineapple, banana and dates. Pineapple, banana and dates. Right? So Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes. Right? So Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes. So they are asking about the Basidiomycetes, which is your club fungi. So they are asking that out of the following, which one is the member? So example based question it is, and for sure the question will come from the examples, and here the answer is Astillego. And what will they cause? They will cause smut. You remember Puccinia, that will cause rust here, right? Astillego will cause smut. These three are the members of your Deuteromycetes, right? Remaining three are the members of your Deuteromycetes. Okay. Next question. The characteristic red color in the members of Rhodophyces due to the predominance of, again a very simple question. Why red algae is red? In simple words, that is the question. Why red algae is red? So in simple words, that is our question. In simple words, that is our question, that why red algae is red. Yes, everybody? Of course, it is going to be phycoerythrin, erythrin word, erythrocyte, RBC, red. This will be blue color. This is there in the brown algae. Now in roots, the tangential as well as radial walls of barrel-shaped endodermal cells, they have deposition, and that is in the form of, again, NCERT statement, and trust me on that, 80 plus questions will be there from this part. 80 plus question. It means a lot. So be attentive here. Wherever you are making mistake, just ask me that. So it will be, answer will be Casparian strips here. Let's move to the next part, people. Feature that is not true about viroid is. Features that are not true about viroid. Viroid is the word here. Viroid is the word here. Viroid is the word here. People, quick, speed up. So, you are talking about the viroids where we just discussed infectious RNA. Am I right? No protein coat, just infectious RNA. So, they have RNA as the main genetic material. Okay. Discovered by Theo Diner. True. Capsomeres are present. No, it is there in the virus. Smaller than virus, true. So, of course, out of all, this statement is incorrect, means it is our true statement. It is our true statement. Next question. Life cycle of Polysiphonia and Cedrus respectively are. So, life cycle path, if you think it is deleted, no. Question, if you need to understand the plant kingdom, do not forget to revise the life cycle. So, Polysiphonia and Cedrus. Cedrus belongs to gymnosperms. Definitely it will have diplontic. Polysiphonia, yes. Polysiphonia. It belongs to, Polysiphonia belongs to anybody here in the class? Polysiphonia. Do you remember P2G2? Porphyr or Polysiphonia it was? Nahin? And Gelidium gracilaria, red algae. So of course, here you are talking about the haplodiplontic and the diplontic life cycle. Haplodiplontic and the diplontic life cycle. So Polysiphonia, no. Yes. So exception is also there. Yes, exception is also there. So Polysiphonia, what that Polysiphonia is? Polysiphonia belongs to, as I said, red algae. red algae. So it should be haplontic, haplodiplontic or haplontic. Yes, it is the algae. Is it an exception? Is it an exception? Is it an exception? It is a red algae, as I said. It is a red algae. P2G2 is the trick. No, algae is having haplontic life cycle, but we have the exceptions there in algae. Just your fucus, that is having diplontic life cycle. That is having diplontic life cycle. So exceptions are there. So answer here is the second one. Right? The answer here is the second one. Okay? Right? So it is related to the exceptions. Right? It is basically what? It is basically the exception. Correct? Yes? Next question. Assertion reason based question. Parenchyma and collenchyma are a type of simple permanent tissue and they are made up of more than one type of cell. Those work together as a unit. Again, a very simple question, and I really wish you'll get such type of simple assertion and reasons in paper. Yes, I really want you people to get these simple assertion and reason based question in your paper. So parenchyma and collenchyma are this type of simple permanent tissue. True. Simple permanent tissue is the one having only one type of cell, not made up of more than one type of cell. So assertion is true, but reason is incorrect. So which one should be the correct answer here? Here it should be second. Which one should be the correct answer here? Here it should be second. Is that clear? Next question. Mannitol is mainly produced by the members of. Mannitol is mainly produced by the members of. Quick people. Mannitol is mainly produced by the members of. So mannitol, brown algae. What will be there in red algae? Floridian starch. Green algae, you talk about the starch now. So, which of the following is not a feature of facilitated transport? No, this part is deleted. Okay, next question. Screen is like, no, I cannot move. Okay, so arrange the following cells in an ascending order of their size. The word is ascending order here. The word is ascending order here. Yes, please. Arrange the following cells in an ascending order. So when I'm saying ascending order, you have to write the smallest cell first, right? So don't you think when it comes to the smallest cell, we talk about, yes people, we talk about what? We talk about what? Mycoplasma, है ना? And after that, bacteria has to be there. Even if you don't know the dimensions, right, you can answer it. And ostrich will be the largest one. So this will be the sequence. So the second is the correct answer. Next, which will show the diplontic life cycle out of the following? Which one will show the diplontic life cycle? Just now I told you. Just now I told you. So here it is the fucus that will show diplontic life cycle. It is the fucus that will show diplontic life cycle. That will show diplontic life cycle. Am I correct here? Yes, people, am I correct? Guys, not getting your... Okay, next question. Which one is the activator for both Rubisco and PEPKs? Important it is. Do not underestimate this question. It is the most abundant protein of biosphere, and this enzyme you discuss in the C4 cycle. In C4 cycle, both will be discussed. Now they are asking about the activator. We generally miss such type of questions, but they are very important. So what should be the answer? What should be the answer? I know my students are smart enough. They know the answer. Yes, let me check the answer. Okay, majority of students are giving the correct answer. That is going to be your magnesium. That is going to be your magnesium. Next question. Statement 1, statement 2. The cell division is a very important process in only multicellular organism. The cell cycle is divided into 2 phases. So choose the correct answer. Choose the correct answer. Yes. Yes. Bache, is the cell division very important in only multicellular organism? Do you think so? Or what about our? Right, what about our unicellular organism? How will they divide? We cannot say that division is only there in the multicellular organism. No, it has to be in unicellular as well. So statement 1 is definitely incorrect. And yeah, it is there in 2 phases. So third should be the answer. Next question. Stilt roots are present in. Stilt roots are present in. Where do you see stilt roots? Where do you see stilt roots? A very common and the simple example, stilt roots. S for stilt, S for sugarcane. Banyan tree mein, you'll talk about the pillar roots, prop roots they are. Okay. Next question. Which of the given Monera is autotrophic and fixes nitrogen symbiotically as well as in the free state? Both the conditions are given here. Both the conditions are given here. That this one can fix the nitrogen symbiotically as well as in free state. So for such type of questions, if you don't know the answer, do not get confused, because here you just need to solve the previous year question, the MCQs, then you will be able to revise it, remember it. So of course it is Anabina. Of course it is Anabina. So symbiotically as well as in the free living state, it will be able to do that. Next question, statement based question it is again. In human respiratory system, the branching network of bronchi, bronchioles, alveoli comprises of lungs. And in humans, 2 lungs are covered by a single layered pleura. Again, a simple question. But in the paper, sometimes you just write it like this, right? You just write it like this. You don't pay attention. You don't read it properly. So, what is this? First statement, is it true? Yes. But second statement, it is false. Here, there are 2 pleural membranes. So, 1 is correct, 2 is incorrect. Next, how many plants have actinomorphic flowers? How many plants have actinomorphic radial symmetry we are talking about? Right? Okay, so actinomorphic radial symmetry we are talking about. Radial symmetry, people. Right? What are we talking? We are talking about the radial symmetry here. And I have given you a trick as well. I have given you a trick as well. So it will be mummy, daddy, chilling America. So mustard, dhatura, chili. They are actinomorphic. Mustard, dhatura, chili. So the answer should be 2. The answer should be 2. Next, select the incorrect statement with respect to photosystem 2. Select the incorrect statement with respect to photosystem 2. With respect to what? With respect to photosystem 2. Right? Incorrect statement. Select the incorrect statement with respect to photosystem 2. The reaction center is P680. It occurs on the outer surface of thylakoid, okay, found in grana lamella, okay, associated with splitting of water and release of oxygen. So basically, right, grana lamella is the word, and see, no doubt it is part of splitting there, 110%. Like water splitting. When you talk about this now, this is your thylakoid. Just have a look. This is your grana. So whenever we read it, right, here in the membrane you have PS2. Here OEC, oxygen evolving complex is associated with it. So definitely it is correct. ठीक? Reaction center is P680. It is correct. Found in grana. ये देखो, grana lamella. That's true. Lamellar word is also used for the thylakoid, and it will be on the outer surface. That is incorrect. That is incorrect. So the correct answer here is the second one. Next question, given below are the two statements, statement 1, statement 2. Read it. Statement 1, statement 2. Yes, bache. So a very significant contribution of meiosis is cell repair. Next, mitotic division take place in meristematic tissue of plant. So what should be the answer here? So very significant contribution of meiosis. Is it so? Meiosis, is it going to help in the cell repair? It will help in the gamete formation. So one is incorrect, but second one is correct. So the third is the answer. So it is such an easy question, and I know my students are going to nail the exam as well. Henna? Henna? Isn't it? Isn't it? You are going to give your best in the exam. You are going to qualify the exam. Do you believe in yourself? Tell me, do you believe in yourself? I am saying that definitely you are going to qualify the exam. If you have worked hard, if you have studied hard, if you are practicing the questions, if you are revising, you are going to qualify the NEET exam. And you are doing all that things. So just have faith. Just have faith. Isn't it? Just have faith, my dear people. So, dicot stem. Dicot stem. Dicot stem. So it is having what? Do not show secondary growth. No, they show that. Do not have endodermis. Okay. They have scattered vascular bundles. No. Right. They have parenchymatous pith. That is true. So it should be fourth one. Right. And here endodermis is not having the Casparian strip. It is known as star sheath. Right. That is something you have to keep in your mind. Next point. Right, for every carbon dioxide molecules which is entering Calvin cycle, how many molecules of ATP and NADPH are required? Right, so Calvin cycle, C3 cycle they are talking about, and they are talking about the ATP and NADPH. Yes, important and the simple one. Important and the simple one. Acha, tell me one thing. Do you want me to make the photosynthesis and respiration short revision series? Do you want me to make the short sessions for the respiration and the photosynthesis? If it is a yes, do let me know in the chat section. If it is a yes, some fire emojis, can't read it. Some fire emojis in the chat section if you people want me to make such type of session, right, for the short revision session of photosynthesis and respiration. Anybody here in the class? Yes, please. Okay. Okay. We'll take care of that. Don't worry. We'll take care of that. Okay. So, they are saying that what should be the answer? So, you are talking about the C3 where first step will be the carboxylation. The second step will be the reduction. And the third step here will be your regeneration. So, when it is reduction and regeneration, in reduction, 2 ATP, 2 NADPH will be used. In the regeneration, 1 ATP is required. So, of course, 3 ATP and 2 NADPH, this is what you need. But remember one thing, for 1 glucose, you need 6 turns of C3 cycle. So 6 turns means 3 into 6, 2 into 6, 12 NADPH, 18 ATP. That should be the answer, okay? That should be the answer, okay? Next question, which volume has the minimum magnitude? Minimum magnitude, of course, it is again the simplest one. Minimum one. Minimum magnitude, beta. Minimum magnitude, beta. Tidal volume. Per breath, 500 ml. Okay? Per breath, 500 ml. Next question. Select the mismatched pair with respect to complex tissues. What you have to select? You have to select the mismatched pair with respect to complex tissues. With respect to complex tissues. So they are saying that tracheids, see, tracheids, vessel, xylem fibers, xylem parenchyma. That is what we have. So tracheids are there, vessels are there. So here, if you will read it, they are saying that they are living cells with thin cell wall. It is not true, right? These three are dead. That's it, right? These two are the main tertiary element. So of course, right, they are dead. They need to conduct the water. They need more space for that. So here the correct answer is third, because living cells, they are not living cells. Next, which of the given step is catalyzed by pacemaker enzyme with respect to the EMP pathway? So which enzyme is the pacemaker enzyme in glycolysis? That is my question here. Which enzyme is the pacemaker enzyme in glycolysis? That is the question here. That is the question here. And it is also the regulatory enzyme. It is also known as regulatory enzyme. Remember, glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate. Then there will be fructose 1,6-bisphosphate. Remember, so here in this particular step, you have phosphofructokinase, if you remember. You have phosphofructokinase, if you remember, right? Phosphofructokinase, that is also the regulatory enzyme, right? So this has to be the answer here, right? This is the pacemaker enzyme. We talk about 3 regulatory enzymes, but yeah, this one will be your pacemaker enzyme. Understood? Next question. Mitotic cell division occurs in. Next question. Mitotic cell division, it occurs in. Yes, people? Mitotic cell division, it occurs in. It occurs in. Yes, students? So mitotic cell division, it occurs in. Where will you see this? Where will you see this? Are you going to see it in a diploid somatic cell? Yes. Tobacco mosaic virus, virus it is, abnormally folded proteinaceous plants. So A and E should be the answer. It's simple. Next, which feature is not related to active transport? Which feature? Okay, so directly we don't have active transport. I'm skipping this question. So, in plant cell, cytokinesis occurs by. In plant cell, cytokinesis occurs by. In plant cell, the cytokinesis occurs by. It occurs by. It occurs by. So in plant cell, cytokinesis, you know that cell plate formation will be there, cleavage furrow. Your cleavage furrow is there in the animal cell where actin filament is required. Now, the next part is all of the given are features associated with meristematic cell except. All of the given are the features associated with meristematic cell except. Except So all of the given are the features except the meristematic cell. They have dense protoplasm, increased evacuation. But increased evacuation thing we discuss in the case of your elongated cells, right? In the case of your elongated cells, you will see this. Next, oxygen evolved by green plants comes from H2O, not from carbon dioxide. This was proved by, again a very simple question. Again a very simple question. The oxygen which is evolved by the green plants, the oxygen which is evolved by the green plants, it is coming from radioisotope technique, right? It was proved with the help of that, right? So jaise you talk about the experiment na, JP, JI, JS, T, Cartoon Network, RHK. So Cornelius, when Niall was like that the H2O will give the oxygen. Who provided the experimental proof? Rubin, Hasid and Kamein's experiment with the help of that mass spectrophotometer. They used the radioactive, radio labeled radioactive oxygen, and then they checked it. Okay, then they checked it. That is the answer. Next question. Enzymes which cannot be activated by iron. The enzymes which cannot be, which do not need iron. Yes. Beta, carboxylase, phospholipase, nitrate reductase, they need it, but not the catalase. Okay. Next question. Which of the given can be, okay, can help in shortening the period between germination and flowering? Just give it a try. Just give it a try. Beta, it is actually, this is again from the deleted part, vernalization, which is the cold treatment, right? So let's come to this part. Z scheme are related with. The last question was from the deleted portion, so you can just skip it. So next is ki Z scheme are related with. Yes, people. Z scheme are related with. I believe you know the answer. Z scheme are related with. Of course, it is related to the light reaction. Next question. Number of ATP required for the, no, select the incorrect match here. Select the incorrect match here. Quick, again from morphology. Again from morphology. Yes? Select the incorrect match. What should be the answer here? Of course, conidia will be there in penicillin. Banana will have the rhizome. Zoospore will be there in chlamydomonas, but sucker will not be there in pistia. Right? It is having opposite. So during photosynthesis, what's going to happen? During photosynthesis, what's going to happen? During photosynthesis, what is going to happen? Yes, please. What should be the answer, guys? What should be the answer here? During photosynthesis, is it the water that is reduced and the carbon dioxide is oxidized? Right? So don't you think this should be the answer here? Carbon dioxide is reduced and we'll form the glucose, and the water will be oxidized here. And the water will be oxidized here. Isn't it? Isn't it? Second should be the answer. Don't you think so? Don't you think so? Next question. Which of the following statement is not true for the Krebs cycle? Which of the following statement is not true for the Krebs cycle? After this, we'll jump to the questions of physiology, human physiology. Yes? Which of the following statement is not true for the Krebs cycle? That is the question. That is the question. Acetyl coenzyme A is an entrant. True. Citrate synthase enzyme is the first enzyme. Yes. Because see, your oxaloacetate, that will accept your acetyl coenzyme A, right? And you are going to get the citrate there. Okay, and here the enzyme will be citrate synthase. Focus here, synthase it should be. Now, fumaric acid is produced from succinic acid. You can just search it. You will get the trick of mine for the Krebs cycle as well. So it is your come in and cake, scooty, scooter, fastly move on. So, oxaloacetate will accept acetyl coenzyme A, will form citrate, then isocitrate, then alpha ketoglutarate, succinate, come in and kick scooty scooter. Firstly, there will be succinyl coenzyme A, succinate, right, bache? Then fumarate, then malate, then again oxaloacetate. So, yes, fumaric acid is produced from succinic acid, right? And succinic dehydrogenase is present in mitochondrial matrix. It's a big no. This is the only enzyme of Krebs cycle. It is the only enzyme of Krebs cycle which is not there in, right? Not there in matrix, but in inner mitochondrial membrane. But in inner mitochondrial membrane, it is forming the second complex, right? Second complex, if you remember. Next question. Which of the given is a polycarpic? Chalo, let it be. The first heart sound is produced when? The first heart sound is produced when? The first heart sound is produced when? Yes? Read the statements. Yes. First heart sound, right? First heart sound is produced when what is going to happen, students? Right? Don't you think that when there will be the bicuspid and tricuspid, these valves will get closed, and they are also known as AV valves. They are also known as AV valves, right? Auriculoventricular valves they are. Done? Next question. All of the following are the intermediate products of glycolysis except. All of the following are the intermediate products of the glycolysis except. Yes, except. Yes, please. So all of the given are the intermediate products of the glycolysis as phosphoglyceric acid, malic acid. Do we talk about them? Do we talk about them? They are the intermediate products of the glycolysis except. So malic acid, so definitely we are not going to talk about. Nahin? जैसे we start with glucose, glucose 6 phosphate, fructose 6 phosphate, 1,6 bis phosphate. Then what do we have? We have the glyceraldehyde 3 phosphate and we have the dihydroxyacetone 3 phosphate. And after that, you know that dihydroxyacetone 3 phosphate will again form the glyceraldehyde 3 phosphate. And then further, right, regarding the glycerates we discuss about, but not the malic acid. So it is not the correct answer. So stalk of the ovule by which it remains attached to the placenta is called. The stalk of the ovule by which it remains attached to the placenta is called. Understood? So it has to be attached to the placenta. The stalk is basically the funicle. The point where it is touching the ovule is the hilum, right? It is the hilum, right, bache? Right, bache? So let's speed up, guys. Let's speed up. Let's move to the next part. Okay. So what is atherosclerosis? What is atherosclerosis? Guys, quick. What is atherosclerosis? Don't you think it is a type of coronary artery disease? It's a type of coronary artery disease, right? Do check the symptoms here. A is the correct answer. Next question, photosystem 1. Very important topic it is, right? The photosystem 1, it's a very important topic, guys. Speed up, quick. Quick, photosystem 1, is it involved in cyclic photophosphorylation, attached to oxygen evolving complex, has a reaction center which absorbs maximum at 600 nanometer, found in both grana and stroma? Beta, it is involved only? No, it is involved in both cyclic as well as non-cyclic. It is not attached to oxygen evolving complex. Here it is P700 reaction center, found in both. That is true. So fourth has to be the correct answer here. Fourth has to be the correct answer here. So how many nuclei are present in a mature embryo sac? How many nuclei are present in a mature embryo sac? How many nuclei are present in the mature embryo sac? How many nuclei are present in the mature embryo sac? See, mature embryo sac near to the micropylar end, chalazial end. Micropylar end, this one is your egg. Surrounding me, you will have your synergids having filiform apparatus. Chalazial end will have your antipodal cells. This larger is the central cell having 2 nuclei, N plus N. So 1, 2, 3, 4, 5, 6, 7, 8. 8 nuclei, but it is 7-celled. That used to be NTA's favorite question. This year also you can expect the questions from this particular part. Okay, so how many nuclei are present in the mature embryo sac? So when it comes to the nuclei, the answer has to be third. Am I right? Next question. Exchange of nutrients, gases between the blood and the cells always occur through which fluid? Always occurred through which fluid? Always occurs through yes bache. So exchange of nutrient, all the gases, etc. between the blood and the cells always occur through interstitial fluid, tissue fluid, lymph. It is all same. So the answer has to be all of these. The answer has to be all of these. The answer has to be all of these. So first action spectrum of photosynthesis was described by first action spectrum. Again, the NCERT line. Matlab sadhana, what am I supposed to do? What am I supposed to do? Yes? Time is going fast, ma'am, go fast. Matlab, what should I do? Should I just move a question to the screen and then next question, then next question, then next question? Do we need to do that? Matlab, we should not read the question. Yeah, we should not give the students time to read the question. Huh? So first action spectrum of photosynthesis, it was described by T.W. Engelmann. So first action spectrum it is, you are talking about the rate of photosynthesis here. Next question, in Mendelian dihybrid cross, out of 96 individuals, right, obtained in F2, how many will show the one dominant allele? Yes, one dominant allele. Tell me quickly. Tell me quickly. Yes, please. Done, बच्चे? चलिए, अब in Mendelian dihybrid cross, what should be the answer? What should be the answer? Yes. So here the answer is, they are saying in a Mendelian dihybrid cross, it's a dihybrid cross. So dihybrid cross matlab something like that, something like that. Out of 96 individual which are there in F2 generation, how many of them will, how many of them, total individuals are 96. Take how many have at least one dominant allele. So if it is a dihybrid cross, this is what you have to consider. You have to consider two different characters here, right, bache? Now they are saying what has to be there in the F2. If you remember when we used to take the ratio 9:3:3:1, 9:3:3:1, that is what we used to consider. Yes or no? Yes or no? That is what we used to consider. Now we have to tell that how many of them will have at least one dominant. At least one. Yes, at least one dominant allele. If you will focus on this particular ratio, it is the one only where we used to get the situation like this. Here it used to be this. Here, yes, bache, it used to be this. Matlab in general, if I have to tell you, this is round yellow. This used to be round green, if you remember, round green. This you should round green. This should be wrinkled yellow, and this should be wrinkled yellow, and it was again wrinkled and green. Remember, bache? Remember? So only this one is having both the recessive alleles, but these three will show you what? Dominant allele. So out of that 16 individual, 15 individuals, exactly my point. Very good, Aditi. Out of that 15 individuals are showing the dominant allele, at least one dominant allele, only one is not showing it. So now apply the same thing on the 96 individuals, right? Right, apply the same thing here on the 96 individual. You'll get the answer. You'll get the answer. Okay, any doubt? Any doubt? Done? Done, done, done. Okay, next. How many matchings are correct? How many matchings are correct? Right? How many matchings are correct? Read it quickly, be fast. Yes, students, be fast. Done? Done? So how many matchings are correct? PCT is there, counter current mechanism, renal coxal counter current mechanism again, right? 2 times it is given. So if I talk about the PCT, reabsorption is there, reabsorption is there. Is that true? Is that true? Then counter current mechanism, concentration of urine, true. Renal coxal mein, filtration of blood, true. Counter current mechanism mein, do we have the role of vasa recta here, guys? Do we have the role of vasa recta here, people? Tell me quickly. Do we have the role of vasa recta here? True that. So all 4 are correct. Am I right? All 4 are correct. Am I right? All 4 are correct. Am I right? Yes. Am I right? Next question. Plant hormones which show the apical dominance is. Apical dominance is the word. A for apical dominance, A for auxin without any doubt. A for apical dominance, A for auxin without any doubt. Done? Next, which of the given gamete fusion reflects nullisomy condition? First of all, you should know what is nullisomy. Anybody? Monosomy, trisomy. In my lectures, I have discussed that with you all. I have discussed that with you all. Nullisomy. What is going to happen in nullisomy? Anybody? Anybody? Don't you think it will be 2n minus 2? Yes. जैसे monosomy में 2n minus 1. Right? Nullisomy में it has to be 2s minus 2. Like trisomy will be your 2n plus 1. Okay. So now here the word is what? The word is nullisomy. So now if we need this condition, मतलब 2 chromosomes are less. Let's suppose if humans, they are having 46 chromosomes, right? A pair of homologous chromosome is not there. Total 44 chromosomes will be there. Matlab this gamete will also lost one. This gamete will also. So according to you, what should be the correct answer? Don't you think it should be second? Don't you think it should be second? Yes, people. Don't you think that it should be, right? It should be second. Right? Because nullisomy is the word. And let's suppose if I change the statement and I instead of nalisomy, if I mention monosomy, then if instead of nalisomy, if I mention monosomy, then yes, please, then now got it? Now got it? Trisomy, tetrasomy. So it's not mandatory that always they will ask the disorders from you, right? That's not mandatory. Sometimes question can be like this. Now, scientists associated with auxin hormone. I believe you remember that. Directly NCERT based question it is, guys. Directly NCERT based question it is. Yes. So scientists associated. These two. So can I say that fourth is the right answer? Can I say that fourth is the right answer? Chaliye. Next, natural plant hormone which delays the senescence is. Natural plant hormone which delay the senescence. The word is it has to delay the senescence. Which hormone will do that? Which hormone will do that? Yes, everybody. Which hormone will do that? Do you remember Richmond-Lang effect? In the quick revision session, in the marathons, we have discussed that. In the quick revision session, in the marathons, we have discussed that. So it has to be zeatin. It has to be zeatin. Basically, you are talking about the cytokinin here. Which hormone are we talking about here? Cytokinin. Next question. How many of the given enzymes are involved in DNA replication? DNA replication. Yes, let's solve together. Do we have helicase? Yes. DNA dependent RNA polymerase, also known as primase? Yes. DNA ligase? Yes. Topoisomerase? Yes. But do we have peptidyl transferase? No. So 1, 2, 3, 4. Third should be the correct answer. Am I right? Third should be the correct answer. Am I right? It is the third that should be the correct answer. Am I right? Okay, next question. Total number of cytochromes which are involved in ETS of mitochondria. Which are involved in ETS of mitochondria. Of mitochondria. Cytochromes involved. Do check it from my short revision session as well. The answer has to be 5 here. The answer has to be 5 here. Okay. The answer has to be 5 here. Is that clear? Sure. Any doubt? Yes. You know that means cytochrome B, cytochrome C, cytochrome C1, right? And cytochrome A, cytochrome A3. Yes. Yes. Next, ginger reproduce vegetatively mainly by. Ginger is the example of. Ginger is the example of. Yes. Ginger is the example of. Yes, beta, ginger is the example of rhizome. So, of course, it will divide vegetatively mainly by rhizome. Next, opening of DNA helix and continuation of elongation process in transcription is achieved by opening of DNA helix and continuation of elongation process in transcription. You have to open the DNA helix and then elongation. Of course, beta, here it will be RNA polymerase. So in 2020, NEET 2020, some same kind of question was there, right? And even we got confused because we just read one thing, that opening of DNA helix, helicase came in our mind. But you have to read the complete statement. So transcription is also the word there. Next question. All of the following processes can release carbon dioxide except देखो, alcoholic fermentation में yes, oxidative it is decarboxylation and Krebs cycle, yes, ketoglutarate 5 carbon containing, 4 carbon containing, yes. So we are left with this one. Okay, next. Embryo sac of angiosperm already discussed. backbone nahi. Okay, very simple question it is. Just have a look. Which is not a plant growth regulator? Of course, porin. Porin. Next question. Citric acid is commercially produced by. Beta, do not miss microbes in human welfare. Do not miss this particular chapter. Trust me on that. If from this particular chapter, 110% question will come. 110% question will come. So, citric acid is commercially produced by. Citric acid is the word. Aspergillus, the fungus. Okay. Okay. Next question. All of the given statements are correct. Okay, leave it. Hanji. In most the higher plants, the growing apical bud inhibits the growth of axillary bud. This phenomena is known as. In most higher plants, growing apical bud will stop the growth of the axillary bud. This is known as. This is known as. This is known as. A very simple question it is. Of course, it is the apical dominance shown by auxin, and cytokinin will counteract it. Okay, so which of the following are non-albuminous seeds? All of the following are non-albuminous seeds except. All of the following are non-albuminous seeds except. All of the following are non-albuminous seeds except. Except, of course, non-albuminous, non-endospermic. Okay, so you know that such type of seeds are in dicots, but here the castor is an exception. It is a dicot, right? It is a dicot having what type of seeds? Having endospermic seeds. Okay, next, during primary treatment of sewage, all the solids that settle. Primary treatment, these are the solids that will settle. The answer will be, easiest question, easiest answer, primary sludge. And what about this part? This is effluent, my dear people. Where will it go? For the secondary treatment. For the secondary treatment, right? So perisperm is. Perisperm. So perispermic seeds are the one in which nucellus is persistent in the mature seed. Just you have the example of your black pepper, beetroot. So do remember the examples. Next question. Chromosomal complement of a person affected from Turner's syndrome. Disorders. Acha, let me give you one good news. Right, let me give you one good news. Right, you know that disorders are very important. Do you know that? Disorders are very important. Do you know that? Disorders are very, very, very important. So I'll be taking a marathon or a short session, whatever it is going to be, for the disorders only. I will be taking a session. for the disorders only. Okay, chaliye. So it is the Turner syndrome. It is the result of what? Monosomy or trisomy? Beta, it will be the result of monosomy. That is -1. Take. So here 45 chromosomes will be there. So this has to be there 44 plus XO. Isn't it? 44 plus XO. Anybody having any doubt here? Yes. Anybody having any doubt here? If anybody is having any doubt, do let me know, guys. Do let me know. 105. What should be the answer? Okay, just allow me 1 minute. $105 it is, and a very simple question. As I said, bache, the answer will be 44 plus XO. So I hope this part is clear to you. Next question. Which bioactive molecule is used as immunosuppressive agent? Which one is used as immunosuppressive agent? Yes, please. Which one is used as immunosuppressive agent? And you know that is going to be, is it cyclosporin? Yes, madam. Cyclosporin. It is immunosuppressive agent which is used, right, during organ transplantation. And who's going to give that? Trichoderma polysporum. Who's going to give that? Trichoderma polysporum. Second question. Okay. Next part. Which statement is incorrect for the thick filament of the muscle fiber? Quick beta. Which statement is incorrect for the thick filament of the muscle fiber? Yes. Incorrect statement we have to answer here. Everybody quick. Incorrect statement we have to answer here. So each thick filament is a polymerized protein. That's true. Globular head with a short arm called heavy meromycin. True. Each thick filament is formed by actin. No. No. Here you'll talk about myosin. So active site for actin are located over meromycin. True. So third is the answer. Next. How many are the autosomal dominant disorders? Beta, the word is autosomal dominant. Autosomal dominant. So I'm cutting the useless options here as well. Right, I'm cutting the useless options here as well. So you tell me what should be the answer here? Hemophilia? No. Sickle cell anemia? Again recessive. Right, again recessive. Phenylketonuria? Again autosomal recessive. This is X-linked recessive, autosomal recessive. Only this one. So one should be the answer. Right, so be ready to solve such kind of question tomorrow. Now, all of the given are no. Leave it. Okay, give it a try. One question from joints. Yes, even this year also it is expected. It is expected. They are saying how many matchings are correct. They are saying how many matchings are correct. So they go. One thing is for sure that these two are the correct only. These two are the correct only. Right. Now when you talk about your pivot joint, this, if it will be fibrous, it will not show any movement. But in pivot we see the movement at least X is 1, isn't it? Right. So a cartilaginous joint is your saddle joint or cartilaginous joint is your vertebral column. So saddle joint, is it the example of cartilaginous joint? No. है ना? Saddle joint is the example of, yes bache, it is the example of anybody. Is it the example of your cartilaginous joint? No, it is also the example of, as for SAD, as for synovial joint. So it will show the movement. And cartilaginous joints, right, they show slight movements, intervertebral discs. So this is true. These three are true. Next, enzymes which have ribonucleic acid synthesis capability are. They can synthesize RNA. That's what they are saying. So reverse transcriptase में तो बेटा from DNA you are getting mRNA, है ना? Sorry, from mRNA you are getting DNA. है ना? From mRNA you are getting DNA, reverse transcription. So they are saying enzymes which have the ribonucleic acid synthesis capability. So these two, Ribonucleic acid they have to synthesize, not deoxyribonucleic. So first and third should be the answer. Next, yes, beta, this is from ecology. This is from ecology. Give it the answer here. Give it the answer here. Right. So what should be the answer, people? What should be the answer here? Barnacles growing on the back of the whale is the example of, is the example of so don't you think it is commensalism where one is getting benefited and the other is at neutral position, no benefit, no loss, right? Next question. Back of the whale it is. Lac Z gene is. Lac Z gene yes please. Guys, 1 second. Barnacle one, let me check. Barnacle may, I just told you they are growing on the back of the whale. It is the example of, is it commensalism or parasitism? Yes, yes, yes. Let me check it. Is it, sorry, let me, answer it to me. Answer it to me. Is it the example of parasitism or commensalism? Because they are growing on the shark. They are growing on the, sorry, they are growing on the back of the whale. This is something that I have confused. So, they are not growing, they are growing on the back of the whale. So, what kind of example are they? So it is commensalism. But let's suppose there is one more relationship. That's why I just got confused. Ki barnacles, some barnacles, they grow on the shark as well. But here, it is the example of parasitism. But now they are talking about the whale. So it is commensalism only. It is commensalism only. So do write this point, right? Do mention it. Okay, do mention it in your notes. Next, lac Z gene, you know that beta galactosidase it will give. So, okay. Lac Z gene of lac operin has a binding site for repressor protein. Next is, it is having a binding site for repressor protein, and the next is present downstream of lac operator. So, bache, this one will be your upstream. You'll use negative sign here. This one will be your downstream. So, if you remember, that is PIPOZYA. That is PYPOZYA. So, operator, this is the operator. So, they are saying downstream. Of course, it is downstream to the operator. Downstream to the operator. So, definitely, second is the correct answer. Next, which of the given is not the characteristic of anthropogenic ecosystem? So, when the word is anthropogenic, matlab man-made. Man-made. So, they will have self-regulatory mechanism? No. So, one should be the correct answer. Next. Okay. Done. It's a good question. I like this question. I like this question. Yes, I like this question. Answer question number 116 and be quick, please. Be quick, please. Read it quickly. Done? See, it's a very good question. They are saying we have to convert 1,000 substrate molecules into the product. 1,000 molecules of enzymes are used. Beta enzymes, right? They will, after the reaction, they are obtained as such. They will not be consumed in the reaction. So the answer here will be 100. So if your concepts are right, if you know the concepts, right? Right. So 100. So that is why 100 is the answer. Next, in which of the given food chain size of the organism finally reduced at higher trophic level? Finally reduce at higher trophic level. That's the question, people. That's the question, people. Quick. That is the question. Hanji. It is finally reduced. It will be very small. It will be very small. Chalo, that is your homework. I want the answer of this question in the chat section. Next, which one is the least productive ecosystem? Least productive ecosystem. Yes, least productive is the word here. Least productive is the word here. So of course it is going to be the deserts. And here, right, detritus, auxiliary, grazing. So they are saying the size of the organism reduces at higher. So I will just give you one hint there, because I have seen that some students, they don't know the meaning of this word. So it should be parasitic, parasitic food chain. Now you have to answer it in the chat section. Will it be done? Will it be done? Chali, next question. Which of the chalo, this is done. Not a hormone. Next, population interaction. Okay, so again, very simple question. I want the answer quickly. I want the answer quickly. I want the answer quickly. Yes, I know you people are going to do amazing in your exam. Quick. The one will be benefited, neither harmed, non-benefited. This kind of relationship is this commensalism. So guys, I'll share the PDF in our Telegram group. Right, I'll share it in the Telegram group, and you can join my Telegram as well. That is Love Biology by Ambika ma'am. Right, so it's simple Ambika Sharma Biology. So you can join it. So now quickly answer this question. It is data related, but it is from the biodiversity. It is from the biodiversity. Quick. Incorrect match. This is for the birds. Next. Yes, beta. Next. Select the correct statement for the given below diagram. Select the correct statement for the given below diagram. Quick. Quick. Done? Just a done. So select the correct statement for the given below diagram. देखो, they are, first of all, it's a reptile for sure. It's a reptile for sure, right? So it is a reptile. Triploblastic it is going to be, yes. Is it the common garden lizard? No. Coelomate, yes. Because it is a reptile, so we will say bony. It is three chambered heart should be there. So answer should be, do we have B, do we have C, do we have E? So yes, students, what should be the answer here? What should be the answer here? It should be fourth. It should be fourth, right? So that is how it is. Next question. Okay, which of the given is an ex-situ conservation strategy for the conservation of biodiversity? Yes, bache. So XC2, when the word is XC2, then of course you know that beta, XC2 means off-site conservation, not at that particular point, it is off-site conservation. So here wildlife safari park will be the example of XC2 conservation. Next, so do not confuse with wildlife sanctuary and safari park. If it is safari, it is XC2. If it is sanctuary, it will be in situ. Next question. In the given below hormones, how many are secreted by placenta? Guys, you are not going to get the break, right? I'll be very honest about it. I will not give you the break. Okay? So if you are interested in having break, you can leave the, right? You people can leave the class for some time and then you guys can join in. Okay? Then you people can join. Okay? Done? So in the given below hormones, how many are secreted by placenta? How many are secreted by placenta? How many? We will get HCG, HPL, right? Even your estrogen, progesterone from them, relaxin from them. So 1, 2, 3, 4, 5. 5 it has to be. 5 it has to be. Right? How many? It has to be 5. It has to be 5. It has to be 5. Is that clear? Is that clear? Chaliye, next. Population is said to be stable. The word is stable here. The word is stable here. Yes, the word is stable here. So it is very important, bache. It is very important. The growth rate is almost 0, right? So it will be, right, something like that. It will be your pyramid, okay? Okay, the growth rate will be almost 0 here. Next, how many cells are haploid in nature here? Quick, how many cells are haploid in nature here? Quick. How many cells are haploid in nature here? Guys, quick. Yes. Spermatogonia, they are diploid. Primary spermatocyte diploid. Ovum will be haploid. Secondary spermatocyte haploid. First polar body haploid. Second polar body haploid. Primary oocyte, it will also be diploid. So 1, 2, 3, 4, right? So first is the correct answer here, right? First is the correct answer here, right? Yes or no? It's the first that is the correct answer here. It is the first that is the correct answer here. Yes or no? Yes or no? Next, in the equation of species area relationship established by Alexander von Humboldt, A represents. You remember? A represents. It is species area relationship. It is species area relationship. A very simple question. A is equals to area. Can anybody tell me the equation? Can anybody tell me the equation? Anybody here in the class who can tell me the equation? Yes. So A is area, but equation. Do you remember that? Do you remember that? Very good. C is equals to, S is equals to CAZ. Then, which one of the following is the incorrect match? Which one of the following is the incorrect match? Which one of the following is an incorrect match? Is an incorrect match. Quick. Incorrect match. Condoms, are they the barrier method? Diaphragm implant? No. This is IUD, true. Periodic abstinence is a contraceptive method, true, but diaphragm is not the implant. Next, which of the following contributes maximum to the total vertebrate global biodiversity? That is the word here. Again, the data-based question. Again, the data-based question. Quick. Quick. It has to be fishes. It has to be fishes. Refer the following cross. Noise. Answer this. Answer this. Give it a try. Give it a try. So they are saying probability of occurrence of a progeny with this genotype. Take the screenshot, guys. Just take the screenshot. Post the answer in the comment section. That will be the exercise for all of you. Right? Let me give you the answer. It will be this. It will be this. You will solve it. You will post it. You will solve it. You will post it. And if you are not able to get this as answer, you have to mention it there. I'll explain it. Okay. Now, how many statements are correct with reference to your mitochondria? With reference to your mitochondria. Yes. So, how many? The number of mitochondria per cell variable depending on the activity of cell. True. It's double membrane mount. True. And other than that, they are saying that yes, the two membrane have their own specific enzyme associated with the mitochondrial function. It is divided by fragmentation. So binary fission will be the answer here. So it has to be second. It has to be second. This should be the correct answer. Next question, beta, select the what will be the amount of DNA in S phase of cell if the meiotic product has 8 pg? Nice question. Nice question. What should be the answer here? What should be the answer here? It's a good question. It's a good question. So that is how it is. Sir, be ready. You tell me. Okay. So the next point here is, after me Wassim sir will come, okay, for the question practice. So this is what we have in meiosis. When you start here, the DNA is 4C, right? Here it will become 2C and 2C, then it will become C, C, C and C. If meiotic product is having 8, right? Meiotic product is having 8 picogram. So they are saying in S phase of cell. S phase mein the DNA amount is doubled. Matlab this is what the cell is containing. So 8 multiplied by 4, 32 should be the answer. Is that clear? 32 should be the answer. Is that clear? Is that clear? Next question. Okay, so given below are the two statements. Given below are the two statements. Yes, bache? They are the two statements. What should be the answer? Gene therapy is a collection of methods that allow the correction of gene defect that has been diagnosed in a child embryo. True that. The first clinical gene therapy was in a 14, first of all 4 years old, and the year is also incorrect here. So one is correct and second is incorrect. Okay. Next, with respect to sexual reproduction in fungi, which one is incorrect? Which one is incorrect? Yes, which one is incorrect? So select the incorrect statement with respect to the sexual reproduction in flowering plants. So fusion of protoblast is called plasmogamy. True. In ascomycetes, okay, that is your sac fungi, dikaryotic phase can be seen. Yes, beta, it will be there. Karyogamy is absent in all fungi. Not true. Conidia are the haploid spores. True that. And they are the result of what? Sexual reproduction or asexual reproduction? Conidia. What type of spores are they? Sexual reproduction or asexual reproduction? Tell me. Next, select the incorrect statement with respect to the wind pollinated flowers. Firstly, tell me sexual, asexual, and then answer this question. Till then, you can read it. Rest, please type it. Incorrect statement with respect to wind pollinated flower, right? So they have the light and non-sticky pollen grain. True that. Well exposed stamens. True that. Large feathery stigma. True that. So flowers have long, sticky, unwettable stigma. That is incorrect. Next, insulin used for diabetes was earlier extracted from pancreas of slaughtered cattle and pig, and that insulin from animal resource through though cause some patient to develop allergy. Okay, so again a very simple question, right? You know that it's a fourth one. Both statements are correct. Next, our phycoerythrin pigment is majorly present in the members of. Our phycoerythrin pigment majorly present in the members of. In the members of. Yes, it is present in the members of rhodophyceae. That is why they are having the red color. So which one is the autosomal recessive disorder? The word is autosomal recessive disorder, right? So of course, autosomal recessive will be thalassemia. Myotonic dystrophy is autosomal dominant. They are X-linked recessive, right? These two are X-linked recessive. Next question, people. Statement 1. No. Okay. In a typical nucleus of eukaryotes, euchromatin. In a typical nucleus of eukaryotes, euchromatin. So, what will happen to euchromatin? It is said to be transcriptionally active and heterochromatin transcriptionally inactive. So, lac operon is switched off when repressor protein produced by regulatory gene binds to which of the given gene. The lac operon, it is switched off, right, when repressor protein. So, you have to keep it in your mind. You have to keep it in your mind that whenever you talk about the repressor, whenever you talk about the repressor protein, whenever you talk about the repressor protein, right, bache? So in the case of that repressor protein, that will always bind with the operator, that always interact with the operator gene, right? That is something you need to keep in your mind. Okay, that is something you need to keep in your mind. Next, in the year 1963, two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. So Hind2 always cut DNA at a particular point by recognizing a specific sequence of 9 base pair. Good question it is. It's a good question. Yes. Do you know the answer? Yes, people, speed up. Do you know the answer? Everybody here in the class. I want you people to give the answer. Done? So, first statement is correct, my dear students, but the second statement here, instead of 9, 6 base pairs should be there. Okay, okay, so 1 is correct, but second is incorrect. Next, cyclosporin A is produced by dash and used as dash. Fill in the correct statements. Fill in the correct statements. Done? So trichoderma polysporum, cyclosporin A, that is immunosuppressive agent, right? So cyclosporin A is produced by trichoderma polysporum used as immunosuppressive agent. So this one is true. Next, a population is said to be mature or stable. That is something we have already discussed. So eco R1 is isolated from people. Eco R1 is isolated from everybody. Yes, eco R1 is isolated from eco R1 is isolated from. Eco R1 is isolated from. Eco R1 is isolated from. Exactly, it is second. Next question, 2,4-D. What's that? 2,4-D. What's that? 2,4-D. What's that? 2,4-D. What's that? It is a VD side, yes, so it is dichlorophenoxyacetic acid. Dichlorophenoxyacetic acid it is. Dichlorophenoxyacetic acid it is. Then, which of the following set of organism have tissue grade of organization, blind sac body plan, and they have radial symmetry? They have radial symmetry. So look at the question, read the characters first. What are they saying? Tissue level. So when it comes to tissue level, you have phylum Cnidaria and Ctenophora. This is the first thing we need to keep in our mind. Blind sac body plan means only one opening is there, which will act like the mouth and the anus. Radial symmetry. So Asterisk starfish, no. Pila mollusca, no. Fasciola platyhelminth. So third will be the correct answer. So this type of questions are expected in paper. This type of questions are expected in paper. Okay, this type of questions are expected in paper. Next, cells of the loose connective tissue that play a major role to ingest cell debris and foreign matter is. Yes, cells of the loose connective tissue that play a major role to ingest cell debris and foreign matter is macrophages, of course. Right? Macrophages, of course. Next, all of the following are the example of neutral amino acid except. All of the following are the example of neutral amino acid except. So all of the following are the examples of neutral amino acid except. So lysine, you remember it is the basic amino acid. How can we remember that? It is HAL, H-A-L. So H stands for histidine, A stands for arginine, L stands for lysine. So it is histidine, arginine or lysine. It is histidine, arginine or lysine. It is histidine, arginine or lysine, basic amino acid. So glutamic acid and your aspartic acid, they are acidic amino acid. Rest all will be what? Neutral. Next question, how many of the given organism they exhibit bilateral symmetry in adult forms? How many of the given organisms they exhibit bilateral symmetry in the adult form? Tell me. Tell me. Tell me, everybody. How many of the given organism they exhibit bilateral symmetry in the adult form? So, okay, sepia, mollusca it is, fasciola, platyhelminthes, echinus, echinodermata, annelida, echinodermata, hemichordata. So now you know the examples as well, that which belongs to which phylum. So what should be the answer? What should be the answer? In the Balanoglossus you will see that है ना adult में, and in the Nereis also, in the sepia and so 4 should be the answer. Fasciola will also show the bilateral symmetry. 4, this matlab 4, second option is the correct answer. Now, heart will receive only deoxygenated blood in all the given organisms except. Will receive only deoxygenated blood in all the living organisms except. Except. Of course, beta, the heart will receive only single circulation. You are talking about the single circulation here. What are we talking? We are talking about the single circulation that is there in the case of fishes. So bufo is only amphibian here. That's why it is the answer. Pterophyllum is your angelfish, right? This is your labia pristis, that is your sawfish. Next, which is not applicable to which of the given protochordate retains the following character throughout the life? The word is protochordate. The word is protochordate. The word is protochordate. Beta, protochordates means you are talking about UC. Means you are talking about urochordates and you are talking about cephalochordates. Am I right? Now they will show retrogressive metamorphosis. Here it is progressive metamorphosis. Retrogressive and progressive is there. They will show all the chordate character throughout the life. So in urochordates, sad is the trick. They are sad. Salpa acidia doliolum. So salpa acidia doliolum. We are left with bronchiostoma. That is the example of cephalochordates. Next, the junction in the epithelial tissue that will facilitate the cells to communicate with each other by connecting the cytoplasm. So it should be, it is the easiest question, beta. It should be next is chemistry. So sir is active. It should be gap junction, of course. Next question. Okay. Chalo, let it be this question. Which of the following would be true if a person has less body fluid? If the person has less body fluid, who's boss? Unacademy NEET English. Matlab Wassim sir is typing here in the chat section. He's saying the boss is here. Kaun sa boss? Kaisa boss? Matlab, you want me to insult you right now? Huh? Thalaiva is here. He is no Thalaiva. He's a normal human being who cannot give, right? Who cannot let others rest. Who's like, nahi, we all have to work. But when it comes to him, he doesn't work at all. Thalaiva for the reason for the NEET students. Just complete your chemistry. You will definitely get selected in the exam. Okay, chalo, let's solve this question. So they are saying if the person is having less body fluid, if the body fluid is less, okay, so we have to increase it. So if we need to increase that, so decreased renin and angiotensin level. See, this is the RAS mechanism. It will help in glomerular filtration rate. It will increase that. Glomerular filtration rate, matlab body fluid is also high. Now, increased angiotensin 2 and non-functioning of RAS would be true. Which of the following would be true if a person? Okay. 1, 2. Okay. So, bache here, if the body fluid is low, it simply means what is the issue? The RAS system is not applicable. The RAS system is basically So, less body fluid means the question is 160 na. So, here we have less body fluid. Less body fluid is less. So, basically, if you remember, when GFR is low, when glomerular filtration is low, right, just to balance that out, the RAS mechanism will get activated. Isn't it? The RAS mechanism will get activated, isn't it? Isn't it? Yes or no? Yes, yes or no? So we have to balance that out. Yes or no? Answer this question. Answer this question. We have to balance that out. So if the body fluid volume is low, we have to balance that out. So according to you, what should be? What should be activated? RAS mechanism should be activated or not? The RAS mechanism should be activated or not? You tell me, check the options and do not make the mistake here. Body is having less fluid. So if body is having less fluid, what should be the scenario? What should be the scenario? Matlab it is basically dehydration is there na? Dehydration is there na? So if the body is having less body fluid, so don't you think then the body would have decreased the renin production, right? So related with the renin, related with this part, So what should be the answer? Don't you think it should be A and D? Don't you think it should be A and D? Ah? Third, how come third? How come third? Why not A and D? Why third? Why not A and D? Why not A and D? Acha, Yawar sir is also here. ठीक है. Why not? Why third? Why third? Why not fourth? See, if you look at the option, increase angiotensin 2 and aldosterone level and increase renin and ADH level. How do we relate it? When there is a low GFR, we have to balance that out. Juxtaglomerular cells, they will release renin. You know that renin has to act on angiotensinogen, right? And that will convert it into angiotensin 1, then to 2, and that will act on the aldosterone, and that will act on adrenal cortex to release aldosterone. That is how it works. That is how it works. So why are we doing it? Because we have to increase the blood pressure. Because we have to increase the blood pressure, we have to increase the body fluid volume. So here they are saying which of the following would be true if the person is having less body fluid. If less body fluid is there in a person, then of course we have to activate the RAS mechanism, right? RAS mechanism. So here basically this is how you have to understand that if the body fluid is low, then of course the body will be like, I will release the juxtaglomerular, I will release the renin that has to act on angiotensin 2, and the further process will occur. So that is why the answer given is third here, right? Third here, this is going to happen if a person is having this particular condition. Is that clear? Yes, aldosterone will help in that absorption, right? ठीक है, चलो. So now, right now let's start chemistry. What say? Let's start chemistry. What say? नहीं? नहीं ना? वाह 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 हमद. Okay, ठीक, very good. वैसे don't you think that all the teachers here, oh my bad, don't you think that all the teachers here on this channel, they all are amazing? है ना? हाँ जी, start करो. हाँ कर दी.
Guys, can you all hear me? Am I audible, visible to every one of you? Good afternoon everyone, good afternoon. Good afternoon. Let me know quickly in the chats if all of you can hear me, if I'm perfectly audible, visible to every one of you. Quickly in the chats, people. Quickly in the chats. Good afternoon, people. Good afternoon and welcome back. Good afternoon and welcome back. So are you done with your biology? Yeah? Are you done with your biology? Are you done with your biology, people? No, no, there won't be any break. There won't be any break. So let me know quickly if all of you can hear me. Yes, let me know quickly in the chats with some fire emojis, everyone. Everyone, everyone. Perfect, 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 people. Perfect, perfect, perfect. All right. Your Ambika ma'am is calling me. Just a second. Anji. हाँ तेरी देन है ना चल बाय हाँ जल्दी तूने स्टार्ट किया ना इसलिए चल बाय 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 अच्छा ओके बाय should we start then, guys? Should we start? Should we start? Should we start? Should we start? All ready? All ready? All ready? Quickly, quickly in the chats. Should we start? My dear students, one thing I'm going to tell you. One thing I am going to tell you that the questions which I will be doing for the subject chemistry in your actual NEET examination, your questions are directly or indirectly going to be asked in the similar manner. Same pattern questions, whatever I've come up with in this particular session. Okay? All right, so I would want you guys to take a note of every single question because every single question is going to be very important, right? And once I'll be done, then it's going to be HSP sir who will be joining in. And after HSP sir, it's going to be your Yavar sir who will be joining in for the physics as well. Okay? So quickly give me some fire emojis in the chat so that I can start the session with the high Josh people. Quick, 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 everyone. I want the chats to be very active. I want you to be active in the chats, right? I do not want this session to be really boring. Yeah? I do not want this particular session to be boring at all. So quickly, let me know in the chats. Yes? Whatever questions I'll be showing on the screen, first of all, you'll be marking all the answers in the chats. Okay? You'll be marking all the answers in the chats. Perfect? Yes, yes. Session PDF I'll be sharing on my Telegram, which is Wassim Bhat Chemistry Official. That is the name of the Telegram channel, if you do not know. Right? If you are not the part of the Telegram channel yet, be the part of the Telegram channel so that you get the handwritten session PDF of this particular session as well. Okay? Perfect, guys. So, without wasting a lot of the time, let's get going. Let's get started with the first question, which is on your screen. My dear students, have a look on the first question, which is on the screen. And the question is from the chapter Electrochemistry. If you look at the question carefully, reduction potential of the ions are given. So we are given with the reduction potentials of these ions. And since we are given with the reduction potentials of these ions, I have to arrange them on the basis of their oxidizing power. Okay. My dear students, one thing which you must know. What is that? Remember one simple statement. Remember, a better oxidizing agent has got more oxidizing power. A better oxidizing agent has got more oxidizing power. And let me tell you, oxidizing agent itself undergoes reduction. Oxidizing agent itself undergoes reduction. I'll say the one that undergoes reduction easily, that is going to be the better oxidizing agent, more oxidizing power. Now, which one undergoes reduction easily? The one that has got more SRP, the one that has got more SRP will undergo reduction easily. Better oxidizing agent, more oxidizing power. Now, compare these SRP values. Which one has got the maximum SRP? I'll say BrO4 negative has got maximum SRP, right? BrO4 negative has got maximum SRP, right? Followed by your IO4 negative, followed by your ClO4 negative. So this is the order of SRP, and you know, more the SRP, more the tendency to undergo reduction, better the oxidizing agent, more the oxidizing power, right? So this is going to be your actual order. So which option is satisfying this condition? Majority is coming up with option B. So absolutely, my dear students, it's going to be option B. That's going to be the correct answer of this particular question. I hope this particular question is absolutely clear to everyone. Yeah? Perfect. My dear students, look at the next question. Look at the next question. The question is from the chapter Coordination Chemistry. The question is from the chapter Coordination Chemistry, right? What do you think is going to be the answer of this particular question? What do you think? Think over it. I'm given with 4 complexes. I have to arrange them, right? I have to match them with the spin-only magnetic moment. Okay? I have to match them with the spin-only magnetic moment. And spin-only magnetic moment is given by the formula under root of n into n plus 2 Bohr magnetrons. I hope you know this. Root of n into n plus 2 Bohr magnetrons. Is that? Is that? Yes? What do you think is going to be the answer of this particular question? Quickly, in the chats, everyone. My dear students, if you look at the first complex, here chromium is in +3 oxidation state, right? Here chromium is in +3 oxidation state. What is the outermost configuration of chromium? You must be knowing it is 3d5 4s1. But if I write chromium tripositive, so take 3 electrons out, 1 from here and 2 from here, it becomes 3d3 configuration. 3d3, 4 is 0, 4p0, right? Perfect. 3d3, 4 is 0, 4p0 and 4d0, right? Now, my dear students, here you have got 3d3 configuration. Here we have got 1, 2, 3. 3d3 configuration. 4s has got 0 electrons. 4p has got 0 electrons. Even 4d has got 0 electrons. Perfect. So, if you look carefully, what is the coordination number in here? Coordination number is 6. If the coordination number is 6, I must say, this central metal ion, it has to arrange some 6 vacant orbitals for hybridization. And what are those 6 vacant orbitals? 1, 2, 3, 4, 5, 6. So this one is D2sp3 hybridized. D2sp3 hybridized, right? At the same time, if I ask you how many unpaired electrons are there, there are 3 unpaired electrons. So the value of n is 3. If the value of n is 3, the spin magnetic moment is going to be under root of n into n plus 2, right? 3 plus 2 is 5, 5 3s are 15, so root of 15. Perfect. Root of 15 will be almost how much? 3.87. So you are matching A with 2. You are matching A with 2. So either option 2 or option 3. Now look at the second one. This is nickel dipositive here. This is nickel dipositive here. Perfect. Right? Now, whether you are going to match this B with 3 or 4, what do you think? Whether you are going to match this B with 3 or 4. First of all, if you look carefully, it's a weak field ligand. And weak field ligand does not do any sort of pairing. So there will be unpaired electrons. If there is unpaired electrons, there will be spin magnetic moment, right? So it's not going to be 0, it's going to be 2.83. So B you are matching with 4. So it is basically going to be option 3. That is going to be the correct answer of this particular question. I believe it's clear to everyone. Okay. My dear students, moving on to the next question. Look at the next question. Next question is basically from the chapter Chemical Equilibrium, right? It is from the chapter Chemical Equilibrium. There are some reactions that are given to us. Their equilibrium constants are given to us. And I'm supposed to get the equilibrium constant for this particular reaction. I'm supposed to get equilibrium constant for this particular reaction. How do we exactly do this? First of all, whenever you get this sort of a question, remember that particular reaction whose equilibrium constant is to be calculated. You're supposed to make that reaction out of the given reactions. You're supposed to make that reaction out of the given reactions. Now, how do we make this reaction out of the given reactions? For that, what I'll be doing, I'll be reversing the first reaction first. When I reverse the first reaction, it becomes 2 times NH3. It gives N2 plus 3 times H2. Since I've reversed the first reaction, its equilibrium constant will be inversed. So its equilibrium constant now is 1 by K1. Okay. Now, my dear students, second reaction, I'm going to write as such. Second reaction, I'll be writing as such, which is N2 plus O2 gives what? It gives 2 times NO. I've written the second reaction as such, so its equilibrium constant is K2. Okay. The third reaction, I'll be multiplying the third reaction by number 3. When I multiply this reaction by number 3, it becomes 3 times H2 plus 3 divided by 2 times O2. It gives me what? It gives me 3 times water. Since I have multiplied this reaction by number 3, this 3 goes to the power of equilibrium constant, which is now K3 raised power 3. Okay? And people, when you add these three reactions, you'll be getting this particular reaction. When you add these three reactions now, you'll be getting this reaction. And on adding the reactions, equilibrium constants are multiplied. So the new equilibrium constant is going to be 1 upon K1 multiplied by K2 multiplied by K3 raised power 3. So which option satisfies it? It's K2, K3 raised power 3 divided by K1. So it's option C. That is going to be the correct answer of this particular question, right? Yes? Am I clear, people? Am I clear to everyone? Yeah? Am I clear? Perfect. I'm going to move on to one more type of the question. Look at it carefully. What do you think is going to be the answer of this particular question? Quickly in the chats. Quickly in the chats. I would want to know the answer in the chats first. Give it a try. You have got MnCN6 tri-negative. So first of all, look at this Mn. It's in tri-positive oxidation state, right? What is the oxidation state of manganese generally? Outermost oxidation state. Sorry, outermost configuration of manganese. It is 3d5, 4s2, right? But manganese right here is in +3 oxidation state. So take 3 electrons out, it becomes 3d4. 3d4, 4s0, 4p0, 4d0, right? Perfect. Now, my dear students, if you look at this 3d4 configuration, 1, 2, 3, 4, 5. So 1, 2, 3, 4. 4s is having 0, 4p having 0, and 4d having 0 as well. Okay? 4d having 0 as well. Now, my dear students, look at the given scenario. Over here, if you look at this cyanide, Cn negative, right? Is it a weak field ligand or strong field? It's a strong field ligand, right? If it's a strong field ligand, it's going to cause the pairing. If it is going to cause the pairing, one of the electron will shift here, right? When one of the electrons shifts here, what is going to happen? It becomes something like this. 1, 2, 3, and 4, right? 1, 2, 3, and 4. And after that, you have got S. After that, you have got 3p over here. I mean 4p over here. And then you have got 4d as well. Perfect. So, if you look at the coordination number here is 6. So, you know, your central metal ion, it has to arrange 6 vacant orbitals for hybridization. That's going to be 1, 2, 3, 4, 5, 6. So, the hybridization involved is simply going to be D2sp3, right? It is going to be D2sp3. I believe this particular question is clear to everyone, right? Am I clear, people? Am I clear? Am I clear? Look at the next one. Look at the next question, guys. Look at the next question carefully. Look at the next one. The question is the right option for the mass of carbon dioxide produced on heating 20 grams of 20% pure limestone. So first of all, you have got 20 grams of 20% pure limestone. What does that mean? That means what is the mass of pure calcium carbonate in the sample? It's going to be 20% of 20 grams. The value comes out to be 4 grams. So basically, we have got 4 grams of pure calcium carbonate. And when these 4 grams of pure calcium carbonate will be heated, it will be giving calcium oxide. And with that, you'll be getting carbon dioxide gas. So technically, we are going to heat up 4 grams of calcium carbonate. Okay. Now, on heating 4 grams of calcium carbonate, what mass of carbon dioxide is produced? That's the question. My dear students, first of all, I've got 4 grams of calcium carbonate. That means how many moles of calcium carbonate do we have? Mass of calcium carbonate divided by molar mass. The value comes out to be 0.04. So we have got 0.04 moles of calcium carbonate, which we are going to heat up. Now, stoichiometry is telling me that 1 mole calcium carbonate upon heating gives 1 mole carbon dioxide. If 1 mole calcium carbonate gives 1 mole carbon dioxide, I'll say 0.04 moles of calcium carbonate will be giving me 0.04 moles of carbon dioxide. So I would say these are the moles of carbon dioxide which are produced. But as per the question, am I supposed to calculate moles of carbon dioxide produced? No. I'm supposed to calculate mass of carbon dioxide produced. So mass of carbon dioxide is equal to moles of carbon dioxide multiplied by molar mass of carbon dioxide. And after solving, this particular value comes out to be 1.76 grams. So I would say 1.76 grams of carbon dioxide will be produced at the end, right? Am I clear to everyone? Quickly in the chats. I hope I'm super clear to everyone. All right. My dear students, this is one more question which is frequently asked nowadays in your JEE Mains examination, right? And it's a simple question. I believe you can easily solve this. What do you think about this one? ICl is more reactive than I2. Is that the correct statement? Absolutely. Your interhalogen, ICl, that will be more reactive than I2. What is the reason? Reason is because ICl bond is weaker than that of II bond. That is the reason. Perfect. So I would say assertion is correct, reason is correct, and reason is also the correct explanation of the assertion. So it has to be option 2. That has to be the correct answer of this particular question. So ICl, that is definitely more reactive than I2. Reason being because ICl bond, that is weaker than that of your II bond. Perfect. All right. Look at this one as well. The question is from the chapter kinetics. What do you think about this one? I have taught you these sort of questions in detail in the marathons. I hope you remember. So what do you think about this one quickly? What do you think about this one quickly? Find out the time required for 2/3 completion of a first order reaction if the value of rate constant is this. My dear students, I'm given with a reaction, for example, that's A gives B. This is the reaction. And this reaction follows the first order kinetics whose rate constant is 4.3 into 10 raised power minus 4 second inverse. Okay. Let's say at time t is equal to 0, the initial concentration of reactant is A0. This has to be 0. Okay. We have to calculate the time taken to complete 2/3 of the reaction. Time taken to complete 2/3 of the reaction. That means time taken, we have to calculate that particular time wherein 2/3 of reactant would have got converted into product. We have to calculate that particular time wherein 2/3 of reactant gets converted into product. My dear students, if 2/3 of reactant gets converted into product, how much reactant would be left at that time? A0 minus 2/3 of A0. This will be A0 by 3. So this is the amount of reactant. This is the amount of reactant left at time t 2/3. Okay. When 2/3 of reaction gets completed, this much amount of reactant would be left. Now, if I use the integrate rate equation says that T is equal to 2.303 divided by K. It is the log of A0 divided by AT, right? So, T 2/3 will be equal to 2.303 divided by, what is the value of K? That is 4.3 into 10 raised power minus 4 log of A0 divided by, what is AT? AT is A0 by 3. So A0 cancels out. So it will be log of 3. And log of 3, you must be knowing, that is 0.47. You just have to solve this equation and you are going to get the time taken to complete 2/3 of the reaction. Am I clear with this? Am I clear with this, people? Am I clear with this? Quickly. So when you solve this question, you'll be getting the final answer exactly as 2.5 unit henries per 3 seconds. This is the time taken to complete 2/3 of the reaction. I hope this is clear. I hope this is clear. All right. This is one more frequently asked question and you should know this. What about this? What do you think? What do you think about this particular question? The number of radial nodes in 3p. The number of radial nodes in 3p. How do you exactly calculate the radial nodes? There is one formula. What is that? That's n minus L minus 1. This is the formula to calculate radial or spherical nodes. What is the value of n here? The value of n here is 3. What is the value of L for p? The value of L for p is 1. So it's going to be 3 minus 1 minus 1. The value comes out to be exactly 1. So there is one radial node in case of what? In case of your 3p orbital. Okay? I hope this is clear. You're supposed to calculate radial nodes, not the total number of nodes. Still, let me tell you, total number of nodes is given by the formula n minus 1. If they had asked about total number of nodes, then n value is 3. 3 minus 1 is going to be 2. This is for total number of nodes. This is for radial nodes. So therefore, angular nodes will be total nodes minus radial nodes. Angular node will be 1. So anything can be asked. Either radial nodes, either angular nodes, or the total number of nodes. Okay? I hope this is clear. I hope this is clear, people. I hope this is clear. All right. What do you think about this particular question? The question is from the chapter Thermodynamics. What do you think? What do you think? What do you think, people? Quickly in the chats. It's a very simple and basic question. In the question, my dear students, they have mentioned for the irreversible expansion. So the process is irreversible. The process is irreversible. For irreversible processes, remember that delta S total, that's greater than 0. For irreversible processes, delta S total, it is greater than 0. Okay? All right, number 1. Number 2, what about delta U? What about delta U? What about delta U? See guys, one thing which you have to know, for any spontaneous process, for any spontaneous and irreversible process, delta S total, that's greater than 0. And since they have mentioned that the ideal gas is undergoing isothermal process, if the ideal gas undergoes isothermal process, its delta U has to be 0. So what is the correct answer here? Delta U has to be 0 and delta S total, that will be greater than 0. That means that will be non-zero. Okay? This sort of a question can also be asked. All right. I'm moving on to one more question. What do you think about this one? These are again the frequently asked questions when it comes to your NEET examination, right? And I have given you a lot of tricks to solve these sort of questions already. For example, you have got XeF6. Let's say you have got XeF6, right? How many valence electrons xenon has? 8. How many sigma bonds xenon is forming? 6 sigma bonds. How many lone pairs will be on xenon? 1. What is the type of the molecule? It's AB6L1. AB6L1 means distorted octahedron. Distorted octahedron. So XeF6 is distorted octahedron. So 1 you have to match with 1. So it's either going to be A or B. It's either going to be A or B. Now if last one seems simple, XeF4, and XeF4 is a very common example, which is going to be square planar. So 4 you have to match with 2. Okay, 4 you have to match with 2. So 4 is matched with 2 here, 1 is matched with 1. So it has to be option A. That has to be the correct answer of this particular question. You are not supposed to check all these things one by one. Just do the work smartly. Look at the options. First one and second one is matching, right? And this one is the differentiating factor. So no need to check the rest of the things. I hope I'm clear with this. I hope I'm clear with this. Now guys, this is one more question that is asked from your chapter atomic structure, and this is frequently asked. This is frequently asked. You guys can easily kill this. My dear students, first of all, whenever you need to compare the radii of two Bohr's orbits, you use the formula R1 divided by R2 is equal to N1 square divided by N2 square. And this is going to be Z2 and this is going to be Z1. Is that? Okay. Perfect. Right? Now, I believe you should be in a position to solve this. You should be in a position to solve this, people. Now, have a look. If the radius of second Bohr's orbit of He positive. So, first of all, we have got He positive. And at the same time, we have got Li di positive. If I call this as number 1, I'm calling this as number 2. Calling this number 1, calling this number 2. Okay. First of all, second Bohr's orbit. So, N1 value is 2. Right? And R1 value is 105.8 picometers. What is the value of atomic number of helium? Hydrogen, helium. That's 2. Okay. What is the radius of third Bohr's orbit? So, N2 value is 3. And R2 value I need to calculate. And atomic number of lithium, hydrogen, helium, lithium, that's 3. So, put all these parameters into this particular equation. And from there, you can easily calculate R2 value. Yes, it's pretty much simple. It is pretty much simple, people. Right? So when you put all these values into this particular expression, you'll be easily getting the value of R2. And after solving, let me tell you, R2 value will come out to be 158.7 picometers. Okay? I believe I'm clear with this particular question as well. All right, one more. Look at this particular question and try to answer smartly. Try to answer smartly. You are not supposed to check all these things one by one. Try to answer smartly, people, in lesser time. We have to arrange them on the basis of their magnetic moments. We have to arrange them on the basis of their magnetic moments, right? Perfect. Since you know your weak field ligands, they do not do the pairing. If they do not do the pairing, there will be unpaired electrons. If there are unpaired electrons, you know more than unpaired electrons, more is going to be the magnetic moment. You know it already. You know it already, right? So first of all, I can see your F negative is a weak field ligand. F negative is a weak field ligand, so it would not have caused any sort of pairing. If it would not have caused any sort of pairing, there'll be unpaired electrons. And there'll be more unpaired electrons in case of Fe tripositive than that of cobalt tripositive. And more the unpaired electrons, more the magnetic moment, right? Perfect. So the correct answer is directly going to be option C. No need to check all the things one by one. I hope I'm clear with this. I hope I'm clear with this. This is Fe tripositive. If you talk about Fe tripositive, iron is 3d6 4s2, right? Iron is 3d6 4s2. Now this tripositive take 3 electrons out. When you take 3 electrons out, it becomes 3d5. 3d5 means there are 5 unpaired electrons. Talk about cobalt. Iron cobalt, 3d7 4s2. But this is again cobalt tripositive. Take 3 electrons out. When you take 3 electrons out, it becomes 3d6. In case of 3d6, N value is 4. 4 unpaired electrons. More than unpaired electrons, more the magnetic moment. As simple as that. Bio lecture will be available in some time, okay? It happens. It happens. Bio will be available in some time. Right? Okay? I hope I'm clear. What do you think will be the answer of this particular question? What do you think will be the answer of this particular question? Quickly. Quickly, guys. Let me know in the chats. This is the question which easily you should be in a position to do. The first ionization energies of sodium, magnesium, after magnesium what comes? Aluminium and silicon are 496, 737 and 786 kilojoules per mole respectively. What will be the value of the ionization energy of aluminium? Tell me one thing. What about magnesium and aluminium? Will magnesium have more ionization energy than that of aluminium? Absolutely, magnesium has got more ionization energy than that of aluminium. Perfect. So, why is that? Why is that? Because in case of magnesium, the outermost configuration is 3s2. 3s2. In case of aluminium, the outermost configuration is 3p1. It is 3p1. In case of magnesium, the outermost configuration is 3s2. Here it is 3p1. This is more stable configuration. To take electron out from here, more energy is required. So magnesium has got more ionization energy than that of aluminium. So remember, aluminium will come in between these two values. Aluminium's ionization energy will come in between these two values. So which value lies between 496 and 737? Which value lies between 496 and 737? It is this particular value. So the answer is going to be option C. That has to be the correct answer of this particular question. I believe it's clear to everyone. I believe it's clear to everyone. Okay. All right. What about this particular question? It's again simple. My dear students, it's again simple. What do you think about this? What do you think about this particular question? The strongest reducing agent among the following. These are the hydrides of your group 15. These are the hydrides of your group 15, my dear students. And let me tell you, in case of hydrides of group 15, the reducing character, the reducing nature, that increases. So which one is going to be maximum reducing among all? It's going to be BiH3. Okay. Remember this particular thing as well. These are again your frequently asked questions when it comes to your NEET examination. All right. This sort of a question from your concentration terms, even it is frequently asked. What do you think about this particular question? What do you think about this particular question? What do you think about this particular question? Quickly. Quickly, my dear students. See, molality of 3 molar aqueous solution of NaCl is, density is also given. If you remember, I had given you one direct result between molality and molarity. If you do not remember, remember it from now onwards. Molality is equal to 1000 multiplied by molarity of the solution divided by 1000 multiplied by density of solution minus molarity multiplied by molar mass of solute. This is the equation that relates molality and molarity. Okay, this is the equation that relates molality and molarity. Now, as per question, molarity is given, density of solution is given, molarity is given, molar mass of solute, molar mass of NaCl is 58.5. Put the values and get the result, get the molality. So whenever you have this sort of a question, people, you should be using this direct equation and try to solve this particular question. Okay. All right. I hope this is also clear. I hope this is also clear. Okay, one more question, which I believe every one of you can easily solve. Think over it. Think over it, my dear students. Think over it. Think over it quickly. The correct increasing order of the bond angles. Bond angles. If you look at the type of these molecules, see, BF3 is of the type what? It's AB3L0 type. AB3L0. This one, this is AB3L1 type. And this is AB3L2 type. These are the types of the molecules. And you must be knowing, if this is AB3L0, hybridization is going to be sp2. And the hybridization is going to be sp3, right? And the hybridization is going to be sp3d first of all. Okay? Correct? If you look at their shapes, AB3L0 is going to be what? It's going to be trigonal planar. This is trigonal planar. AB3L1 is going to be pyramidal. And AB3L2 is going to be T-shaped. First of all, in case of planar ones, the bond angle is 120, right? In case of T-shaped molecule, the bond angle is going to be 90, right? And this particular one, it's going to be between these two. So which one is the correct one? Which one will have maximum bond angle? Maximum bond angle is going to be for BF3, right? Is anyone showing BF3? Yes, only the first one. So directly go for the first one. No need to check the rest of the things, right? As I told you, you have to answer smartly in the exam. It should not take a lot of time. What about this one? Which of the following pairs is isoelectronic and isostructural? Isoelectronic and isostructural. Isoelectronic and isostructural. First of all, what you need to do, my dear students, as far as I can see the options, the option has to be 4. Why is that? Why is that? If I talk about your IBr2 negative, right? Iodine has got how many valence electrons? 7. There's a negative charge, so 7 plus 1 makes it 8. How many sigma bonds are there? 2, right? How many lone pairs will be there on iodine? There will be 8 minus 2, 6. That means there are 3 lone pairs on iodine. The type of the molecule has to be AB2L3. The type of the molecule is going to be AB2L3. Okay. Now, look at the type of this particular molecule, XCF2. It's again going to be AB2L3 only. Remember those molecules which have got the same type. Those molecules which have got the same type, they are going to be isostructural. They are going to be isostructural. So it is definitely going to be the last option that has to be the correct answer of this particular question. I believe I'm clear. I believe I'm clear. Look at the next one, my dear students. You know, when you talk about Kp relation, Kp is equal to Kc RT raised power delta Ng, right? You know that already. So how to get the value of Kp divided by Kc? Kp divided by Kc is going to be RT raised power delta N. RT raised power delta N. So first of all, get the value of delta N for this particular reaction. How do you calculate delta Ng? Number of moles of gaseous products, which is 1, minus number of moles of gaseous reactants, which is 1 plus 1 by 2, that is 3 by 2. The value comes out to be minus 1 by 2. Okay, so it's going to be RT raised power minus 1 by 2, which is going to be simply 1 divided by root of RT. Perfect. So Kp divided by Kc is nothing. That's 1 divided by root of RT. So it is definitely going to be option 4. That has to be the correct answer of this particular question as well. These sort of questions are also frequently asked. So have a proper eye on these questions. Okay. Now, my dear students, this is one famous question. The outermost electronic configuration of gadolinium. Okay. I believe all of you would be remembering it directly because it has got exceptional configuration. You know it. You should know it. You study this in your DNA block elements as well, right? You study this in your periodic classification as well. So what do you think about this particular question? What is going to be the answer of this question? Quickly in the chats. Quickly in the chats. Everyone. Everyone. All of you should answer this at least. Because gadolinium, it has got the exceptional electronic configuration. You know it. So this particular question, I would want you guys to remember directly from now onwards. Gadolinium's outermost electronic configuration is 4f7, 5d1, 6s2, right? Just remember this, 4f7, 5d1, 6s2. Okay, this is a lot of times asked in different examinations. Okay, so remember this directly. Remember this directly. This sort of a question, I believe all of you can easily solve. What do you think? What do you think about this one? I'm looking at your chats because this is something which is very basic. Cadmium, chromium, vanadium, titanium, manganese. Quickly, you have to arrange them on the basis of their increasing number of unpaired electrons. If I talk about scandium, scandium is 3d1 4s2. Titanium, 3d2 4s2. Vanadium, 3d3 4s2, right? Chromium, 3d5 4s1. Manganese, 3d5 4s2. How many unpaired electrons do you see here? There is one unpaired electron, which is in D. There are 5 unpaired electrons here. All the 5, sorry, 3d5 4s1. That means there are 6 unpaired electrons here. 5 here, 1 here. In case of this particular configuration, D3. D3 means 3 unpaired electrons. S is completely filled, right? D2, 2 unpaired electrons, right? So you have checked all the unpaired electrons. In case of manganese, there are 5 unpaired electrons. So you can easily select your options now. You can easily select your options now. What do you think is the correct answer now? Which of the following has got maximum unpaired electrons? That's chromium. That's option B. That's option B. So option C has to be the correct answer of this particular question. Okay? I hope I'm clear with this. There's only one option that's saying that. That's option B. That's option C, sorry. All right. This is again a frequently asked question, guys, and a simple one. And you love these sort of questions. I know that already. What about this one? All these are your isoelectronic species. All these are your isoelectronic species. And in case of isoelectronic species, how do you talk about the size? When you talk about the size of isoelectronic species, remember, size is directly proportional to negative charge, inversely proportional to positive charge. Simple. More the negative charge, more the size. So where is the negative charge maximum? N tri-negative. So N tri-negative will have maximum size, then O di-negative, F negative, Na positive, Mg di positive. Directly option A, which is going to be the correct one. So remember this particular one. Size, in case of isoelectronic species, is directly proportional to negative charge, inversely proportional to positive charge. Yes? Right? Just remember this statement and we are done. We are sorted. All right. This sort of a question is also asked. If you look at the previous 2-3 year JEE mains examinations, in all the shifts, I must say, in majority of the shifts, this sort of a question has been asked. And it's a pure NEET standard question. It's a pure NEET standard question. The pKb of dimethylamine, which is the weak base, and pK of acetic acid, which is a weak acid, right, are this and this. The correct option for pH of dimethyl ammonium acetate. So it is basically a salt of weak acid and weak base. It's a salt of weak acid and weak base. And you should know, when salt of weak acid and weak base undergoes hydrolysis, how do we calculate pH? pH formula is simply 7 plus half of, it is P of Ka minus P of Kb. When the salt of weak acid and weak base undergoes hydrolysis, how do we calculate the pH of the resulting solution? It's 7 plus half of P of Ka minus P of Kb. And all the things are given here. So it's 7 plus half of. What is pKa value? pKa value as per the question is given to me as 4.77. pKb value is 3.27. So it is 7 plus 1 by 2. This value will come out to be 1.5. 1.5 divided by 2 is, so this is 7 plus 0.75. So the final answer is nothing but 7.75. So pH of the final solution is nothing but 7.75. Am I clear with this? Am I clear with this question also? Just remember this particular formula. Try to solve this question in very, very, very lesser time. It's a basic question asked many a times. It's asked many a times. Sir, one word in Kashmiri. Once a Kashmir pad kya shi bozun se jaldi answers mein. Okay, look at this particular question. Which of the following molecules is non-polar in nature? Okay, my dear students, remember I'm giving you directly some set of molecules. Whenever you see molecule of the type Ab2L0, you see molecule of the type Ab3L0, you see molecule of the type Ab4L0, you see molecule of the type Ab5L0, you see molecule of the type Ab6L0, you see molecule of the type Ab3L2 or sorry Ab2L3, you see molecule of the type Ab4L2. All these molecules, whenever you see molecules of this particular category, their net dipole moment has to be 0, and if their net dipole moment is 0, they are going to be non-polar. So basically, technically, you have to check the types of these molecules. That's it. That's it. Now, for example, look at this one, SbCl5. SbCl5. Sb belongs to group 15. So how many valence electrons? 5 valence electrons. How many sigma bonds? 5 sigma bonds. How many lone pair on antimony? 0, right? Type of the molecule is nothing but Ab5L0. If the type of the molecule is Ab5L0, that means the molecule has to be non-polar in nature. That's all. Right? That's all, people. So just remember this particular part and we are done with this particular question as well. And I hope easily you can solve this sort of a question from now onwards. One more theoretical question, which all of you, I'm pretty much sure, can easily solve. What do you think? Anomalous behavior of oxygen is due to anomalous behavior of oxygen, anomalous behavior of fluorine, right? What is the reason for that? Reason is their small size and more electronegativity. Reason is their small size and more electronegativity. Because of these two factors, small size and more electronegativity, your oxygen in its group shows anomalous behavior. Similarly, fluorine in its respective group shows anomalous behavior. Reason is the small size and more electronegativity. Remember this particular one as well. Okay. What about this one? What about this particular question, guys? What about this particular question? 4.5 moles of hydrogen and iodine is heated in a sealed 10-liter vessel. At equilibrium, 3 moles of HI were found. Okay. The equilibrium constant is. So basically, I'm giving you the reaction. The reaction is H2 gas plus I2 gas in equilibrium with 2 times HI. Okay. As per the question, we are starting with 4.5 moles each of 4.5 moles each of H2 and I2. Perfect. Now, my dear students, at equilibrium, let's assume Let's assume at equilibrium, out of 4.5 moles of H2, let's say X moles have reacted. So I'll be left with 4 minus 6. Here also I'll be left with 4 minus 6. 1 mole gives 2 moles. So X are going to give us 2X moles. So these are the moles of reactants and products in the container at equilibrium. And if these are the moles of reactants products at equilibrium, as per the question, they have mentioned that 3 moles of HI were found at equilibrium. Number of moles of HI at equilibrium are given to me as 3. But we know number of moles of HI at equilibrium is nothing but 2x. So 2x is basically 3. So x is 3 by 2. That's 1.5. So I got the value of x. If x is 1.5, 4.5 minus 1.5 is 3. 4.5 minus 1.5 is 3. 2x value directly is 3. So these are the moles of reactants products at equilibrium. If you got the moles, divide these moles by the total volume. Total volume is 10 liters. So what did you get? You got the concentrations. You got the concentrations of reactants as well as products at equilibrium. And once you get the concentrations of reactants products at equilibrium, you can easily write the Kc expression, which is concentration of HI, 3 by 10, raised power stoichiometric coefficient, that's 2, divided by concentration of H2 at equilibrium, that's 3 by 10, raised power its stoichiometric coefficient, that's 1. Concentration of I2 at equilibrium, 3 by 10, raised power stoichiometric coefficient, 1. So the overall value comes out to be 1 only. So it has to be option 2. That is going to be the correct answer of this particular question, and expect this sort of a question in your upcoming NEET examination as well. Okay. All right. My dear students, look at this question as well. Look at this particular question as well. Look at the question carefully. The volume of hydrogen liberated at STP by reacting 2.4 grams of magnesium. 2.4 grams of magnesium with excess of HCl. Just tell me one thing. When magnesium reacts with HCl, what does it form? It forms MgCl2. With this, you get H2 gas. It's a balanced chemical equation if you see carefully. As per the question, how many grams of magnesium are participating in the reaction? 2.4 grams of magnesium are participating. So that means how many moles of magnesium are participating? Mass of magnesium divided by molar mass of magnesium. So in the reaction, 0.1 mole magnesium is participating. Now stoichiometry is telling us that 1 mole magnesium gives 1 mole H2. Stoichiometry tells us that 1 mole magnesium gives 1 mole H2. If 1 mole magnesium gives 1 mole H2, therefore 0.1 mole magnesium will give us 0.1 mole H2. So we got the moles of H2 produced. We got the moles of H2 produced. But as per the question, are we supposed to calculate the moles of H2 or the volume of H2? I'm supposed to calculate volume of H2. So volume of H2 has to be nothing but moles of H2 multiplied by 22.4. The value comes out to be exactly 2.24 liters. So this is the volume of H2 which is produced at the end of the reaction. Yes? Am I clear, my dear students? Am I clear till here? Quickly in the chats. Yes? All right. Okay. One special compound. Chromium chloride. The oxidation state of chromium is chromium chloride. Tell me the oxidation state of chromium in case of your chromium chloride. This is something which is asked frequently. What do you think about this one? What do you think about this one? Quickly, guys, you should know the structure of your chromium chloride. This is CrO2 and Cl2, right? So due to this double bond, chromium gets +2. Due to this double bond, chromium gets +2. Due to this single bond, +1. Due to this single bond, +1. So what is the total charge on chromium here? It's +6. So +6 is the oxidation state of chromium in case of your chromium chloride, right? It's a special example. Remember this. All right. Look at this particular question. Look at this particular question. A solution is prepared by adding 2 grams of X, by adding 2 grams of X with 1 mole of water. 1 mole of water means 18 grams of water. So basically in the solution, what do we have? In the solution, we have 2 grams of X and 18 grams of water. What am I supposed to calculate? Mass percentage of X. So mass percentage of X in the solution will be mass of X divided by total mass of the solution multiplied by 100. So this is 1, this is 10. The value comes out to be 10%. So mass percentage of X in the final solution is nothing but that is 10%. So option 4 has to be the correct answer of this particular question. Okay. I believe this is also super clear to everyone. Okay. Even this sort of a question, guys, again a question from the chapter Chemical Kinetics. Again, this sort of a question is frequently asked. You should have a proper eye on this sort of a question as well. You are given the first order reaction. Let's say this first order reaction is A gives B. Its rate constant K is equal to 10 raised power minus 2, second inverse. Okay. Let's say at time t is equal to 0, the initial amount of reactant is how much? 20 grams. Now, how long will it take for the reactant to reduce its amount from 20 grams to 5 grams? So this is the value of 80. How long will it take for the amount of reactant to reduce from 20 grams to 5 grams? Now, my dear students, again, it's a first order reaction. So directly you can use the first order kinetics. It's integrated rate equation. You can say T is equal to 2.303 divided by K. It is going to be log of A naught divided by 80. So T is equal to 2.303 divided by K is 10 raised power minus 2. Log of A naught is 20, 80 is 5. So 5, 1, 5, 4. Log 4 is 0.6. So it is 2.303 divided by 10 raised power minus 2 multiplied by 0.6. Solve this and get the answer. You're done. You're sorted. Right? This sort of a question should be done as well. And its final answer will come out to be 138 seconds over here. All right, people. Look at this particular question. Look at this particular question. For the chemical reaction A plus B gives products, the order is 1 with respect to A and B. The order is 1 with respect to A and B. What is the value of X and Y? Can you solve this question? I've solved these questions many a times, right? I've solved these questions many a times. So you are given with the reaction A plus B gives products. So when I write the rate law is going to be R is equal to K, concentration of A raised per order, concentration of B raised per order. Right? So order with respect to A as well as the order with respect to B. Order with respect to A as well as the order with respect to B is 1 each. Okay? Is 1 each. Now, my dear students, what you need to do exactly? These are some values. These are some experiments which have been carried out. And as per experiment number 1, what is the rate? What is the value of R? That's 0.1 is equal to K. What is the concentration of A? That is 20. Right? Raised power 1. What is the concentration of B? That is 0.5. Calling this is equation number 1. Put the experiment number 2 data in the same equation. What is the value of R? That is 0.4 is equal to K. A concentration is X and B concentration is 0.5 in the experiment number 2. In the experiment number 3, this is 0.8, this is K, this is 40 and this is Y. Right? You have got 3 equations. 3 equations. Right? From these 3 equations, you just have to get the value of X and Y. Right? You just have to get the value of X and Y. Perfect. So tell me the answer, people, quick. Tell me the answer quick. Tell me the answer quick. See, guys, even without solving these equations, you can solve this question. Even without solving these equations, you can solve this question. Why is that? If you look at first and second experiment, B concentration is kept same. B is kept same. Okay. Rate is increasing 4 times. Rate is increasing 4 times. That means, since power of A is 1, that means concentration of A would be increased 4 times. Therefore, rate would be increased 4 times. So when concentration of A increases 4 times, 20 into 4 comes out to be 80. So X value has to be 80. X value has to be 80. Now the value of Y. Now the value of Y. Now the value of Y. Can you solve it quickly? Can you solve it quickly, guys? Now the value of Y. In order to get the value of Y, have a look properly what I'll be doing. Have a look properly what I'll be doing, people. When you go from first to fourth, first to third, rate is increasing 8 times. Rate is increasing 8 times. Why is that? Because concentration of B would be also increasing 8 times, right? Concentration of B would be also increasing 8 times, therefore rate would be increasing 8 times. So 8 5s are 40, that means 4. Y value has to be 4, right? So which one is the option? It has to be option 4. That has to be the correct answer of this question. This is one way of solving. Or you have already 3 equations. Divide 1 and 2, divide 2 and 3, get the value of x and y. That's it. Okay. Did I do some mistake? Just a second, just a second. Did I do some mistake here? Okay, yes, we have done one mistake. We have, okay, concentration of A was not constant here, sorry. Concentration of A was not constant here. Concentration of A was also changing, right? This was changing 4 times. This was changing 4 times. So if you look at it, R is equal to K, concentration of A raised power 1, concentration of B raised power 1, right? This was doubling. You were doubling this already. You were doubling this A already. And what is happening to rate? Rate is increasing R times. So if this is doubling, then this has to go what? This has to go 4 times. Then only it becomes 8 times. So if this is increasing 4 times, then 0.5 multiplied with 4 is equal to 2. So Y value is equal to 2. Sorry, my bad. Okay. Anyways, you can solve the question by solving these 3 equations as well. Okay. I thought the concentration is constant here for A. That's the reason why I got 4. Anyways, moving on to the next question. Moving on to the next question. Tell me, how do you solve this particular question? How do you solve this particular question? What do you think? How do you solve this particular question? What weight of glucose must be dissolved in 100 grams of water to lower the vapor pressure by 0.2 mm of Hg? Okay. So lowering in vapor pressure has to be 0.2 mm of Hg. Vapor pressure of pure water is 54.2, right? Molar mass of glucose is 180. My dear students, the question based on colligative property, first colligative property, relative lowering. And as per the formula, you know, P naught minus PS divided by P naught. For a dilute solution has to be moles of solute divided by moles of solvent. Moles of solute divided by moles of solvent. This is one formula which you would have studied. Now P naught minus PS value. This lowering in vapor pressure is 0.2 divided by vapor pressure of pure solvent is given to me as 54.2 is equal. Number of moles of solute will be mass of solute divided by molar mass of solute. That means molar mass of glucose. Na, mass of solvent, means mass of water divided by molar mass of water. Look at this particular equation. One equation, one unknown. One equation, one unknown. You just have to get the value of WB over here. That will give you that mass of solute which is to be added in order to lower the vapor pressure by how much? In order to lower the vapor pressure by 0.2 mm of Hg. Okay. This is again one important question, my dear students, which all of you must be remembering from now onwards. Okay. Look at this question as well. Look at this question as well. Time taken to complete 87.5% and time taken to complete 50% of the reaction. What is the relation between the two? Remember something directly. For the first order reactions, my dear students, remember, time taken to complete 75% of the reaction is nothing but 2 times the half-life. This is the first expression which is valid for the first order reactions. Second, time taken to complete 87.5% of the first order reaction is equal to 3 times the half-life. Remember this as well. Time taken to complete 99.9% of the reaction is also 10 times the half-life. So I was supposed to get the relation between this and this. So I will say T 87.5 is equal to 3 times the T half. So X value here has to be 3. Direct answer. Well, these expressions you derive basically. But right now we do not have the time to derive things. So it's better to remember these 3 expressions. But these 3 expressions are valid only for the first order reactions. Okay, valid only for the first order reactions. So remember them as well. These are super important. These are super important. Look at this one, guys. Look at this one. When a hydrocarbon A undergoes complete combustion, it requires 11 equivalents of oxygen and produces 4 equivalents of water. What is the molecular formula of A? So first of all, your hydrocarbon is CXHY. When it undergoes combustion, it gives carbon dioxide and it gives water. I believe you already know how do we balance this particular reaction. This is X times, this is Y by 2 times, this is x plus y by 4 times. This is how you balance this particular reaction. Now, as far as the question is concerned, when a hydrocarbon A undergoes complete combustion, it requires 11 equivalents of oxygen. 11 equivalents of oxygen it requires. That means x plus y by 4 is equal to 11. And produces 4 equivalents of water. That means y by 2 is equal to how much? Y by 2 is equal to 4, which means that y is equal to 8. So I got y. Put the value of y here. So x plus 8 by 4 is equal to 1. That means x plus 2 is equal to 11. So x value has to be 9. So x is 9, y is 8. Okay, x is 9, y is 8. If x is 9, y is 8. So the formula has to be C9H8. C9H8 has to be the molecular formula of the compound. So this is how you solve this particular equation as well. Okay? Am I clear with this? Am I clear with this, people? Quickly in the chats. This is one more question which is also asked frequently from the thermochemistry part. My dear students, you are given with a reaction and you have to calculate enthalpy of the reaction. You have to calculate enthalpy of the reaction. And you should know enthalpy of reaction is nothing but enthalpy of formation of products minus enthalpy of formation of reactants. So start with the product. Its stoichiometric coefficient is 1. So 1 multiplied by enthalpy of formation of carbon dioxide plus 4 multiplied by enthalpy of formation of HCl. This is the enthalpy of formation of products minus enthalpy of formation of reactants. So 1 multiplied by enthalpy of formation of CCl4 plus 2 multiplied by enthalpy of formation of H2. Okay, perfect. All the parameters as per the question are given to us. So I'll say delta H0 R is equal to enthalpy of formation of carbon dioxide is given to me in the question. Enthalpy of formation of HCl is also given to me. Enthalpy of formation of CCl4 is also given to me. Enthalpy of formation of water is also given to me. So all the parameters are given here. Put all the parameters into this equation and get the value of enthalpy of the reaction under standard conditions. That's it, people. Right? That's it. A simple question, just remember the formula. Enthalpy of reaction is nothing but enthalpy of formation of product minus the enthalpy of formation of reactant. I hope I'm clear with this. I hope I'm clear with this. All right. One more important question, right? And these sort of questions are also asked. What do you think about this one? I think this question we solved a few days back also when I took the marathon. Quickly, what do you think about this one? What do you think about this one? Quickly. My dear students, as per the question, the light of wavelength 400 nanometers is falling on these metals. And you have to check where photoelectric effect is going to happen. Okay. So first of all, what is the incident energy? What is the energy carried by light? It is Hc divided by lambda. Hc value is 1240 electron volt nanometer divided by lambda is nothing but 400 nanometer. This value will be 4341. So 3.1 electron volt. Right? So incident energy is how much? 3.1 electron volt. When 3.1 electron volt falls on lithium, whose work function is 2.42. Remember, for the photoelectric effect to happen, energy of incident light should be greater or equal to work function. So 3.1 is greater than this, so it will show. 3.1 is greater than this, it will show. 3.1 is greater than this, it will show. 3.1 is not greater than this, it won't show. Right? So there are three metals basically which are showing the photoelectric effect among these. Right? There are three metals which are showing the photoelectric effect among all these metals whose work functions are given. And my dear students, this is the criteria for the photoelectric effect to happen. Energy of incident light should be greater or equal to what? It should be greater or equal to work function. I hope I'm clear with this. This is again one simple question which is to be solved in less than 10 seconds, I believe. Which of the following are intensive properties? Intensive properties. Quickly. What are intensive properties? The properties of the system whose value does not change. The properties of the system whose value does not change on changing the size of the system. See, volume. It changes when you change the size of the system, so it's extensive. Mole read capacity, intensive. Molarity means concentration, that's intensive. EMF, intensive. Gibbs free energy, extensive, right? Molar mass is also intensive. Nothing to do with the amount of substance. So 1, 2, 3, 4. There are basically four properties which are intensive in nature among these, right? So the answer has to be 4. Okay, I hope I'm pretty much clear with this as well. All right. What do you think about this particular question? What do you think about this particular question? These are like basic questions which are asked. You have got a metal ion whose magnetic moment is 4.9. Magnetic moment is nothing but under root of n into n plus 2. When can be under root of n plus n? When can be under root of n into n plus 2? 4.9. When the value of n is how much? When the value of n is how much? Quickly. When the value of n is 4. Because then only 4 into 4 plus 2. 4 plus 2 is 6. 6 4s are 24. Root of 24 comes out to be 4.9. So n value has to be 4. There should be 4 unpaired electrons. There should be 4 unpaired electrons, people. Okay? There should be 4 unpaired electrons. Solve this smartly. Solve this question smartly. No need to do a lot of things here. 2 complexes are given. You have to check the magnetic moments. My dear students, first of all, here you have got a weak field again. It is going to act like a weak field. This is going to act like a strong field again. Weak field, strong field. Weak field does not cause pairing. So definitely its magnetic moment is going to be more than that of this one. Okay. First one's magnetic moment is going to be more than that of second one. Perfect. Now how much? This is basically Fe tri-positive. What is the outermost configuration of iron? It is 3d6 4s2. But this is Fe tri-positive. So take 3 electrons out, 2 from here, 1 from here. So it becomes 3d5. It becomes 3d5, 4 is 0. Perfect. So how many unpaired electrons? 5. Because there will be no pairing, weak field. If n is equal to 5, what will be the magnetic moment? Root of 5 into 5 plus 2. Root of 5 into 5 plus 2. That will be 5.92. Right? So answer has to be 4. It has to be the fourth option, which has to be the correct answer of this particular question. In simple way. In simple way. Which hydride among the following is less stable? What do you think? Which hydride among the following is less stable, people? You should know this one as well. And what do you think about this one? What do you think about this one? Quickly, guys. Which hydride among the following is less stable? No need to think a lot about this question, guys. This is simple. If you talk about BeF2, it is hypovalent. Its octet is not complete. Its octet is not complete, right? Perfect. The octet of beryllium is not complete here. It is your hypovalent compound. So it is going to be unstable. It's going to be unstable. What about the thermal stability of hydrides of group 15? What about the thermal stability of hydrides of group 15? What about this question? I want answer from everyone. Let me call HSP sir till then. Let me see if he's ready with his questions. Sir, are you ready? In 10 minutes? Okay. So guys, what is the answer of this question? The thermal stability of hydrides of group 15. When you talk about the hydrides of group 15, on moving from top to bottom, thermal stability decreases basically, right? So NH3, that's going to be maximum thermally stable, followed by PH3, followed by ASH3. Okay. All right. This is one more easy question, right? Easy kill question, you can call this. Identify the correct order of standard enthalpy of formation. Basically, when 1 mole of NaI gets formed, how much heat is released? When 1 mole of NaBr gets formed, how much heat is released? When 1 mole of NaF gets formed from its constituent elements, which should be present in their standard states, I'm not going into the details of that. Just tell me, during the formation of their 1 mole, where energy is released the maximum? Where energy is reached the maximum? Quickly. Quickly, guys. What do you think is going to be the answer of this particular question? Quick. See, the size of cation everywhere is same. The size of cation everywhere is same. The size of anion is changing. Lesser the size of anion, lesser the size of anion, lesser the size. Basically, force of attraction between the ions is inversely proportional to size of ions. Force of attraction between the ions is inversely proportional to size. So lesser the size of ions, more the attraction. More the attraction, more the energy released. So NaF would have maximum enthalpy of formation. Okay? Simple basic question, but remember this one. Remember it, guys. Okay, this is, I believe, which you can easily solve by just looking at the question. So what do you think about this? What is the IUPAC name of the following? It is going to be potassium. Potassium. Now come into the coordination sphere. It's going to be trioxylato. Trioxylato. This is going to be cobaltate. Its oxidation state is +3. So it is potassium, trioxylato, cobaltate, oxidation state 3. So what is the answer? Right? It's option 3, option 4, that has to be the correct answer of this question. A simple basic question this is, but you should be in a position to solve this one. Okay? 1 mole of ideal gas is allowed to expand. Just let me modify this question. It is irreversibly. 1 mole of ideal gas is allowed to expand. So you have got 1 mole of ideal gas, which is allowed to expand irreversibly and adiabatically. Irreversibly and adiabatically. So Q is 0. From the temperature of 27 degrees centigrade. 27 degrees centigrade means 300 Kelvin. Okay. The work done is 3 kilojoules. W is equal to 3 kilojoules. The final temperature of the gas is T2 value. I'm supposed to calculate, and CV is also given to me as 20 joules per Kelvin per mole. Okay. I have to calculate final temperature of the gas. The process is irreversibly adiabatic. Since the process is adiabatic, so Q is 0. If Q is 0, delta U has to be equal to W, and it's W irreversible, right? Delta U is nothing but NCV T2 minus T1. W irreversible is given to me as 3 kilojoules, means 3000 joules, right? N value is 1. CV value is 20. T2 value is how much to be calculated? T1 is 300. So this value is nothing but it is 3000, right? So this and this gets cancelled. 2, 1. This is 150. So T2 is equal to 150 plus 300. The value comes out to be 450 Kelvin. The value comes out to be 450 Kelvin. The option is not matching because I modified the question. I made the question as irreversibly adiabatic. That's why these were the options. But the answer of this question has to be 450 Kelvin. But there is something missing, guys. There is something wrong here. What is that? 1 mole of ideal gas is allowed to expand irreversibly and adiabatically. Since it's an adiabatic expansion, right? During the adiabatic expansion, temperature of the gas should decrease. If the initial temperature is 300, how can be final temperature 450? It should be less than this. Did I do any sort of mistake here in solving the question? Just check it out. Q is 0. Delta U has to be W. Delta is equal to Q plus W. Since Q is 0, N value is how much? 1. CV value is 20. T2 is to be calculated. T1 is 27 degrees centigrade. That means 300 Kelvin. Is equal to 3 kilojoules per mole work done. So 3 kilojoules means 3000 joules. All right. So this is 20. This is 150. So T2 has to be 150 plus 300, right? 450 Kelvin is coming. So there is... Okay. Okay. One thing, guys, it's an expansion, right? It's an expansion. And whenever there is expansion, work is negative. So I forgot to check that. W has to be minus 3. Minus 3, right? So minus 3000, right? So it will be how much? It will be how much? It will be 150 Kelvin. Now it's correct? Because how I got to know this, because whenever there is adiabatic expansion, during adiabatic expansion, be it reversible or irreversible, temperature of the system decreases, right? But it was showing increase in temperature. That's why I told you there is something wrong here. All right. So this sort of equation is done. This sort of equation is done. Look at the next one. Aqueous solution freezes at, and aqueous solution freezes at minus 0.186. That means delta TF, depression in freezing point, which is Kf multiplied by molarity, which will be 0.186, right? Kf value is given, so molarity is equal to 0.186 divided by 1.86. So molarity of the solution is 0.1. What am I supposed to calculate? Elevation and boiling point, delta TB. What is delta TB? Delta TB is nothing but Kb multiplied by M. What is Kb? Kb is given to me as 0.512. Molarity of the solution is 0.1, right? Kb is given to me as 0.512 and molarity of the solution is 0.1. So it will be 0.0512, right? This is the elevation in boiling point delta TB. A simple basic question which can be solved just with the help of a formula. Nothing else. Okay, nothing else. Solubility product for a salt. You are given with the salt AB. You are given with the salt AB. This is going to be A positive aqueous, B negative aqueous, right? Let's say its solubility is S. So its concentration is S moles per liter. Its concentration is also S moles per liter. You know it. If I write the KSP expression is concentration of A positive raised per stoichiometric coefficient, concentration of B negative raised per stoichiometric coefficient. So it's S square. So S has to be nothing but under root of KSP, right? And KSP is given to me in the question. How much is that? 4 into 10 raised per minus 8. So it will be simply 2 into 10 raised per minus 4 moles per liter. So this is the solubility of AB, or you can say this is the concentration here. This is the molarity here, which I was supposed to calculate. As simple as that. Okay. All right, people. One more question. Which of the following pair of species have same bond order? Yesterday, I posted one video in which I told you all the isoelectronic species, they have got the same bond order. If you talk about Cn negative, it is a 14 electron species. CO is also 14 electron species. Since they are your isoelectronic, and in case of isoelectronic species, the bond order is going to be same. So bond order here is going to be simply 3 of both of them. Okay. This one is done. All right. What about this one? The volume of HBr. The volume of HBr required to neutralize 10 ml of 0.01 molar BaOH whole twice. So this is the case of neutralization. Your strong acid and strong base, right? First of all, what is the N factor of HBr? That's 1. Volume of HBr is to be calculated. Molarity of HBr is nothing but 0.02. N factor of this is 2. Volume of this is given to me as 10 ml. And its molarity is given to me as 0.01. So first of all, calculate their normalities. N1, N2. Normality is molarity multiplied by N factor. So it will be 0.02. Normality is molarity multiplied by N factor. So it will be 0.2. So use the equivalent concept. N1 V1 has to be equal to N2 V2. Right? What is N1? 0.02. V1 is to be calculated. What is N2? 0.2. V2 value is equal to 10. So V1 is equal to 2 divided by 0.02, which will be 200 by 2. 200 by 2 is 100. Right? Or again some mistake. What am I doing today? Let me check the question. 0.02. This will be 0.02. That is 0. Okay, it's not 0.1. It is 0.01. That means normality would be 0.02 here. Okay, 0.02. So this is 0.02 and V2 is how much? That is 10. So this gets cancelled. V1 value comes out to be 10 ml. What was V1? That was volume of HBr, which was to be calculated. Right? Perfect. Are all these questions clear, guys? Are all these questions clear? Let me know once in the chats. I'm calling your HSP sir to resume the session from here. Just give me a second. Sir, are you ready? Okay, I'm stopping the stream. आप stream start करो वहाँ से. हाँ? Okay, मैं stop कर रहा हूँ.
Hello, 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 hello. Just to let me know, am I audible to all of you? Welcome, Vanakkam, Suswagatam. Joe Bole Sone also Srigal. It's your Baba HL speak. It's just to let me know. Can you hear me? Can you see me? Am I audible and visible to each and every one of you? Let me check out. Everyone, I want to see green hearts in the chat section. And if yes, you can hear me, you can see me. I want everyone that you should smash the like button and mark your attendance in the form of yes, smashing the like button. I want to see green hearts. Hey everyone, yay, bacha party, students, just to let me know. I want to see green hearts if yes. This is your Baba HSP. If your Baba HSP is completely audible and visible to all of you, I want to see green hearts. That's really awesome. That is indicating that yes, 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 kids, yes. Now, what I can see in the chat section that completely I'm audible and visible. First of all, do let me know that are you ready? Now, if you're ready, then the second question is that you want new questions from NCERT lines because this time your NTA will give a little bit tricky questions. I will not say tough questions, but tricky questions. The questions, these questions will be easy, but you will get stuck. You will get trapped. They will try to trap you. Now, some concepts are missing in NCERT, but they have been given in Tamil Nadu State Board. So, aware of this thing that your Baba HSP is going to give you those concept-based questions also. So, they might be missing somewhere in your books and by PYQs, but I will give you even those questions. So, just to let me know if everyone is ready for each and everything. What I'm telling you, the new questions, totally NCERT based. And second thing, what I'm telling you, the things which are missing in your NCERT but given in Tamil Nadu State Board, these also if you want, show me fire. I want to see fire in the chat section. Where is the fire? Where's the Josh? Let me refresh. I have refreshed it. Yeah, do let me know in the chat section with the fire. I want to see fire in the chat section, kids. Show me some fire in the chat section. Everyone. Just to let me know. I want to see some fire in the chat section, kids. Hey, everyone. Where is everyone? Let me see what kids are writing. Yeah. Yes. I can see fire. Now, ready, steady, go. See, I'm having number of things. I'm having printed questions, number of printed questions. See, number of printed questions. But first of all, what I want to say, kids, the thing which will definitely come in your exam, and kids, let me tell you one very, very important thing. If you know your seniors, they will tell you directly the questions will come from here only. Just to let me know, whatever I'm going to give you right now, not this one. I will frame out new question. I will frame out absolutely no new question. But whatever I'm going to write down over here in 2 hours or 3 hours will come in your examination. Yes, I mean this. Whatever I'm telling you, it will happen. So are you ready? I want to see Josh, fire, but you have to respond. You have to give the answer of the questions. You have to write down immediately that, yes, your response should get reflected. I want to see to every action there is equal and opposite reaction. Once I'm giving the question, I want to see immediate answer. You are able to understand what I'm giving you. You want to get more explanation that you have to write down. Okay. If the explanation of the question's answer, you are able to get it, I want to see green hearts. When to show green hearts? If you are 100% satisfied with the explanation, you have to show green hearts. If you're not satisfied with the explanation, you want to get a little bit more explanation. Once again you want to see the explanation with another visualization, then fire. Clear? When to show fire, when to show green hearts. Is it clear? Then only I'm going to start. Concept clear? Once concept is clear, with the concept clarity, what you will show? You are supposed to show green hearts. If not clear, what you will show? Fire. If you are able to understand this thing, then only I will start. Otherwise not. Otherwise it's going to be useless. Otherwise this question and answers marathon is going to be useless. You are able to understand? Yes. If you are able to understand, green hearts. If you're not able to understand, fire. These things you are going to follow. Yes, if you want to follow, if you're going to follow it, green hearts. Yes. Now, here is your question number 1. Question number 1. I will move in the sequence, NCERT sequence. I will follow while giving you questions. Questions will be according to NCERT sequence. NCERT sequence, that means organic chemistry. I will take organic chemistry first. 11th and then 12th, right? Questions will move like this. They will go like this, right? Ready? But you have to give marks. I will move with total handwritten 100 questions. How many questions? 100 questions. Total 100 questions. I'm making a promise. Handwritten 100 questions. And I'm making a promise. All the organic questions will come from here only in your exam. That's my promise. Nalla iruku promise. It must be looking idiyappam, but I'm telling you all the questions of organic chemistry in your NEET exam will come from here. That's my promise. All the questions. Now, what do you want to show for this? Very, very important. Very, very important. Okay. So, ready, steady, go. Now do let me know if you are true by heart. Everyone, if you are true by heart, then take your copy and take your pen. Yes. Write down 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Right now, write down 10 on one page. And let's see how many questions you are able to give the correct answer. You are the judge, you are the students. Right now, you are the judge, you are the student. If you can't do this, it's useless to take this marathon of questions. Take your copy, take your pen. On first page, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. First 10 questions. Then, what's the answer of 10 questions? What's the total out of 10 questions? How much you have got negative marks and positive marks? Just take your copy. Then, it's going to be fruitful. So, here is question number 1. Calcium carbide CaC2 on reaction with water has given, and this will give you that how much perfect revision you have done. You will see the mirror. A is reacting with NaNH2 excess, giving out B. B has given reaction with this in excess, given out C, and C is reacted with H2 PD C, giving out D. Now what is D? I'm giving you the options. What is D? A, option A is trans hex 3-in, B, trans but 2-in, C, butane, D, cis but 2-in. Here is your question. And let me sit in front of the screen and let me check it out. What's your response? Yeah, some people are giving the correct answer. Very good kids. Give positive marks to yourself. That's really awesome. You got correct answer. The correct answer is cis but 2-in. How to do it? Let me tell you. Calcium carbide Ca C triple bond C on reaction with water, it will emit out this acetylene. It will form acetylene. So this is acetylene with two acidic hydrogens. They will react with NH2 negative is a strong base. NH2 negative is a strong base. So two acidic hydrogens will eliminate out, and this will get released in the form of ammonia. So you will get disodium acetylide. Now, methyl chloride will react over here. Methyl chloride. Now you will get but2-ine. Now this is Lindlar's catalyst. Now people, you must be thinking, what is Lindlar's catalyst? I have to studied about Lindlar's catalyst is H2PD, barium sulfate, charcoal, boiling xylene, quinoline. That is extra cheesy, but if it is given, then also it is Lindlar's catalyst, and your answer is cis. Clear to all of you? I've told you, once you're getting it, show me green hearts. If you're not able to understand fire, but you're not following it. And if you're not able to follow the rules and regulations, no dedication is there, no consistency is there. You're not able to follow the rules, what you will do in your life. I've told you, na? I've told you. If you're not a good student, you can't do anything in your life. So once I'm telling you that to show green hearts, then how you will become doctor? You are going to be like God. Is there anyone who is going to explain you, to guide you, to tell you? You are your boss. You have to become boss of others. Now, question number 2. Question number 2. This is your benzene reacting with 6Cl2 anhydrous AlCl3. This question is going to be anaconda question. What do you mean by anaconda question? Let me tell you, anaconda question means everyone will attend it, but more chances of getting negative marks. More chances of negative marks, right? In dark. So you have to tell the product. A option BHC, B option gamexine powder, C option 666, D option hexachlorobenzene, E option all of these. Yes, I want to see your answer in the chats. Everyone, I want to see your answer in the chats. Do let me know. Yay, everyone. What's your answer? So, most of the students are going for D, and you are not trapped. You are not trapped. You are really those who have given answer to be D. That is really correct answer, because it is ESR, electrophilic substitution reaction. So you will get compound like this, and this is hexachlorobenzene. Hexachlorobenzene. Now question number 3. Question number 3. Benzene hexachloride. So I will write down statement into it, find out which statement is incorrect statement, right? Incorrect about this. Incorrect statement about this. A, BHC is aromatic because it is planar and follows Huckel's rule. Follows Huckel's rule. B, this compound is having 6 chlorine atoms. Molecular formula is C6H6Cl6 D, none of the above. None of the above. 4 statements are there. I want to see answer in the chats. Do let me know. I want to see answer in the chats. Everyone, just do let me know. What's your answer? Baba Tillu is going for A. K. Rutik is going for C. Fast, 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 kids, fast, fast. Fast. I want to see fast reaction. Now, the incorrect statement is, now most of the students got trapped. A is the correct answer. Why A is the correct answer? Let me show you. Let me show you. This is HCl, 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 HCl. This is benzene hexachloride. How it is formed? Just see, kids. When benzene it's treated with, this is your benzene C6H6, when it is treated with 3 moles of Cl2, 500 Kelvin sunlight, it is what type of reaction? Free radical addition reaction. Free radical addition reaction. Now this is C6H6 Cl6. All the carbons are sp3 hybridized. So the ring is non-planar. Remember that. Ring is what? And second thing, no pi electrons. So neither it is planar nor it is following Huckel's rule. So it is non-aromatic compound. So this is the best, I would like to say, best trap of the examiner. Once again, if you are able to understand it, green hearts. If you are not able to understand, then fire. I want to see your reaction, kids. It means a lot, kids. Let me sit in front of screen what you're writing, just to let me know. Now, next question, just see. Question is, this is your RCONH2 amide, 1 degree amide. Amide is 1 degree. If I react it with Br2 KOH, what I will get? I will get RNH2. The name of this reaction is Hoffman bromamide degradation. Am I right? Now what question they will ask? Hoffman bromamide degradation. Now question is, which intermediate, which of the following intermediate is not formed during this reaction? Question is, which of the following intermediate is not formed during this reaction? Right, kids, just see. First option, bromamide. Second option, that is B option, this was A, bromamide, isocyanide. C option, carbamic acid. D option, nitrine. Nitrine. Everyone, just to let me know, I'm just in front of screen. These questions holds a hell lot of weightage. They will change your life, kids. Question 3 wrong. One kid is writing question 3 is wrong. Okay, I will come to that. But first of all, B will not form. B, isocyanide, is wrong. This is incorrect. Which of the following is not formed? So this is answer, because not the isocyanide is not the answer. The answer should be isocyanide Cyanate. One kid has written question number 3 is wrong. Let me come back. See, kids. Let me come back. In the chats, one kid has written question number 3. You are talking about this? Who has written it is wrong? It is wrong. Question is wrong. It's not wrong. Remember that benzene hexachloride is the name. Benzene hexachloride is the name, but there is no benzene. Without benzene, this compound doesn't have benzene. Its name is benzene, but it doesn't have benzene. How many of you are getting it? One kid has written it is wrong. It's not wrong. That's why it was a trap, and that's why examiner will give you number of times. This question is clear. Is it clear, kids? Hey everyone, what are you doing? What's going on? I'm doing such a hard work for you people, and you are not able to show green hearts. What are you doing, kids? It's correct. Very good. And those who got trapped, they are still unable to imagine that how much accuracy I am giving you. This is really great. You are getting the clear mirror image. Now, question number 3 we have done. Question number 4 we have done. Question number 5. Question number 5. Let's see how many of you can do this question. Question number 5. The intermediates or the intermediate of Hoffman bromide degradation A set, B set, C set, D set. Okay. Anion of bromamide isocyanate bromamide isocyanate nitrine carbene benzene carbocation. Ready, steady, go. Your time starts now. Vote for this question. Everyone vote for this question. Fast, kids, fast. Hurry up. Hey kids, hurry up. I want to see the answer. C. Kirtuk is going for C. Bavatillu is going for C. Everyone is going for C. What the correct answer is? Shririsha has given the correct answer. Intermediates of Hoffman bromamide degradation. You got trapped, kids. A is the correct answer. Isocyanide is not the intermediate. Carbene is not the intermediate. Carbocation and benzene, they are not the intermediate. How many of you got it correctly done? Kids, this is your situation. So I have to tell you that Hoffman bromamide degradation reaction. What happens during Hoffman bromamide degradation? Just see. In Hoffman bromamide degradation, what happens? Just see. And during this reaction, what happens? Primary amide on reaction with Br2, 4 moles of KOH gives 1 degree amine. There is one song for this reaction. Primary amide has been treated with primary amide has been treated with cobra. Treated with cobra. Cobra means potassium hypobromide. Cobra like cobra. Cobra like cobra. Cobra. Primary amide has been treated with cobra. We got primary amine with 1 carbon stolen, with 1 carbon stolen. Now, what are the intermediates of this reaction? Intermediates: bromamide, bromamide, bromamide. They can start from here also. Once during AIMS exam, they have started from here. Bromamide. Now, anion of bromamide, RCON negative Br. After this, nitrine, RCON nitrine. Now there will be rate determining step, RDS, rearrangement, RN, double bond C, double bond O. Remember that isocyanate, isocyanate, 8, eat, 8, eaten, eat, 8, eaten, 8, 8, 8. Isocyanate, not cyanide. Isocyanate. Carbamic acid, RNHC double bond OOH. One degree. I mean, these are the intermediates. I'm doing it quickly. I'm explaining it. Just remember, nowhere you will get such type of teaching kits. Are you ready for the next question? Are you ready for the next question, everyone? Very good, Baba Tillu. Everyone, I want to see green hearts if you're really liking it. The way I'm teaching, the way I'm explaining the question, the way I'm giving you the new questions kits. Gautam Sri Harindra, wow, wow, great, great, great. Now, next question. Question number. Hey, what's the question number? 6. Which organic compound can be used as solvent during Friedel-Crafts alkylation, during Friedel-Crafts alkylation? A. Toluene, B. Xylene, C. Nitrobenzene, D. Anisole. What's your answer? This is anisole. Let's see how many of you are going to get passed this time. Let me send in front of screen how many of you are going to get passed. Everyone. Yes, yes, Gautam, yes. The correct answer, let's see who. C, very good. C is the correct answer. C is the correct answer. Why, kids? This is inert during Friedel-Crafts, will not show. So it is inert during Friedel-Crafts. Nitrobenzene is having minus M group, and such type of compounds do not undergo Friedel-Crafts alkylation and acylation. Aniline, the answer is, let me write down, aniline and all meta directors do not show FCA, Friedel-Crafts alkylation as well as acylation. So question number 7. Benzene on reaction with this question, the one who is going to solve will be genius. Concentrated HNO3, concentrated H2SO4 is giving out A. A is treated with 10 HCl giving out B. B is treated with NaNO2 HCl giving out C. This is treated with Cu2 Cl2 HCl giving out D, and D is treated with C NaOH 623 Kelvin, 300 atmospheric pressure. H plus is giving out E, and E has been treated with acetyl chloride, has given F. Now the question is total number of covalent bonds in F. Do it. Total number of covalent bonds in F. Meanwhile, I will make the options. You just do it, and I am making options for it. Okay. These are the options. Once if you have done it, then really you will be a very, very good student. I will say yes. I will say yes. You are going to rock. Let me see in the chat section who is going to get the answer for this question. Yes, I'm waiting. I'm waiting. Oh my God, Vishal MH. Your answer is absolutely correct. Those who have given this answer, their answer is absolutely correct. How many? First of all, do let me know how many of you are liking this type of questions. I could have given printed questions, but right now I'm framing out the questions so that you can use your brain. Already the questions which you have done somewhere, MCQs, there your brain is not getting utilized. You are just you're having in the memory the answer. You just see the question and you tick mark, and that's why I'm giving you new questions just to use your brain. First of all, do let me know how many of you are really enjoying these type of questions, this type of revision, because if you are having knowledge, then you must be liking such type of session. But if you haven't done your revision properly, you must be thinking, oh my God, what's going on? What's going on? Show me green hearts if you're really liking it. I want to see green hearts if you're really liking it. Nitration, nitrobenzene, If you can't do these questions, your selection is far away, far away, far away. If you can answer these questions, you are going to get selected. Reduction. Before I do these questions, first of all, tell me, have you seen any organic teacher on any YouTube that framing such type of questions directly? First of all, do let me know in the chat section. Have you seen any organic chemistry teacher on any YouTube channel that the person, the teacher is framing out direct questions on the spot in front of the students? Let me see in the chat section what you are writing. Let me see in the chat section. So, very good. It needs knowledge, experience, and on this channel you are having such a great teacher. So tell everyone that who is over here on this channel, who is there, who is in front of you, kids. Diazotization. Benzene diazonium chloride. Sandmeyer reaction. Chlorobenzene. Dawes process. Phenol. Oh, one thing is missing, kids. One thing is missing. Before this, I should have done the reaction. CCl4 KOH. I must have done this reaction, Riemann-Tiemann reaction. Then you must have got salicylic acid. Then you must have gone one step. I have missed, kids, one step. Sorry for that. Salicylic acid and then aspirin. Aspirin, the painkiller. The painkiller aspirin. OCO-CH3. C double bond OOH, painkiller and protects from heart attack. So you see in aspirin, if I write down aspirin, benzene is having 15 covalent bonds. 15 O. Now see, 15, this much 15 covalent bonds, 16, 17, 18, 19 and 3, and 20, 21, 22, 22, 23, 24, oh my god, 25, 26, one more. 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26. Which is the nearest? It's 26. So kids, this is the answer, 20 to 30. Got it? Everyone got it? Just to let me know in the chat section you are able to understand or not. Let me have a glass of water, kids. Just to let me know in the chat section everyone got it or not. Now, next question. Question number 8. Now, this question is mind-blowing question. Mind-blowing. See why? Mind-blowing question. Not everyone can do this question. Should I give you? Are you accepting the challenge? It's a challenging question. Ready for this question, everyone? Do let me know. An organic compound molecular formula C5H10 on ozonolysis gives A plus B. A gives positive tolerance test, positive hydroform test. B gives positive tolerance test, negative hydroform test. So question is, find out IUPAC name of this compound. IUPAC name of this compound. So I am going to give you the options. A, hex 2 in B, 2-methyl but 2 in C, pent 2 in D, cyclopentene E, pent 1 in. See this question properly, and I'm sitting in just front of screen to check your answer. Everyone, Sahana is there, and Sahana is going with B. Let me check who is going to win this race. It's a really challenging question. Let me check it out. Who is going to be the winner? Who is really going to perform? Wow, Sahana, really awesome. But kids, let me see who is going to give the correct answer. Hello HSP sir, this is Vardhan, your 2023 batch student, now in a GMC. I remember attending similar sessions last. Kids, see, yes, Vardhan is there. Wow, congratulations. Kids, in the chat section, see, yes, Vardhan is there, and he's got selected. He is selected. See, everyone, give flowers to Yashwardhan, kids. Everyone, give flowers to Yashwardhan. He is not doctor. Kids, what are you doing? Show flowers, show green hearts to Yashwardhan. He is now doctor. He is selected from this session only, from these type of sessions only. Yashwardhan got selected from such type of sessions only, kids. Kids, when somebody is there and who got selected from such type of sessions, so you can't understand to show your gestures, to welcome him. This I have to tell you, kids. Am I the one who is going to tell you all these things? Show flowers, welcome him. He got selected from such type of sessions. Really, congratulations, Yashwardhan. Really, really warm welcome, warm welcome. Kids, he got selected. He has become doctor. He was like you people only. And from this channel, by watching all these sessions, such type of sessions, he got selected. He has become doctor now. Now check it out. Let me tell you what is the correct answer. Let's see. Let's see. Anionic compound C5H10. What is the DU? DU, degree of unsaturation, is X plus 1 minus Y by 2. 6 minus 5. 6 minus 5 is 1. That means 1 pi bond is there. Ozonolysis is happening. There is only one compound which gives both positive tolerance test and iodoform test. Positive tolerance test means aldehyde, and there is only one aldehyde in this world which gives tolerance test positive and iodoform test, and that is your acetaldehyde. Am I right or wrong? That is your acetaldehyde. Am I right or wrong? And this gives positive tolerance test. That means it is aldehyde. Now, out of 5 carbon, 2 carbon have been gone over there. How many carbons are left? 3 carbons. 5 carbon organic compound. Out of 5, 2 are over here. 3 are left. So this aldehyde is having 3 carbons. How it is aldehyde? It is giving positive tolerance test. If anyone is giving positive tolerance test, that means it is aldehyde. So 3 carbon aldehyde. Now, how to join these two? How to join these two? So you should know how to join these two. That you should know how the way you should join them. This is one aldehyde and this is another one aldehyde, kids. Now the way you have to join them, just release oxygen from here and combine them. What you will get? You will get CH3, CH double bond CH, CH2, CH3. What is the name of this compound? Everyone, just to let me know. What's the name? 1, 2, 3, 4, 5. Pentuine is the correct answer. How many of you got? How many of you understood? I want to see green heart. Really, such type of questions will come in your examination, and the one who is really getting it will be the winner. Do let me know in the chat section how many of you are able to understand. Gets. Now, how many of you want more questions like this? How many of you want more questions like this? Do let me know in the chat section. Really awesome question. Understood. Very good. Yes. Do let me know. Okay. Now see. Another question. Question number 9th. An organic compound with molecular formula C6H12 on ozonolysis gives A. This A gives shows positive iodoform test and negative tolerance test. Kids, if you are able to do such type of questions right now, if you are not able to do these type of questions, no problem, but do proper revision. And those who are able to do these type of questions, they are really going to be genius. Now, question is IUPAC name of compound. IUPAC name of starting compound. Right? I'm going to give you options. I'm giving you options. 2,3-dimethyl butene, 2,3-dimethyl butene, B, hex 3-ene, C, 2-methyl hex 2-ene, D, 2-methyl hexene. and sitting in front of screen. Let me see how many of you can answer it. Waiting for response. Sahana is going with A. Sanjana also going with A. Baba Tillu is going with B. A is the correct answer. Very good. A is the correct answer. See, Sahana has given first answer. Now, she was the first one to give the correct answer. Now, see, this is giving only one type of compound. Analysis. Only one type of compound means what analysis? Just like you must have watched CID serial. In CID serial, hey Daya, something is fishy. So if I say only one type of product is there, first of all, do let me know how many of you are really getting nervous while seeing such type of questions. Show me green hearts. How many of you are getting nervous if you see such type of questions? Whenever gets encountered with such type of questions, how many of you are getting nervous? How many of you are really getting nervous? Your anxiety level becomes up by seeing such type of questions. No one? No problem. So only one type of compound means alkene. This alkene is symmetrical. That indicates alkene is symmetrical. Now it gives positive iodoform test. Positive iodoform test, that means this thing must be there. To show iodoform test, this thing must be there. Now 6 carbons, so 2 moles of this I will get. Let's join them together. C double bond O, CH3, CH3, C double bond O, CH3, CH3. Let's remove this oxygen. Now what I will get by removing this oxygen? CH3, CH3, double bond, CH3 and CH3. How to write down the IUPAC? 1, 2, 3, 4. 2,3-dimethyl butene. How many of you got it? If you're really liking the analysis, if you understood, just to let me know in the chat section. Just to let me know, kids. Everyone, just to let me know. Now, next question. This time I will not give the options. This time I will not give the options. I will give question, right? You have to answer it. Just question will come in front of you. Question number 10th. B, KOH aqueous. C, PCC. D, KMNO4, acidic medium. E, ammonia. F, heat. G, HNO2. H, Cu2 Cl2 HCl I. NaOH 623 Kelvin, 300 atmospheric pressure, H plus J. Zinc dust, K. Reductive ozonolysis, O3, zinc and water, L. Now I want to see what is L. L. So let me give you options also. First option, methyl glyoxal. Second option, crotonaldehyde. C option, cinnamaldehyde. D option, glyoxal. So let's see how many of you can do this question. Even I have given the options also. Oh my God, I will not declare the answer, but really I am so happy. I'm so happy by seeing these answers. Yeah, Vishal MH, see my students. These students can answer any questions. This is Baba HSP. Everyone claps for Sahana HSPN. Sahana rocking. See, without options, such a great question, such a lengthy question, my students can do. Everyone, huge round of applause for Sahana. Sahana is the first one to give the correct answer. Such great questions can be even solved within fraction of seconds, even without seeing the options. And this is C. Let me explain it. Let me explain. Methyl chloride, this is Friedel-Crafts alkylation. Friedel-Crafts alkylation, so toluene. Free radical substitution reaction, so benzyl chloride. Nucleophilic substitution reaction, benzyl alcohol. MOA, mild oxidizing agent, PCC, benzaldehyde. SOA, strong oxidizing agent, benzoic acid. Ammonia, acid-base neutralization, ammonium benzoate. Heat, water will get released, benzamide. Hoffman bromamide degradation. So one thing is missing. Hoffman bromamide means Br2KOH must be given. Br2KOH is missing, kids. So Hoffman bromamide, then this is aniline, and then diazotization, benzene diazonium chloride. Sandemeyer reaction. This is chlorobenzene. Dow's process. This is phenol. Zinc dust. This is benzene. Just a second, kids. Just a second. Give me a second. Kids, how is the explanation? How is the explanation? How many of you want more such type of questions? More such type of questions. Show me green hearts. I will make you genius. This is Baba HSP's promise. This is my promise. I will make you genius. I will make you genius. Show me green hearts if you want such type of more questions. Kids, do let me know in the chat section if you are really enjoying it. Prajwal Reddy, welcome, welcome. Sir, how are you making these questions like Indra NNS? See kids, one kid has written really the best thing. Sir, how you are making such type of questions immediately? This girl is really genius. Then she is able to understand, sir, how great you are, how you are making such type of questions immediately on the spot. See kids, I deserve huge round of respect. Question 11. Incorrect match. Nota means none of the above. So, let's see. Let's see kids. Everyone just fast. Let's see who is going to get the correct answer. Baba Tillu is going for B. Rest of the others too, let me know, kids. Nota means none of the above, kids. Nota means none of the above. Now, kids, do let me know. Do let me know in the chat section. What's your answer? What's your answer? So, correct answer is none of the above. No one has gone for this because this is Hofmann bromamide degradation. Hofmann bromamide degradation reaction. So, Rn double bond C double bond O is correct. Isocyanate. This is carbylamine reaction, kids. Carbylamine test or reaction. So isocyanate, bad smelling compound, very bad smelling, foul smell. And this is very important reaction known as mustard oil test. Mustard oil test. So here Rn double bond C double bond S will get formed and it is having pungency of mustard oil. Now kids, before I move ahead, none of them is wrong. Have you done your lunch? Have you done your lunch? Should I give you 15 minutes for doing lunch, kids? Even I have not done my lunch. So kids, should we take 15 minutes break for the lunch? With your due permission, do let me know. Show me green hearts if you are ready. Just have some fruits or whatever you want to eat. Let's take 15 minutes break for the lunch. Even I have not done my breakfast. Neither breakfast nor lunch, nothing else. You know that yesterday I took marathon of inorganic chemistry. Yesterday I took marathon of inorganic chemistry, and today I haven't got time for all these things. Thank you kids. So let's meet at what time? Right now what's the time? 4:13. 4:13. So we will meet at 4:30. Right? Are you back, kids? Just to let me know. With the green hearts, I want to see, is everyone back? Ready? The K was missing over here, break time. So, show me green hearts once you are ready. Now, this time the question number, whatever the question number, let me check it out. Question number 12th, I will give from inorganic chemistry. Inorganic chemistry. Once you do, let me know that you are ready. Show me green hearts if everyone is ready, kids. Yes. Inorganic chemistry question. FeCr2O4 in presence of excess of O2, K2CO3 gets A in reaction with H2SO4 gets B. This B on reaction with NaCl concentrated H2SO4 gives C. C is deep red vapors. A is yellow solution. B is orange solution. Now, when we react toluene with C, what is C? That is C. On reaction with C and H3O plus, CCl4 H3O plus gives D. Now my question is, what is D? You have to find out D. The C is over here, the same C. That C and this C is same. And this on reaction with toluene, on reaction with toluene gets D. Now what is D? You have to find out D. So options are benzaldehyde, option B, cinnamaldehyde, option C, macetyl oxide, D, macetylene. Let me check it out. This C and that C, they are same. Let me check it out in the chats. What's your answer? I want to see in the chats what's your answer, kids. No one, no one. No one. See, I think your inorganic chemistry and organic, it is mixed of inorganic and organic question. It's a mix of organic and inorganic. The one who is good in organic and inorganic as well, both can do this question, kids. Let me see who can do this question. Fast, kids, fast. Everyone. What's going on, kids? No one is giving the correct answer. Vishal MH is the one who has given the clear answer. Vishal, you have given the correct answer. That's really awesome. This is the correct answer. A is the correct answer. Now let's see. This is known as iron chromite ore. Iron chromite ore. Now this on reaction with this will give yellow solution of Na2 or K2CrO4. Now this is K2Cr2O7 potassium dichromate, and this reaction is known as chromyl chloride test. Have you heard the name? This is CrO2Cl2 and chromyl chloride test. Chromyl chloride test, right? Now, so this reaction is known as Etard reaction. Have you heard the name Etard reaction? Etard reaction, on reaction with CrO2 Cl2 you will get benzaldehyde. So benzaldehyde is the correct answer. Those who got it, show me green hearts if you got it. Really, if you got it. Really, if you are able to understand, kids. Now, another question, question number 13th. Question number 13th. What color will be absorbed by Z? Will be absorbed by Z. Now what is Z? What is Z? Dimethylamine on reaction with NaNO2 HCl gives Z. So what color will be absorbed by Z? Options are option A, green. Option B is red. Option C is yellow. Option D is violet. Let's see who can give the correct answer. So this question is blend of organic and inorganic, made up of both. Let's see who is going to give the correct answer, kids. Want to see who is genius, how much you are genius, kids. Your intelligence will reflect in this question, kids. Who is going to give the correct answer? Let me check it out. Yes. So everyone got failed. Now see, kids, just check it out. Everyone got failed. Every one of you got failed because the correct answer is violet. Why the violet color? See, this is 2-degree amine, and 2-degree amine with this yellow oily liquid. Yellow oily liquid, am I right? And this is known as Lieberman's nitroso test. Lieberman's nitroso test. Now kids, what they asked? Emitted or absorbed? Absorbed. So which color is complementary to yellow? Violet is complementary of yellow. Violet and yellow, they are complementary color. Yesterday I taught you the color wheel. The color wheel, they are complementary colors like this. See, kids. The complementary colors, this always remember that, otherwise you will get failed. Vipcure, violet, indigo, purple, Vipcure, blue, Vipcure, green, yellow, orange, and red. So the complementary color, violet and yellow, they are complementary. Are you getting my point, kids? Do let me know. Yellow and violet, they are complementary colors. Blue and orange, they are complementary colors. Blue, green, red is the complementary color. These are the complementary colors. See, kids, if it is clear, then show me green hearts, the color wheel. You should remember always the color wheel. Is it clear, kids? If you got it, you are getting it. I want to see green hearts. So if this question number 13, so see, have you ever seen the blend of organic and inorganic chemistry? Now another question. Question number 14. Question number 14 is wrong IUPAC. Wrong IUPAC. You have to find out which compounds IUPAC is wrong. A, C double bond O H, CH2, CH2, COOH. Four aldo butanoic acid. Option number 2, three ethyl 2 mercapto cyclohex 4N1-ol. Third, that is C. Two hydroxy pent 3 in 1,5 dioic acid. All of these. So this is the question, and let's see how many of you can answer this question. See, it's real replica of NEET is going on, and you are dealing with new questions, absolutely new questions, kids. This will change your life if you are able to answer these questions. And let me check it out. Who can answer these questions, kids? Your life is going to get changed, kids. Apply your brain, kids. Apply your brain. If you have, and everyone is having, kids, everyone do have brain, you are not able to apply. See, the answer is all of these. All of them are wrong. Why they are wrong? See, if I do the numbering 1, 2, 3, 4, I have counted this in the parent chain. So who is left? Only this one is left. So its actual name will be what? Its actual name will be 4-oxo butanoic acid. Are you getting my point? Are you getting my point, kids? Just see what I'm telling you and you are not able to understand. Let me tell you, kids. Once you are counting it 1, 2, 3, 4. If this much you have counted, then this much is left. If this much is left, so this is also not Aldo. Otherwise, Aldo will be this much. This much is going to be Aldo, kids. But this much we have not counted. So, now see, in this one, if I talk about this is 1, this is going to be the 1, then multiple bonds should get preference. Then after 1, 2, 3, 4, 5, 6, this is being the correct numbering. Because during numbering, your preference order must be like this: secondary suffix, then multiple bond, and then the prefix or the substituent will come. Now, if this is secondary suffix, they all will act as prefix. Are you getting my point? They will act as prefix. So, secondary suffix 1, 2, 3, 4, multiple bond is coming on 3 position. If I go like that 1, 2, 3, 4, it will come on 4. So, this is a wrong answer. Its correct IUPAC will be 5 ethyl 6 mercapto. It should be 5 ethyl 6 mercapto cyclohex 3 in 1 all. E should be deleted. Now see, in this 1, 2, this is the prefix. So should I give preference to prefix or should I give preference to multiple bonds? So its correct numbering will be, remember that its correct numbering will be from this side 1, 2, 3, 4 and 5, not from this side. It will be counted from that side because secondary suffix on number 1, secondary suffix on number 1. After that secondary suffix, we have to see, we have to give importance to multiple bond, not to the prefix. Junior functional group will always act as prefix. This junior functional group will act as prefix, and this is your multiple bond. After secondary suffix, the position of multiple bond will get preference, gets. So you should know this, that 1, 2, 3, 4, 5. This should be the correct order of the numbering. So all of them, they are wrong. Everyone got it? If you are able to understand, I want to see green hearts in the chat section. Kids, if everyone understood, I want to see green hearts in the chat section. Really good question. Each and everything about IUPAC got clear or not, kids? Do let me know. You are able to understand or not? Now, question number 15. Really awesome question. You are going to deal with question number 15. Incorrect match Na2CrO4 yellow PPT B K2Cr2O7 orange PPT C potassium manganate pink, purple or red color. D, all of these, all of the above. Let me see who can give the correct questions answer. Let's check it out. How much preference, how much preparation you have done for the examination. Let me check it out, kids. What's your preparation? Up to the mark or not up to the mark? Let me check it out. Your preparation level. What's the level of your preparation? Gets fast. Answer the correct answer immediately. I want to see the chat running with more speed. Yeah, Ultra Magnus Prime. Correct answer is D. D is the correct answer. This is your correct answer. This is correct. Why? Let me tell you. This is sodium. Remember that all salts of amino rubicis, amino rubicis. This you know very well. All salts are soluble in water. These are always soluble in water. Always soluble in water. Once they are soluble in water, how they can be PPT? It is yellow, not the PPT, but what? In place of PPT, it should be solution. Orange, solution. And this is potassium manganate. Potassium manganate is K2MnO4, and K2MnO4 is green colored. It's not like that, it's green colored. Now, really awesome question or not? Do let me know. How many of you are enjoying my teaching? I'm just making new questions, basic questions from your NCERT. Just do let me know with the green hearts that really, if I'm teaching, my teaching level is really good. My framing of questions really means a lot, and if they are from NCERT and they are mind-blowing questions, then show green hearts. Otherwise, no need to show green hearts. Oh. Okay, so let's deal with this question. Let's see how many of you can answer this question. Let me go with printed question. And if you don't want printed question, you can tell me in the chat. I will give you my own questions, my own framed questions. And if you want this, C. Okay, let me check it out. Let me check. Computer G, just check out the answer. B. What answer you have given? C. How many of you want the explanation? How many of you need explanation of this question? Just see. To absorb color in visible light, the central metal atom must have at least one unpaired electron. Central metal atom has what? Central metal atom or ion must have at least one free electron. Am I right? To show DD transition, not titanium. This is neutral ligand, this is neutral ligand, 4 plus. Science teacher, very crazy man, do you remember? Science teacher, very crazy man, felt cold in night with cousin. Do you remember this? And if you really remember this, kids, If you really remember this, then you should know the atomic number 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. Titanium is over here 2, 2. So 2, 2 means 3d2, 4s2. 4 electrons gone. So titanium 4 plus means 3d0, 4s0, unpaired. No electron is here. No electron, that means colorless, can't absorb light. Can't be the answer. N is equal to 0. Now chromium 3 plus, just see chromium. Chromium is 24. Chromium is 24. So 3d5, 4s1, and how many charge? 3 plus, so 3 gone. So 3, 1, 2, 3, 3d. How many electrons will remain? 3, 3d3, 4s0. So it is having unpaired electrons. How many unpaired electrons? 1, 2, 3. 3 unpaired electron. If you want to see zinc, zinc is 30. 30 means Zn 30 means 3d10, 4s2, and Zn2 plus 3d10, 0 unpaired electron. Scandium 3 plus, scandium 21, 3 plus means 0 unpaired electron, 0, 3d, 0, 4s0. Am I right? If you are able to understand, show me green hearts if it is clear. If it is clear, each and everything is clear, show me green hearts. Gets fast. Now the Josh, where is the Josh? Your Josh, where the Josh has gone, kids? I cannot see the Josh. I think it's missing, kids. Why it's missing, kids? How is the Josh? Now, next question. Computer ji, next question. See, the explanation is given over there. Which of the following complex ion is not expected? Not expected. This time not. Remember the word not. Let me see who is going to give the correct answer. Quick, kids, quick. B. Okay, this time even I will do this question. I will not show you the answer. Let me check it out. Okay. Along with you, I will do the question. Science teacher, very crazy man, felt cold in night with cousin. 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. Nickel. This is nickel. How to write down the configuration? Just reverse it. 2, 8. 2, 8 is written. Write down 8, 2. 3d8, 4s2. Right? Now nickel, but cyanide, see the oxidation state. X minus 4 is equal to minus 2. So X is equal to plus 2 is the oxidation state. So nickel 2 plus 3d8 and 4s0. 4s0. So 3d8, how I will write? 3d8 under the influence of cyanide. Cyanide is strong field ligand. Am I right, kids? Cyanide is what? Cyanide is strong field ligand. under the influence of cyanide, the 8 will occupy how many orbitals? 3, 6 and 8. So this will remain empty. N is equal to 0. Which of the following complex ion is not expected to absorb visible light? Oh, this is 0. I think this is the answer. According to me, this is the answer. What you say, kids? I will go for A only. I will go with A only. What you say, kids? D. Okay, let's see. Let me check it out. Let me cross check. Chromium 3 plus. Chromium 3 plus. Here chromium. Chromium is 24, 3d5, 4s0, no, 4s1. So 3 has gone. So chromium 3 plus, 1 has gone, 2 has gone, 3d3. So it is N is 3. So it is colored. Colored means will absorb light, will absorb in the visible range, right? So this can't be the answer. This can't be the answer. Now iron 2 plus. Iron 2 plus. Iron is 26. So I will write down 62. Iron, I will write down 62. So 62, 3d6, 4s2, 4s2, 2 has gone. So 3d6, 3d6, and water is weak field ligand. So 6 electron, 1, 2, 3, 4, 5, 6. So the value of N is equal to 4. Nickel 2 plus. Nickel is over here, 28. Nickel 28. So I will write down 82, 82, 3d8, 4s2, 4s2. Nickel 2 plus, 2 plus. So these two go. 8, 8 means how I will fill 8. 1, 2, 3, 4, 5, 6, 7, 8. N is equal to 2. So my answer, I will go for A only. I will go for AL only. The starting only I have told you I will go with A, right? Do let me know, kids. Am I right or not? Let me show you the answer. I have cross-checked it. I have cross-checked it, kids. My answer is A. Same. A. Yeah. Kids, show me green hearts if you're really liking it. If you're really loving it. The Baba is also has become a student. Baba is solving questions along with you, kids. Do let me know, kids. Now, kids, before moving ahead, let me show you something. I want to discuss something with you, kids. You can help yourself, you can help others with this. You can help number of people. If you know about it, then you know that sharing is caring, and if you are helping others, Almighty God is going to help you a lot. Iconic and VIP week is on. Choose your edge in NEET UG prep. Offer ends tonight. Use code. What's the code? HSP10. HSP10. This Iconic is available just for rupees 13,499. An Iconic subscription, you know that people are getting notes, the booklet, hard copy. My dear student, at this moment of time, I know that it's not for you, but at least you can help others, your junior ones, relatives, classmates. I don't know that where the people must be suffering. You know, number of offline coaching students, they are suffering with teachers. They are not having teachers, and if they are having teachers, they are not good quality teachers. And if everything is okay, then the food material, the food, the whatever, the water and the climate conditions, they are not able to match with that. They are not able to cope up with that. So they are suffering with a lot of diseases. Anxiety level is getting high. So to live in other city, to pay 2.5 lakh rupees, to pay the rent, the whole thing will come out near about 4 lakh to 5 lakh. And it's available just for 13,499. You can eat idli, dosa, utpam, whatever your mother is making, whatever your area, nearby area you are going to get from there. What to say? They are not charging even a single, what to say? It's very, 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 what to say? High quality and cheapest price. And this is VIP batch. It's not, it was earlier or going to be after this discount 49,999, but right now it is 44,999. Now, you know about Unacademy, Iconic, and these features you know very well. VIP features, you can go through it, tell them. And here I would like to say the plus subscription for 12 months, it is just for 4,999. CBSE combo, VIP combo, everything is over there. You can avail it if you apply code HSP10, and you will get mentorship also, kids. Mentorship also, right? Now, what's required for you, kids? Even I can tell you that this is for you. This is for you. Free mock test before the actual exam. Your gateway to NEET UG 2025 success, real exam, simulation for NEET UG, questions as per the latest, predict your rank and enroll now. This is 20th April 2 to 5 p.m. This is for you, and this is your Wassim sir. Wassim bhai, how are you? My bro. Thalaiba bro, Wassim bro, right? So what's over here? Join NCERT Crux test series for NEET UG 2025, March 25 onwards. Now let's come back. Let's set go with our chemistry kits, and where we were. So this is same type of question, but before this question, before going ahead with this question, let me give you one question from my side. One question from my side, okay? Only genius can solve these type of questions, not everyone. So it's not for everyone. What is G? You are supposed to find out G. Let me write down the options. G is So the question is here, the options are there. Do it and let me check it out. Who is going to win? Who is going to be the winner? Some are going for B, some are going for C. But what's the correct answer? Let me tell you, kids. These type of questions means a lot. See. Ethyl alcohol alumina heat dehydration, non-acidic dehydration, ethane, am I right? Ozonolysis will break from here 2 moles of formaldehyde. Grignard reagent HCH, it's like this, this negative part whatever is there, add over here and O will get added with H. Are you getting my point? Ethyl alcohol PCC is mild oxidizing agent. So you will get acetaldehyde. Now once again reaction with the Grignard reagent, add this over here. Isopropyl alcohol, camenophore, SOA, strong oxidizing agent, you will get acetone. Now Grignard reagent, once again this is del positive, Grignard reagent will come over here. Tertiary butyl alcohol is the answer. C is the correct answer. Those who got correct answer, show me green hearts. And if you want such type of more questions, then pink hearts, pink or red hearts. I want to see pink or red hearts, green hearts. Green hearts for understanding it. Pink heart if you want more such type of questions. Okay, right. Let's see in one question how the world can get changed just in a single question. Just see. Oh. I is? You are supposed to find out I. Let me write down the options. Let me see, kids, who is going to give the correct answer for this question. Let me check it out, kids. Who is going to be the brilliant topper? Who is going to be the topper? Yeah, you will get PDF for all these. Yesterday's PDF is already shared with you, kids. You will get PDF of this. Whatever, thank you. Kids, when you write, you are the best. I get energy. I don't know what type of energy, but positive energy. I feel really and fully enthusiasm. So let's see who is going to win this. Question's answer is correct. See, benzene, this is known as Friedel-Crafts acylation. Friedel-Crafts acylation, right? So it's acetophenone. What is the name? Acetophenone. What is the name? Acetophenone. This is Clemenson's reduction. You will get ethyl benzene. Side chain oxidation. Benzoic acid. Acid base neutralization. Benzamide. Hoffman bromamide degradation. Aniline. NaNO2 diazotization. Benzene diazonium chloride. Sandmeyer reaction. Cyanobenzene. Stephens reduction. Benzaldehyde. CH3CHO. Perkin reaction. It's Perkin, no. Claisen-Smith. Claisen-Smith. That is cross-aldol condensation. Cinemaldehyde. A is the correct answer. Those who got it, show me green hearts in the chat section. Those who got it and those who understood. Everyone, just do let me know in the chat section if you got it and is it clear. Just do let me know in the chat section. Now I am going to give you mind-blowing question. A really small, simple question, but you won't be able to do it. One equivalent of A three equivalents of B equivalents dilute NaOH has given now A and B are respectively. A and B are right. This is the question. So let me give you options. A and B are respectively. None of the above. Do let me know in the chat section what's your answer, kids. Let's see, let me check it out. Do let me know, kids. C. Wow, kids, you are really awesome. This time you have done it. Done it, kids. Killed. You have killed it. Rocking, kids. My lovely kids, you are rocking, rock star. Loving. Lovely. Lovely, kids. One equivalent of this having 3 acidic alpha hydrogens and 3 of formaldehyde. Very good, kids. Now one more question like this. Really awesome, kids. Now I'm feeling accomplished with your stupendous performance. Baba is feeling awesome, accomplished. Question is, how many compounds will show positive Victor-Meyer test? Options are yes, kids. Do let me know in the chat section what's your answer. Sahana is going with A. So everyone just sees Sahana is going with A. What's about others? Let me check it out. Kids, Mukesh Murali is going with D. Kirtik is going with C. All the options are being ticked. And it's really a very, very hard competition. Let's see who is going to win this match. All the options, almost all the options, everyone has marked for it. So the correct answer, Baba HSP is going to tell you. Hey, Sahana, your answer is correct. All four, because the journey starts from here and this is nitric acid. It moves up to there and Sahana has given the correct answer. Now, another question. Let's see who will give the correct answer. Let me first give you the passage. The passage says, primary amine gives carboylamine test. The passage, it's written, all primary amines give positive carboylamine test. Now question is, how many compounds will show or will give foul smell? Foul smell means that is carboylamine test. A, B, C, D. E. Now options. Just see to it properly. These questions are $1 million questions. No one can tell you such type of questions gets newly framed, absolutely new. Absolutely new, fantastic, Romba Sulbam questions gets really awesome. Sahana HSPN is going with D. B. Give me $1 million. Oh, pointless. So, my dear friends, should I tell you one thing? With heavy heart, Baba HSP is telling you that everyone is failed. No one is able to qualify this question. Sorry, kids. With heavy heart, I'm telling you that every one of you got failed. Mommy. Everyone got failed. Ayyo, Ranga Swami. What are you doing? You are unable to understand the passage is given over there that all primary amino gives positive carbon test. If a passage is given, then also you are getting a failed. Then what you will do? How will qualify the examination? Everyone just let me know. It's given over here. All primary amine gives a positive carbon test. Then also you got getting a failed. See, this is what the degree amine. Just do let me know what is the degree of this amine first one. Everyone, I want to know what is the degree of this first amine. Let me know in the chat section. Hey, Emperor, you said, oh, very good, Emperor. Ready? Very good. Do let me know. Everyone, just first of all, do let me know what is the degree of this amine. I want to know that what is the degree of this one first amine. Everyone just do let me know what is the degree of this amine in the chat section. It is 1 degree. See, it is attached with 1 carbon only, so it is a primary amine. This is also attached with 1 carbon only. This is a primary amine. This is also primary amine. This is also primary amine. This is also primary amine. So there are total 5 primary amines. How was the question? If you really loved it, everyone just do let me know the green hearts. I want to see green hearts in the chat section. Hey kids, everyone. Now, another question. Let me give you the options. Just see properly and then give the answer. No hurry, kids. Let me check it out. But it is in your syllabus. Pyruvic acid is in your syllabus. Number of times they have asked this question. Remember that. Pyruvic acid has been asked during games, exams also. Let me see who is going to give the correct answer, kids. Who is going to give the correct answer? Till now I haven't got the correct answer. Try it out, kids. Try it out. Mukesh Murli is correct. Rest of other students, those who have given answer as this as your answer, B, then you are right. See why. This will give positive, this will give positive, this will give positive, this will give positive, this will negative, positive, positive, negative, positive, positive, positive. How many compounds have given? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 compounds. Now pyruvic acid and lactic acid structure, let me tell you. CH3, C double bond O, CHO. This is pyruvaldehyde. CH3, C double bond O, COOH. This is pyruvic acid. CH3, CHOH, COOH. This is lactic acid. Got it, kids? They all will give positive iodophone test, and this is chloral, CCl3, CHO. This is chloral. Now next question. Are you ready? I want to see the Josh in the chat section. If the Josh is high, then only. Yes, let me see. Are you ready? Are you ready, kids? Dishum dishum. Let's play dishum dishum. Hey, are you ready? Just to let me know. Okay, let me start this time question from what? Hey, Champa, where are you, Champa? Just give me some special type of compound so that I can start from there. Yes, which compound? Cumene. Cumene. Champa is saying cumene. Start from cumene. Oh, Champa. So mean. Cumene. Champa said start from cumene. Okay, Champa. What is L? You kids are supposed to find out L. Let me give you the options for L. Okay, kids, let's see who is going to give the correct answer for this question. Awesome question. The ball is in your court, and let's see who is going to kick it out. All the options are there. The ball is there. Let's see who is going to give the correct answer. Yes, kids. Let me see who is going to give the correct answer, kids. Fast, kids. Shruti Sashi right now has given the correct answer. Shruti Sashi has given. Congratulations, Shruti Sashi. Your answer is really awesome. But let me check it out, kids. How to move on? Cumene. Now, cumene is this compound. It is having this hydrogen, so side chain oxidation. Everyone just see to it. So you will get benzoic acid. Side chain oxidation, you will get benzoic acid. Am I right, kids? This is your decarboxylation reaction or soda lime reaction or lab method preparation of alkane. Benzene. Now benzene, this ethyl alcohols H plus will attack on the lone pair of oxygen to convert into oxonium ion. Now oxonium ion oxygen will say I can't tolerate this plus charge. So water will eliminate out, plus charge will attack on benzene. So ethyl benzene, am I right? Once again, side chain oxidation, kids. Benzoic acid. Thionyl chloride. Benzoyl chloride. What is the name, kids? Benzoyl chloride. Remember that, its name, very, very important. Benzoyl chloride. Rosenmund reaction. Have you heard this name? Rosenmund reaction. Very, very famous reaction. Benzaldehyde. Till now, if you have got it, now from here, Wolff-Kishner reduction. Have you heard this name, Wolff-Kishner reduction? So toluene, you will get toluene. Chromyl chloride, Itard reaction, benzaldehyde, Clemenson's reduction, toluene, Cl2, FRSR, free radical substitution reaction 3 times, benzyl, benzyl, benzo trichloride. Reverse hyperconjugation, meta director. So ESR at meta position. Now once again the allylic chlorines will go and hydrogen will take place. This is given in class 11th. In place of chlorine, hydrogens will come. So benzaldehyde, by chance it's metachlorotoluene, right? So who has given the correct answer? Do let me know in the chat section. Who has given this answer, kids? Who has given this answer? I want to see in the chat. Okay, kids. Now Yava sir will come for the physics class just in 30 seconds. So kids, this is all for today from my side. How was the session? If you really liked from my side, next class Yava sir for physics is going to start. Do let me know in the chat section how was the organic chemistry and inorganic chemistry. With full dedication, I have given you the best. With new questions, have I given you the best? Then yes, I would like to see green hearts in the chat section. Do let me know, kids. Do let me know. Quick. Only I am having 30 seconds. Just after this class, Yava sir is going to the best teacher, for physics in the entire world. Yes, most charming, most very intelligent Yava sir. Yava sir is going to take the class. He is going to start the class. So, bye to all of you. We'll meet you, yes, in next class. But yes, your class is, yes, what to say, bonded with, this chemistry class is bonded with physics class, right? So, okay, are you ready for the physics class? Yay! Okay, Janvi, don't worry. Do proper revision. You will be able to do these questions. Proper revision is required. Just that, okay? Thank you, Shadows, Hamad Awad, Marifaz Janab. Hello. Okay, ready, bro? Okay.
Already started. Hi guys, I believe all of you are doing great, having a good time. Please let me know in the chats if I'm perfectly audible, visible to each and every single student. Everybody, guys, let me know in the chats if I'm perfectly audible and visible to each and every single student. Yo, 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 yo. So guys, we have now the questions of physics, okay? I would want you guys to smash the like button and mark the attendance if you have not liked the session yet, because liking the session is marking the attendance. Okay? Yes, that's great. That's great. Now guys, first and the foremost thing in this particular case, let's start with an important question. Let's start with an important question. That is the question from current electricity. Okay. Let me first solve this question, then I'll show you some of the new patterns of, we say, electrostatics also. Because when it comes to the current electricity and electrostatics, these are the, we say, high weightage chapters when it comes to the physics. Okay. Now guys, see. Now guys, see. I would want you guys to focus exactly over here. See guys, see guys, see guys. Take a look, take a look. In this particular case, you have a circuit something like this. You have a circuit something like this. And you have this 6 ohm resistor, you have this 12 ohm resistor. You have 12 ohm resistor, you have 4 ohm resistor, and this is a battery of 70 volts. You are supposed to find the current in this branch. Guys, first and the foremost thing is, let's find the current in the entire circuit. Okay, how much current this battery is sending. How do we find that? First, find the equivalent resistance over here. Guys. First, find equivalent resistance. If I say, what is the equivalent resistance of these two, 6 and 12? Guys, when these two are in parallel, when the two resistors are in parallel, okay, when the two resistors are in parallel, how do we find? We say R equivalent in that case is R1, R2 upon, we say R1 plus R2. Now in this particular case, this is 6 into 12. Okay, we say 6 into 12 upon, we say 6 plus 12. Okay. And my dear friends, in this particular case, if you find the R equivalent over here, this will be, that is R1, R2, that is 4 into 12 upon, we say 4 plus 12. This is how we are supposed to find out the equivalent of these two and equivalent of these two. Okay. Okay. Now guys, tell me in this particular case, we say this is R1, R2 upon, we say R1, R2 upon R1 plus R2. Now I'll be directly writing over here. See. Guys, first you have to get the equivalent of these two. Then you will have to get the equivalent of these two. Okay. Now in this particular case, it will be 4 and 3. It will be 4 and 3. Now guys, when I say equivalent resistance of this entire circuit, I won't be showing that in detail. Okay, I have shown you over here. Now these two and like the equivalent of this one and equivalent of these two are in series. So you can basically say the final circuit that I'm going to make over here, that would be something like this. That would be something like this. Guys, take a look. So equivalent resistance in this particular case, that is, we say 7 ohms. Okay, and this is 70 volts. And when it comes to the current, guys, current will come out to be I is equal to V divided by R. V is 70 and R is 7. That is going to be, we say, 10 ampere. So means how much is the net current in this particular case? We say that is 10 ampere. 10 ampere is the net current. Okay, yes. And how do we calculate? These two in parallel, they're equivalent in series. Okay, that is 7 ohms, 70 by 7, that is 10. Now guys, when this 10 comes and 10 enters over here, we say some part of the current goes in this one, some part of the current goes in this one. We said two laws will be applied over here. Law of potential drop, okay, law of current distribution. Okay. Now guys, in this particular case, I say this is 10 ampere, and we know that when it comes to the law of current distribution, what does that say? That say current is inversely proportional to resistance. The one which has got more resistance will have less current in it. So my dear friends, I say in this particular case, if 10 ampere current is entering, so let's suppose this is 12 ohm resistance. Okay, here current will be less. Let's assume current in this is X, and 6 is how many times bigger than, smaller than 12, that is 2 times, or 12 is 2 times bigger than 6. So here current is going to be 2x. So this current is dividing into X and 2x. Why here current is less? Because resistance is more. Now, can I say in this particular case, guys, if I say current here, can I say X plus 2x, this will come out to be 10 ampere. Now, this will be 3x is equal to 10. X will come out to be, that is 10 by 3 ampere. X will come out to be 10 by 3 ampere. So what does that mean? That means current flowing in this is 10 divided by 3 ampere. Okay. And how much is this? This is 2x. So this will be 20 divided by 3 ampere. Okay. So we got the current in the upper branch. We got the current in the lower branch. Now guys, take a look. If I say 10 ampere current is entering, what do you guys think? How much current will be leaving? We say, sir, 10 ampere current will be leaving. Now here also, we say this current is basically the sum of current flowing through 12 ampere and the current flowing through 4 ampere. That is adding up and it is becoming 10. Now see, can I use the law of current distribution in this particular case also? Can I use the law of current distribution in this case also? We say if this is 10, we are assuming, we say X current will flow in this one. Okay, X current will flow in this one. And 4 is how many times less than 12? We say 3 times. So here it would be 3x in this case. Now, can I say in this case 3x plus x? So x plus 3x in this particular case is going to be, that is 10 ampere. Now 4x here is 10 ampere. Okay, so x in this case will come out to be, that is 10 divided by 4. Okay. Now, this current is 10 divided by 4. This current will be 30 by 4. Now, take a look at the upper branch. So, basically, we wanted to calculate the current in this branch. We wanted to calculate the current in this branch. Now, if I make the new diagram, it is like this. You have got a resistance. Okay, this is the branch. You have got one more resistance. And here, in this resistance, we say current is 10 by 4. Okay, it is 10 by 4. Here current is 10 by 4. Here current is 20 by 3. Okay, this is the incoming current, and here current you guys are supposed to calculate. As don't you think in this particular case, this current is being divided into, this current is getting divided into, we say this 10 by 4 and this I. So can I say this 20 by 3 is equal to basically I plus 10 divided by 4. Exactly. So you can say over here 20 divided by 3 minus I can say 10 divided by 4 will come out to be I. Now this is 80 minus I can say 30 divided by 3 into 4 is, that is 12. This will come out to be I. Now 80 minus 30, that is going to be 50 divided by 12 is the current. Okay, 50 divided by 12 is the current in this particular branch. Okay, I hope this is crystal and clear. So basically, guys, what I'm trying to say is that first you have to solve this entire circuit. Like you have to find the equivalent of these two resistors. Then those two equivalents are in series. Their equivalent will come out to be 7. In nutshell, we say equivalent resistance of the entire circuit is 7. Battery is 70 volts. Okay. Now, how much is the current? I is equal to V by R. 70 by 7, that is 10 ampere. Means net current is 10. Now, this 10 is divided into 12 ohm and 6 ohm. So, we say less current flows. Less current flows through more resistance. More current flows through less resistance. Here, through this it is X. Through this it will be 2X. Why? Because 12 is 2 times bigger than 6. That's why. Now, this current plus this current is total 10. So X comes out to be 10 by 3. So if this is 10 by 3 ampere, this will be 20 by 3. Similarly, we use the law of potential drop here also. Then we got the value of the current. Is this clear? Please tell me in the chats. Is this clear? Please tell me in the chats. Is this clear? Please tell me in the chats. Everybody out there. No. Take a look at this particular question. Take a look at this particular question. This question is extremely important. This question is extremely important when it comes to your NEET examination. This is the question from current electricity. This is the question from electrostatics. Take a look. Guys, you have a charge over here. You have a charge over here plus Q. You have a charge over here plus Q. Okay, these two charges are in equilibrium. These two charges are in equilibrium. Take a look. These two charges are in equilibrium. Means they are at rest. If I say these two charges are in equilibrium and this is the distance between the two charges, you guys are supposed to tell me find the value of Q. Okay, sorry, theta. Okay, this angle theta. Tension in the string, that is this T. Q, charge on the sphere. X, distance between the two spheres. If the charge is in equilibrium. If the charge is in equilibrium. Take a look. Whenever you face a question something like this, means these are the two charges suspended with a thread. This is a thread. This is one charge. This is another charge. So these two charges are in equilibrium. If these two charges are in equilibrium, what you guys are supposed to do in this particular case, guys, take a look. I say, draw all the forces acting on the charge. On this charge, it will be mg in the downward direction. Okay. Now, this charge will apply electrostatic force on this in the outward direction. Fe is equal to, we say, k q square divided by r square. Yes. Okay. So, this is, I would write k q square divided by r square. Yes. Now, guys, this is tension. If this is tension, this is theta. This component of this would be also theta. This would be t cos of theta. This component would be t sine of theta. I would want you guys to focus exactly over here. Guys, if the charge is in equilibrium, means this force will be equal to this force. Upward force will be equal to downward force. So you guys will say over here, t sine of theta will be equals to k q square divided by x square. And t cos of theta will be equals to, that is mg. I want you guys to divide the two equations. T and T will cancel. Sin theta by cos theta is basically tan theta. Okay. So tan theta will come out to be kq square divided by x square into mg. X square into mg. Okay. Yes. So this comes out to be the value of theta. This comes out to be the value of theta in this particular case. Okay. Now, guys, if you want to calculate the value of q, okay, this is the theta. This is the value of theta. Now, let's find the value of q. Okay, this charge. Take this x square mg over here. You'll be writing x square m into g. Then you say tan theta. Okay, tan theta divided by this k. It will come out to be q square. And instead of this square, I'll put the root over here. So you are getting the value of q. From this equation only, you are getting the value of q. Okay. Now, if somebody wants you to find out, the value of x square, guys, from this equation only, can I say take this x square here, tan theta here? So x square will come out to be, that is kq square upon, we say that is tan of theta m into g. So can I say x will be equals to under root of kq square, and then we say tan of theta m into g. So this is the value of x. This is the value of q. Okay. Okay. Now, let's suppose if somebody wants to calculate the value of tension over here. Guys, take this charge. Take this charge. This is plus q. What are the original forces acting on the charge? Okay. What are the original forces acting on the charge? We say this is the tension force. In the downward direction, it would be mg. From this side, it is electrostatic force. Guys, see. Can I say resultant of this electrostatic force and this? It would be something like this. This would be resultant R. And how do you calculate this resultant? It would be under root of, guys. If you have the angle between the two vectors at 90 degrees, then resultant of two vectors is, that is Fe whole square. Then we say mg plus mg whole square. And guys, if you are saying this charge is in the equilibrium, upward force will be equal downward force, right side force equal left side force, and this force would be equal to this force. So tension would be equals to R, and that would be equals to, that is Fe square plus mg whole square. I hope this is crystal and clear to each and every single student. I hope this is crystal and clear to each and every single student. We were supposed to calculate the value of theta. This is the value of theta. We were supposed to calculate the value of charge, okay, X, and we say tension. These two were asked in, we say JEE Mains 2021. Mains 2021. Okay, yes. Guys. I want you guys to remember this one single formula in this one, and this will give you the question. This will give you the answer to all the cases. Okay, this will give you the answer to all the cases. This equation. Okay, now guys, see. Let me make one more question over here. Question is, we say if the two suspended spheres are not identical, then what does that mean? That is, mass of first sphere is not equal to mass of second. Then or you can say it something like this: are not identical, mass of first is greater than mass of second. Then compare theta 1 and theta 2. Guys, in the previous case, if I say mass of this sphere is m, this is theta, we say this will be also theta. Why? Because charge is same, mass is same. So here theta will be same, okay? Angle made by this sphere with the vertical, angle made by this sphere with the vertical will be same. Yes. Now, if theta is not same, what can I say in that particular case? Guys, take a look. I would want you guys to focus. Okay. So this is the charge. I would say over here, its charge is Q1, its mass is M1. And this is another, it's Q2 and its mass is M2. In this particular case, let's suppose this is making theta 2 over here, this is making theta 1 over here. We have to compare which one is greater and which one is smaller. Guys. If I write the value of tan theta, what is the value of tan theta? K Q square X square upon mg. I can write over here tan theta 1. Tan of theta 1, I can write K Q square. So here it is Q1, here it is Q2, divided by X square. Then we say M1 into G, because here mass is M1. Similarly, this tan theta 2, I can write over here. Tan theta 2 will be, that is K Q1 Q2 divided by X square M2 into G. Guys, divide the two equations, okay? K Q1 Q2, K Q1 Q2, X square, G. So I can say here, tan of theta 1 divided by tan of theta 2 is coming out to be equal to 1 by M1 upon 1 by M2. So that will be M2 divided by M1. So I can say tan of theta is inversely proportional to mass. Now, if we say M1 is greater, so we say tan theta 1 will be less. So I say M1 is greater than M2. So theta 1 will be less than theta 2. Why? Because theta is inversely proportional to mass. So that's why I'm saying theta 1 is less than theta 2. It is obvious. If I say this sphere is having more mass, it will be difficult for the sphere to create greater angle. So that's why we say theta will be less. Okay. So this was this question. Great. Let's suppose if you ever get a question something like this. Guys, take a look. Find the equivalent capacitance. Okay, resistance, because this is the resistor. Guys, this looks a difficult figure over here, but it is not difficult at all. I want you guys to focus. Don't you think in this particular case, these three are in parallel? So whenever identical resistors are in parallel, so their equivalent resistances are by N. So how much is R over here? 30. So 30 by N, 1, 2, 3, N is 3, 30 by 3, that is 10. So their equivalent resistance is 10, 30 by 3. Now guys, take a look. This is 20 as it is. Now this 10 ohm, the potential here will be same as potential here because it's a straight wire. Whenever potential across the resistor is same, so this is short circuit. So you can remove this resistor. Here also, you can see directly. It is connected over here with a simple wire. So this resistance will be also short circuit. Now let me redraw the figure. Instead of this, I'll make 10 ohm. Instead of this, I'll make this 10 ohm. And then you have 20 ohm connected with it. This is 20 ohms. And then this 20, 10 and 20. This 10 and 20. Okay. So I'll make it over here. This is 10 and this is 20. This is 10 and this is 20. Now guys, tell me. 20 plus 10 is 30. 20 plus 10 is 30. So it would be something like this. So this is 30. This is 30 and this is 30. Now, if identical resistors are connected in parallel, their equivalent resistances are by N. N means 1 and 2, two resistors. So this would be 30 divided by 2. That is 15 ohm is the equivalent resistance in this particular case. I hope this is crystalline clear. Is this clear, guys? Tell me in the chats. Is this clear? Please tell me in the chats. This was the question which was asked in J Mains 2021. Before this, let me show you this one. Guys, here I is equal to 5 ampere. This is 6 volt. This is 3 ohm. You guys are supposed to find the potential of this point A. You guys are supposed to find the potential of this point A. And here it is grounded over here. Means potential of point B is 0. Guys, whenever you have to find the potential of point A, let me show you how do you find this. Let me show you how do you calculate it. Select the path. Select the path. Let's suppose I'm selecting the path from B to A. I'm selecting the path from B to A. Okay. So if you are selecting the path from B to A, first you have to write the potential of B. So this is VB. Okay. This is VB and that is 0. I'll directly write this is 0. Then, this is higher potential, this is lower potential. And in this case, this is higher, this is lower. Why? Because the direction of the current is this. So current always flow from higher potential to lower potential. Current always flow from higher potential to lower potential. Okay. And this is negative, this is positive. This is positive, this is negative. Because here current is flowing from higher potential to lower potential. In case of the battery, this is higher, this is lower. Now see. First, you write the potential of VB, that is 0. Then potential of this, how much is the difference of this? That is 1 volt. So I'll write minus 1 volt. Why? Because you are moving from higher to lower. Because this is your path. If you move from higher potential to lower potential, you take its potential drop. That is 1 volt. This as negative. Then here. You are also moving from higher potential to lower potential. So its potential drop is also negative. And how do you find the potential drop in case of resistance? V is equal to I into R. Current is 5, resistance is 3. 5 into 3 is 15, so it will be 15. 5 into 3 is 15. Okay. Then here. You are moving from lower to higher, so its potential drop will be positive. And it is 6 volts. It is 6 volts. Okay, because you are moving from lower to higher. Here you are moving from higher to lower. This would be negative. And how do you calculate? I into R, that is 4 into 5, that is 20. 20 volts is equal to, then finally you reached VA. Now tell me, this would be minus 1, minus 20, minus 15. Minus 36 volts plus 6 volts is equal to VA. So I can say VA will come out to be minus 36 plus 6. That would be minus 30 volts. Please check it from your end. Yes. Now on the similar pattern, question was asked in J Mains 2021. And same question was asked in NEET 2018. You have a circuit something like this. Okay. Here it is 0 volt, 2 ohm resistor, 12 ohm, okay, 1 ampere current. See guys, if current is flowing from something like this, the direction of current means in case of resistance, this is positive, this is negative. Why? Because we say current flow from higher potential to lower potential. Okay. Now here, this is negative and this is positive. In case of the battery, this is positive, this is negative, this is positive, this is negative. Now select the path. If I'm selecting the path from, let's suppose, A to B, first I'll write the potential of A, that is 0 volts. Potential of A is 0. Then here, potential in case of, potential difference in case of resistance, that is I into R. Current is how much? That is 1 ampere. R is how much? 1 into 2 is 2 ohm. That is minus 2 ohm. Here it is minus 2 ohm. Then in case of the battery, it would be plus 12 volts. Then in this resistance, its resistance is R. We have to find this R only. Now this is I into R. So 1 into R, that is R only. And it will be minus R. Why? Because you are moving from higher to lower, plus to minus. And in case of this resistance, you are moving from higher to lower. Its potential difference would be also negative. So that is 4 into 1, that is 4 volts. Okay, is equal to VB. VB again is 0. Okay. Now guys, this is minus 4 minus 2, minus 6 volts plus 12 volts minus R is equal to 0. Now, minus 6 plus 12, this will be plus 6 volts minus R is equal to 0. So, R will come out to be 6 volts. R will come out to be 6 volts. R will come out to be 6 volts. Okay. Yes, 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 yes. Let's first move here. Take a look at this question. This is a question from transformer. This is an important question. This is an important question. See guys. A step-up transformer operates on 230-volt line and supplies a load of 2 ampere. So there is a transformer which supplies a load, means output current is 2 ampere. Okay. The ratio of primary to secondary windings. The ratio of primary to secondary windings. Okay. So means NP divided by NS, number of turns in primary divided by number of turns in secondary. This is given as 1:25, 1 by 25. Determine the primary current. We are supposed to find the value of primary current or input current. Guys, see, when it comes to the output in case of a transformer, we say output voltage in case of a transformer is simply input voltage, number of turns in secondary coil upon number of turns in primary coil. So from this particular equation, we can say output voltage divided by input voltage will come out to be NS divided by NP. This is the first equation. Second equation we know, guys, when it comes to a transformer which has got 100% efficiency, we say in that case, output power will be equals to the input power. So can I say over here, voltage power is V into I. What is the formula of power? V into I. So that is output voltage into output current will be equals to, we say input voltage into input current. So in this particular case, can I say output divided by V input, this will come out to be I in divided by I out. Okay, call this as equation second and call this as equation first. Now can I say from first and second? From first and second equation. Here V out by V in is NS by NP. Here V out by V in is I in by I out. So can I say input current divided by output current? Okay. Okay. It is going to be NS divided by NP. NS divided by NP. Now guys, I input current we are supposed to find out. Output current and this question is given as that is 2 ampere. This equation is, by the way, very important. You guys need to remember this. Okay, is equal to number of turns in secondary. Guys, it is NP by NS is 25:1. Can I say NS by NP will be 25:1? Yes, 25:1. So 2 into 25 is 50. So input current in this particular case will be 50 ampere. Input current will be 50 ampere. Yes. Let's solve one more question from the transformer. Okay. See guys, the primary and the secondary coils of a transformer has 50 and 500 turns. Number of turns in the primary coil is 50 number of turns in the secondary coil is 1500. Okay, respectively. If the magnetic flux linked with the coils is, so flux is given over here. Okay, this is the flux through the primary coil. This is given as, that is phi naught plus 4t. We are supposed to find the output voltage. We are supposed to find the output voltage across the secondary coil. So here flux is given, guys. When flux is given, always remember, listen, always remember. See, I can write over here when flux is given, if you are supposed to find the current, we say voltage is equal to d phi divided by dt. Okay. If you are finding the input voltage, so this will be flux primary, flux input. Now, differentiation of this flux, that will give you the input voltage. So for phi naught, it is 0 because phi naught is given as constant. Okay. And for 4t, it is going to be 4. So input voltage is 4. Now you have to find the output voltage. How do we calculate the output voltage? Output voltage will be V in, okay, number of turns in secondary upon number of turns in primary. This is 4. Number of turns in secondary is 1500 divided by, this is 50. This and this will cancel. 5 ones are, 5 threes are, 5 zeros are. Now 4 into 3, 120 volts. So output voltage in this particular case is 120 volts. It is 120 volts. This is the question from AC, alternating current. Okay. Okay. It says that in the following AC, E naught is equal to 200 volts, resistance is 20 ohms, L is 0.1 Henry, capacitance is given. This is the LCR circuit. Okay. Frequency is variable. Frequency is variable. The current at frequency F is equal to 0 and F is equal to infinity will be. Guys. When it comes to the LCR circuit, everybody knows that impedance or the resistance of LCR circuit is Z. And that is under root of. We say XL minus XC whole square plus we say R square. What is XL? Inductive reactance. What is XC? Capacitive reactance. R is resistance. Okay. This is the value of impedance, or you can also say XC minus XL if LCR is capacitive in nature. Now guys, I can write Z will be equals to under root of. What is the value of XL? It is omega into L minus what is the value of XC? 1 upon omega into C. Okay, it is whole square plus I can say R square. So Z will come out to be under root of omega, that is 2 pi into F. Omega is also 2 pi F. This is L minus 1 upon 2 pi into F into L into C because it is C. Okay, it's whole square, then we say plus R square. So this is the actual formula of impedance. Now take a look. Guys, it says frequency is variable. We say if frequency is 0, could you please tell me in that particular case, what is going to be the value of impedance? Impedance means resistance of the LCR. If frequency is 0, this term would be 0. This would be 1 by 0. 1 by 0 is infinity. Infinity square is infinity. So infinity plus something is infinity. So Z will come out to be infinity. Means impedance will be infinity. Impedance means a resistance in LCR is infinity. If resistance in LCR is infinite, what about the current? Current in that case will be 0. Resistance is large, current is 0. Yes. Now see, take a look. If I say frequency is infinite, let's keep this frequency as infinite. So this term will become infinite. 1 by infinite, 0. So infinity square, infinity. Infinity plus something, infinity. So resistance, impedance will come out to be infinity. Again, current will come out to be 0. So when frequency is either 0, okay, or it is infinite, in both the cases, current will be 0 and 0. Current will be 0 and 0. Take a look at this question. In the series LCR circuit, the RMS voltage across the resistor and the inductor are 400 and 700 respectively. RMS voltage. Okay, RMS voltage. See guys, if you make an LCR circuit, okay, everybody knows if I make the phasor diagram of LCR circuit, that is something like this. This will be voltage in case of resistance. This will be voltage in case of inductance. This will be voltage in case of capacitance. Now, guys, what does it say over here? Listen. If I'm saying RMS voltage in case of a resistor is 400, means V, this is given as 400 of volts. And this is RMS, this is not peak. Keep that thing in your mind. In case of inductor, it is given as 700. And what about net? Guys, this is the net voltage. See, this is the peak. E naught over here, this is 500 root 2. This is the peak voltage of net EMF. If you have to find RMS, how do you find RMS? We say ERMS, that is peak divided by root 2. If you remember this. Now, this would be 500 root 2 divided by, I would say, root 2. Root 2 cancel. So, this would come out to be, that is 500. So, RMS voltage came out to be in this particular case, how much? That is 500. Guys, this is voltage across resistance. This is voltage across inductance. And net voltage, net RMS voltage is coming out to be 500 volts. Now, you tell me one thing. Is it possible that the net of 400 and 700 is 500? Is it possible that the net of 400 and 700 is 500? No. So we say net would be 500 only if here it is 300, because we know 400 and 300, their resultant would be 500. So means my dear friends, this 500 will come out to be the net voltage only and only if voltage across capacitance is 400. Now see, 700 minus 400. 700 minus 400, that would be 300. Now this 300 and 400 will have the net of 500. So can I say voltage across the capacitance in this case is 400? Now guys, see, if the equation of the applied voltage is this, then the peak voltage across the capacitor, you got this as RMS voltage across the capacitor, VRMS across the capacitor. Now you have to find the peak. Everybody knows that if you have to find the peak from RMS, multiply it here. So it would be, so peak voltage of capacitor would be VRMS multiplied by root 2. So that is going to be 400 root 2. So which option? Option B. Option B. Okay, lens law is important when it comes to electromagnetic induction. Lens law is important when it comes to the electromagnetic induction. Let's solve some questions from the lens law. Take a look, guys. Take a look, guys. You have a coil over here, and this is the observer. You have another coil over here. This is the magnet. Guys, always remember, this magnet is moving away from the coil. Always remember, whenever a magnet moves away from the coil, this coil will never want that magnet should go away from me. So it will create an imaginary bar magnet inside. If the coil is moving away from the coil, it will create an imaginary bar magnet. And North pole will be from this side, South pole will be from this side. Why? Because then only this North pole will attract the South pole, because unlike poles attract, like poles repel. So what I'm trying to say is that if this is a coil, this is the magnet which is going away. Coil does not want the magnet should go away. So it will try to attract the coil. It will try to attract the magnet. How is that possible? That is possible if it creates imaginary bar magnet. Okay, so here if it is south, it is north, it will create north from this side. Then only it would be attracted. And then if this magnet is moving towards the coil, coil will never want the magnet should come towards the coil. Okay. Coil will never want magnet should come towards the coil. So coil does not want magnet should go away from me. Coil does not want magnet should come towards me. So here it will create an imaginary bar magnet due to which it will repel. So if it is north here, it will create north from this side, south from this side. Then only it will repel. So when it is going away, then it will create unlike pole. When it is coming towards, then it will create the like pole. Then only repulsion is possible. Now see, how do we calculate the direction of the current? Because we know when we say magnetic flux change through the coil, current is flowing. Now see, Now see, we say this, I'll write if the observer sees North Pole, if the observer sees North Pole of the imaginary bar magnet, we say current is anti-clockwise. Okay. And if the, or I'll write it like this. We say if the observer sees South Pole of the imaginary bar magnet, in that case, current is clockwise. Let me show you over here. Guys, take a look. Remember this particular trick. In this case, this observer is directly seeing the South Pole in this coil. If it is seeing South Pole in this coil, current is going to be clockwise. So I say current in the coil A, it is going to be clockwise. This observer is seeing the North Pole directly because South Pole is from the another side. So current in the coil clock B, it is anti-clockwise. Let's suppose if I create the similar question, one more in this one. You have got a coil over here. You have got a coil over here. You have got one more coil over here. Okay. And there is a magnet, something like this. There is a magnet. This is North Pole. This is South Pole. It is moving towards this coil. You are supposed, and observer is here. Let me keep the observer here. You guys are supposed to tell me what is the direction of the current in the first and the second coil. See guys. If this magnet moves away from this coil, it will create an imaginary bar magnet. And South Pole will be here, North Pole will be here. Then only it will attract. And in this one, it will create an imaginary bar magnet in which we say South Pole will be here, North Pole will be here. Now you tell me one thing. Guys, this observer is able to directly see the South Pole here. So current in this is clockwise. Okay. And this observer is directly able to see the North Pole in this one. So here current is anti-clockwise. Because if it is seeing directly this one is from the other side. I hope you got the feel of this question also. I hope you got the feel of this question also. Okay, let's talk about this question. Equation of an alternating current is 10 root 2. You have got an alternating current. I is equal to 10 root 2. Then we say sine of 100 pi t plus pi by 6. Okay. And you guys are supposed to tell me the current taken by the time, the time taken by the current to reach the root mean square value. Guys. We say when it comes to the graph of an alternating current, it is something like this. This is the sinusoidal current. This is the peak value. Root mean square value will be here. So how much time it is going to reach till the root mean square value? How do we find it? First of all, how do we find the RMS value? This we consider always peak. So here peak is 10 root 2. And if you have to find it IRMS, that is going to be peak divided by root 2. Peak divided by root 2. Now guys, if you have to find the time, so put instead of this I, you put I 10 root 2 by root 2. 10 root 2 divided by root 2 is equal to, and this would be over here 10 root 2. Then I say sine of 100 pi t plus pi by 6. Plus pi by 6. Take a look, guys. Take a look, guys. Take a look, guys. So basically, if I say find the time in which it reaches the RMS value, you put the RMS value here. Find the time in which it reaches the peak value, you put the peak value here. Find the time in which it reaches the 50 ampere. You put 50 ampere here. Now see, 10 root 2, 10 root 2. So it is 1 by root 2. When is the value of sine as 1 by root 2? Sine pi by 4, sine 45. So I'll write over here, sine of pi by 4. It will be from this side, sine of 100 pi t plus pi by 6. Sine will go, it will remain. What remains over here? It is pi by 4 will be equals to 100 pi t plus pi by 6. Okay. Now this pi and pi will cancel. Take it over here. So this will be 1 by 4 minus 1 by 6 is equal to, I would say, 100 into t. Now 6 minus 4, that is 2 divided by 24 is equal to 100 t. 2 ones are, 2 12s are. So 12 into 100. So t will come out to be 1 divided by 1200 seconds. So in this much time, it is going to reach the RMS value. In this much time, it is going to reach the RMS value. Like you have this current over here. Take a look. In this case, I'll just follow the first part. Okay. And frequency of AC. This, see, I say I is equal to, guys, in an alternating current, I is represented as 5 sine of 300 pi t plus pi by 6. Find the time in which it reaches the, okay, calculate the peak value of the current. You are supposed to find the peak value of the current. That is 5. RMS value of the current, 5 root 2. Frequency, omega is equal to, we say 300 into t. Omega is, that is 300 in this particular case. Okay. Or when it comes to the frequency, we say omega is equal to 2 pi into f. So omega will be 1 divided by 2 pi. F will be 1 divided by 2 pi into omega. You can find this one. Okay. Okay, this question also. Let's follow this question also. A resistance of 40 ohm is connected to an AC source. Okay. The time taken by the current to change from its maximum value to RMS value. See guys, see guys. See guys. I can say in this particular case, if you take a look over here, you have got, see, you have got an alternating current. If I make the graph of an alternating current, it would be something like this. Guys, this is the zero value, minimum value. This is the peak value. This would be RMS value. So it goes from RMS to peak, then again it comes to RMS. How much time it is going to come from peak to RMS? That is this time to this time. Peak to RMS. Now guys, see. When it comes to the peak value, everybody knows that this current, this is the peak value. How much time the current takes till peak value? T by 4. See. If I make over here. So here current is 0. This is the peak value. Here current is T by 4. Here this would be T by 2. Here time is 3T by 4. Here it is 2T. So basically this is T by 4, T by 4, T by 4, T by 4. So this is, sorry, it is T. So this is one time period. It is one time period. T by 4, T by 4, T by 4, T by 4. That is one time period. So how much time current takes to reach the peak value? That is T by 4. Okay, fourth part of the complete time period. Now guys, in this case it says that, let's suppose here current is 0. Here current is peak. This is I naught. And here it is 0. How much time it is going to take from this to this? How much time is this? So this is T divided by 4. And somewhere here it is peak IRMS. This is IRMS. So this is 0 IRMS and this. Guys, let's suppose if I'm saying from 0 to IRMS it is taking time T1. From IRMS to I naught it is taking time T2. So we have to find the value of T2. How much time it is taking from IRMS to this. Guys, my Telegram channel is t.me/YAWARMANZOOR. Okay. Join this Telegram channel for all the updates, for all the notes. Okay. Okay. Everybody of you. Okay. Everybody of you. Okay, take a look. So guys, in this particular case, we are supposed to find the value of this time. Let's first do one thing. Let's find the value of t1, because from here to here time t by 4 we know. Let's find the value of time t1. Let's find t1. Okay. We say, guys, I would want you guys to focus over here. In this particular case, we say at time, after time t1, how much is the current? IRMS. IRMS means I naught by root 2. So can I say an alternating current? We write something like this. I is equal to I naught sine of omega t. Yes. And I'll write t1 over here. And we have to find the time in which it reaches the IRMS value. So that is going to be I naught divided by root 2, because IRMS we find peak divided by root 2. And here, this is going to be I naught and sine of omega t1. I naught will cancel 1 by root 2. When is the value of sine as 1 by root 2? This is sine of pi by 4, sine 45. And this is going to be sine of omega t. Instead of omega, we can write 2 pi divided by capital T into t1. Now sine, pi. So here it remains 1 divided by 4 is equal to, that is 2 divided by t into t1. So take it over here, So t divided by 8 will come out to be t1. This is t1. Now guys, can I say in this particular case, we can say t1 plus t2 will be equals to the capital T. This t2 plus t1 will be equals not the capital T, t divided by 4. This t by 4. Now what is the value of t1? That is t by 8. This is t by 8. Okay. Plus. Okay, if this is t by 8, plus we say t2 is equal to t divided by 4. Take it over here. So t2 will come out to be t divided by 4 minus t divided by 8. That will be t by 8. And when it comes to the time period, we say time period is inverse of frequency. So I can write 1 upon f into 8. I can write 1 upon. What is the value of frequency? Frequency is given as 50 hertz. So that is 50 into 8. So this is 1 divided by 400 seconds. Answer is 1 by 400 seconds in this case. Okay. Let's solve some questions from semiconductors. Guys, take a look. If the temperature of a semiconductor is increased, its resistance decreases. If the temperature of a semiconductor is increased, its resistance decreases. Guys, when it comes to the temperature, when it comes to the semiconductor, if you increase its temperature, we say electrons shift from valence band to the conduction band. So means when electrons shift from valence band to the conduction band, conduction increases. We say conductivity increases. So resistance decreases. So it is correct. Assertion is correct. The energy gap between the conduction band and the valence band of a semiconductor is small. Yes. When it comes to the band gap, when that is small, then only crossing of electrons is possible. Yes. So A is correct. Conductivity of a semiconductor is increased on doping. When we do doping means adding impurity. If you add impurity, we say in case of the p-type semiconductor, holes are created. In case of n-type semiconductor, electrons are created. So doping means creating the conductivity. So conductivity of a semiconductor increases on doping. Correct. Doping raises the temperature. Doping does not raise the temperature. So option C. For a given applied voltage, conduction in n-type semiconductor is more than that in p-type semiconductor. Yes. When it comes to the n-type semiconductor, we say majority charge carriers are electrons. When it comes to the n-type semiconductor, majority charge carriers are electrons. When it comes to the p-type semiconductor, majority charge carriers are holes. Majority charge carriers are holes. Okay. So we say holes have got the large mass. Electrons have got the less mass. So mobility of holes will be less as compared to the electrons. So assertion is correct, reason is correct. So option A, assertion is correct, reason is correct, and reason is the correct explanation of assertion. The temperature coefficient of resistance is positive for metals, means alpha. You might have seen this formula. R is equal to R naught 1 plus alpha delta T. This is the equation we study in semiconductor, in current electricity. For metals, it is positive. For semiconductors, it is negative. Okay. What does it mean? It means that if alpha is positive, that means on increasing temperature, resistance increases. If alpha is negative, means on increasing the temperature in a material, resistance decreases. The temperature coefficient of a resistance is positive for metals, negative for semiconductors, correct? On raising the temperature in metals, drift velocity decreases, but in semiconductors, more charge carriers are released. We say option A. Which of the following is false? Pure Si doped with trivalent impurity gives a p-type semiconductor. Pure Si, pure silicon doped with a trivalent impurity gives a p-type semiconductor. You have to say which one is false. This is true. This is true. Okay, but this is not our option. We have to check which one is false. This is not our option. In reality, it is true. But according to the option, this is not our option. Majority charge carriers in N-type semiconductor are holes. This is false. This is our option. Minority carriers in a p-type semiconductor are electrons. This is true. Because majority are holes, minorities electrons in case of p-type. So this is not our option. This is not our option. Okay, this is also the resistance of an intrinsic semiconductor decreases with the increase in temperature. True. True. So which option? Option B is the correct option. For forward biasing, positive terminal of the external battery is connected to p-type, correct? When it comes to the forward bias, okay, this is p-type, this is n side, okay, and this is called the forward biasing. Positive terminal is connected to p side, negative terminal is connected to n side. And in case of and negative terminal is connected to the n-type semiconductor. Yes, assertion is correct. This would support the tendency of the majority charge carriers to cross the junction. Yes. So basically it creates the positive charge over here. It creates negative over here, due to which depletion region decreases, due to which electron hole recombination takes place. So assertion is correct, reason is correct. Reason is the correct explanation of assertion. Okay, here, which one of the following diode is forward biased? See. This is P side, this is N side. If P side is connected to the higher, this is higher potential. This is lower potential. This is lower potential. This is, okay. This is, oh, what am I doing, bro? 5 volt is higher. Higher potential, this is lower potential. Guys, N side is connected to higher potential, P side is connected to lower potential, means this is reverse bias. In this case, N side is connected to lower potential, P side is connected to higher potential. This is forward bias. Okay, this is P, this is N, this is P, this is N. So N side is connected to lower potential, P side is, okay. Here -3 is higher because -4 is smaller. Because when it comes to the potential, it's a scalar quantity. So -3 is greater than -4. So N side is connected to the higher potential. So this is forward, this is reverse bias. So here, this P side is connected to the lower potential, N side is connected to the higher potential. This is also reverse bias. Okay. This was the question which was asked in J Mains 2021. 2021. Take a look at this question, guys. an electron moving with a speed v and a photon moving with a speed c has the same de Broglie wavelength, means lambda of electron is same as lambda of photon. How do we write the lambda of electron? H divided by mass into velocity. So de Broglie's wavelength is Planck's constant divided by momentum. And when it comes to the photon, we say energy of a photon is equal to hc divided by lambda. Here, lambda will come out to be hc divided by E. So put it over here. It is going to be hc divided by E. Now, guys, it also says that an electron is moving with a speed v. Can you tell me what is the kinetic energy of electron? That is 1 by 2 mv square. Now take this 2 over here. 2 into kinetic energy. Energy of an electron, let's consider this as E. Okay. Divided by, take this 1v over here. What remains over here? M into v. Because m into v is v square, mv square. And then this is 1 by 2. So this equation, mv. Now put this mv over here. So can you write? If you put this mv, value of mv here, what will you write? H divided by, instead of mv, you will be writing 2e, energy of electron, divided by v is equal to hc divided by energy of photon. This is energy of photon. Okay. H and H will cancel. Take this in the numerator. V divided by 2ee is equal to C divided by energy of photon. We are supposed to find the ratio of kinetic energy of an electron to that of photon. Take it over here. Take this over here. So V divided by 2C will come out to be and kinetic energy of electron divided by kinetic energy of photon. This was also asked in JEE Mains 2021. JEE Mains 2021. A proton, neutron, an electron, and alpha particle have same energy. Their de Broglie's wavelength compares as. See, guys, when it comes to the alpha particle, guys, this is alpha particle. This is proton. First, I'll write neutron. Then we say proton. Then we say electron. Always remember, mass of alpha particle is greater than mass of neutron, is greater than mass of proton, is greater than mass of electron. Okay, is greater than mass of electron, is greater than mass of electron. Just a second, guys. Okay, now guys, we say when it comes to the lambda de Broglie's wavelength, we say de Broglie's wavelength is h upon under root of 2m. Same energy, that is 2m into kinetic energy. So this is that formula of de Broglie's wavelength, which is in terms of kinetic energy. Now it says that kinetic energy of all the particles is same. Planck's constant is same. 2 is same. So can I say this lambda is inversely proportional to root m mass? Means the one which has got more mass will have less de Broglie's wavelength. And we almost consider, we say mass of proton. We say, guys, mass of neutron is almost equal to mass of proton. Slightly, mass of neutron is greater than mass of proton. But you can say mass of neutron is equal to mass of proton. Now tell me, if this has got de Broglie's wavelength, if this has got large mass, so its de Broglie's wavelength will be less than de Broglie's wavelength of neutron. That will be equals to de Broglie's wavelength of proton. That will be less than de Broglie's wavelength of electron. This is going to be the comparison. Option B is matching. This question extremely important. This question. See, guys, and this was also asked in JEE Mains 2021. JEE Mains 2021. In Bohr's atomic model of hydrogen, let kinetic energy let K, P and E are the kinetic energy, potential energy and total energy of the electrons respectively. Okay. Okay. Yes. Choose the correct option when the electron undergoes transition to the higher level. Guys, see. When it comes to the kinetic energy of an electron revolving in the nth orbit, it is ZE square upon 8 pi epsilon naught R. Okay. When it comes to the potential energy, it is minus ZE square upon 4 pi epsilon naught R. When it comes to the total energy, it is minus ZE square upon 8 pi epsilon naught R. Okay. Now you tell me one thing, guys, understand one thing. We say kinetic energy is positive. Okay. It says that if an electron makes the transition from, we say, to a higher level. Let's suppose electron is revolving in the first orbit. It is shifting to the second orbit. What will happen to the kinetic energy? Will it increase, decrease? That's what we are supposed to say. See guys. We say if an electron makes the transition to the higher orbit, we say if R increases, what will happen to the kinetic energy? It is inversely proportional to R. Kinetic energy is going to decrease. Yes, kinetic energy will decrease. Okay, because it is inversely proportional. Now guys, what about the potential energy? We say if R increases. We say what about potential energy? See. If R increases, it is inversely proportional to potential energy. We say potential energy will decrease, but the value of potential energy is negative. So potential energy is decreasing in the negative value. We say potential energy decreases in negative value. Potential energy decreases in negative value. And whenever a scalar quantity is decreasing in negative value, means it is increasing. We say negative minus 5, minus 10. Which one is greater? Minus 5 is greater than minus 10. So that's what I'm saying. Potential energy decrease in the negative value means overall it increases. So if R increases, potential energy increases. Okay. And what about total energy? Total energy is also inversely proportional to R. So means if R increases, we say total energy decreases in negative value. If total energy decreases in negative value, means total energy is actually increasing. So if an electron makes the transition to a higher level, we say kinetic energy decreases, potential and total increases. Kinetic decreases, potential and total increases. The ratio of wavelength of the last line of the Balmer and the last line of the Lyman series, okay? So we say when it comes to the Balmer series, guys, we have to find the wavelength. Wavelength of which line? Last line of Balmer series. Guys, when it comes to the last line, we say last line of Balmer series. What is going to be the final n final? That is 2. When it comes to the Balmer series, it is 2. And n initial is going to be infinity. Because when it comes to the last line means if an electron makes the transition from infinity to the first orbit, that is going to be the last line. Okay, that's going to be the last line. That is going to be the last line. Now guys, see, I can say in this particular case, how do we find the wavelength? 1 by lambda Balmer, it will come out to be, that is R Rydberg constant, 1 by n final square, n final is 2 square, that is 4, minus 1 by n initial square, that is infinity. So 1 divided by lambda Balmer will come out to be R divided by 4. So lambda Balmer will come out to be 4 divided by R. Lambda Balmer will come out to be 4 divided by R. Let's talk about last line of Lyman series. Lyman series. Guys, when it comes to the last line of Lyman series, I would say n final in that case is going to be 1 and initial in that case is going to be infinity, because when an electron makes the transition from infinity to 1, first orbit, that means last line of Lyman series. So 1 divided by lambda Lyman, it will be r upon 1 by n final square, 1 by 1 minus 1 by infinity. Okay. So now lambda L, it is going to be 1 divided by r. Now guys, when I say lambda Balmer divided by lambda Lyman, that will be 4 divided by r upon 1 divided by r. R and r will cancel. It is 4:1. So answer is this. Okay. What else can I show you? Okay, take a look. Let me just tell you the approach only over here. An electron of mass and when accelerated through a potential difference V has a wavelength lambda. Okay. Guys, when it comes to an electron, how do we write its de Broglie's wavelength? It is h upon potential difference is given under root of 2m q delta V. And when it comes to the charge on an electron, it is E delta V. This is the de Broglie's wavelength for an electron. Okay. The de Broglie's wavelength associated with a proton. Let's come talk about proton. Okay. It is going to be h upon under root of 2. Mass of proton is capital M. Because it is saying it is mass is capital M. Charge is same E. Delta V is the potential difference. Okay. Now, in this particular case, wavelength of proton is given. Has wavelength lambda. Okay, wavelength of electron is given. You have to find the wavelength of proton. See guys, in this case, I can say lambda P divided by lambda E. That is going to be h upon under root of 2m E delta V upon h upon under root of 2m E delta V. H will cancel. Okay, it is going to be d delta V, E delta V, 2, 2. So it is going to be 1 by root m, 1 by root of small m upon 1 by root of capital M. It will be a root of capital M divided by small m. So lambda P divided by lambda E came out to be under root of capital M divided by small m. So lambda P will be, take it over here, lambda of electron, that is given as lambda. Okay, so it will be lambda under root of capital M divided by small m. Okay. 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 Let's talk about this question, guys. It says that a mass at rest explodes suddenly into M1 and M2. You have got a mass over here. Okay, this is capital M. It suddenly explodes into two parts, M1 and M2. Okay, let's suppose its velocity is V2, its velocity is V1. We know in case of the explosion, two different objects move in the opposite direction. Okay, so we say these two masses move in the opposite direction. And we know momentum is, and we know momentum remains conserved, and we know momentum remains conserved. We know momentum remains conserved. And what is the momentum over here, sir? This is mv, m1 v1 will be equals to m2 v2. So basically momentum in case of explosion is constant, is conserved. Now it says that find the ratio of the de Broglie's wavelength. Guys, when it comes to the de Broglie's wavelength of this particle by this part, lambda 1 divided by lambda 2, how do we find the de Broglie's wavelength? That is h divided by p, momentum. If momentum is conserved, ph is conserved, so we say lambda 1 divided by lambda 2, it is 1:1. It will be same. It will remain constant only. Okay. Find the maximum angular speed. Find the maximum angular speed of an electron in a hydrogen atom in a stationary orbit. Guys, see. It says this is the hydrogen atom an electron is moving in the first orbit, means n is equal to 1. An electron is moving in the first orbit, that is n is equal to 1. You have to find the value of omega. Guys, see, we say V is equal to R omega. Omega will be equals to V divided by R. Angular speed. And what is the value of velocity? Everybody knows velocity of an orbit, of an electron in the nth orbit, that is V naught Z by n. Radius is R naught n square divided by Z. And you can write it like this also, 2.2 into 10 raised power 6 Z by n. And guys, when it is talking about the hydrogen atom stationary orbit, means the first orbit, for hydrogen Z is 1, for stationary orbit, it is n is 1. So you can say it is 1 by 1, that is 1 divided by 1. And this will be R naught means 0.53 into 10 raised power minus 10, this angstrom, 1 square divided by 1. Now let's find the value of omega, angular speed, that is V. V will come out to be 2.2 into 10 raised power 6 upon, this is 0.53 into 10 raised power minus 10. So this will be over here 4.1 into 10 raised power 16 radian per second. Please do check the calculation from your end. Okay. With this, we'll take a leave, guys, okay? We'll come up with more questions very soon. So for now, okay, take care and bye-bye. Okay? Come on, everybody of you. That's all for today in the today's session, but there will be more questions very soon. Top one, we say, predicted paper, every single thing will be there. Okay, that's all for today, guys. Thank you. See you guys in the next session. Yes, this was the end of the session. Yes. Okay, thank you so much. See you guys in the next session that we will take very soon. Take care and bye-bye. Thank you.